Hello, good morning, ma'am.
Yeah, good morning. Good morning.
1 minute, 1 minute. Yes. Ma'am, now I think you can unmute and talk, ma'am.
Yeah, now I can.
We are not able to hear you, but I'm not able to hear you. Let me check. Oh God, my mic is muted. 1 minute. Yes, ma'am, you can talk now.
So, good morning.
Good morning, ma'am. Actually, there is a slight change in the schedule, and our Vice Chancellor will be joining us at 9:30. So I informed the dean also.
Okay.
So that is why 5 minutes delay.
Okay.
ரெண்டு பேரோட ஆடியோ செக் பண்ணிட்டு ஆடியோ ஓகேயா மேம்?
Okay, no? My voice is audible?
Yes.
உங்களுக்கு
It's audible.
மேம்.
Yeah. Good
ஒரே
morning.
ஒரு சஜஷன் சுதா மேம். கர்டன் பாத்தீங்களா?
கர்டனா?
அத வந்து க்ளோஸ் பண்ணிடுங்க.
க்ளோஸ் பண்ணி தான் இருக்கு. கொஞ்சம் கேப் இருக்கு. ஓகே.
இல்ல. அந்த கேப் ஏன்னா அது உங்களுக்கு பின்பக்கம் கரெக்ட்டா பளிச்சுன்னு தெரியுது.
ஓகே, இப்படி
ஃபேஸ் டார்க்கா தெரியுது.
இவங்க போடு இவங்க.
ஓகேவா?
ஒரே ஒரு நிமிஷம் மேம்.
Is this okay?
ஓகே மேம். I think மேம் உங்களோட கபோர்டு
ஏன் இந்த கபோர்ட் தெரியாமலா?
உங்க Dress எல்லாம் தெரியும் அங்க.
இல்ல க்ளோஸ் பண்ணல இது.
அது இல்ல க்ளோஸ் பண்ணல அது. இல்ல இது வேண்டாம் மேம். நீங்க அப்படியே பண்ணுங்க ஏன்னா இந்த கர்டன் தெரியாது ரைட் ஹேண்ட் சைடு கர்டன். இந்த சைடு நீங்க அந்த கபோர்ட க்ளோஸ் பண்ணிடுங்க.
நான் க்ளோஸ் பண்ணேன்.
இது ஓகேவா சார்?
சூப்பர் மேம். இருங்க ஒரு நிமிஷம் இருங்க ஃபுல் ஃப்ளோர்ல போட்டுறோம்.
Looks like you are sitting in there. Yeah, you are in your room, mama.
மேம் பண்ணுங்க. மேம் நீங்க எஸ்எஸ் மேம் கொஞ்சம் towards on your left hand side. Towards on your left hand side. இன்னும் கொஞ்சம் போதும் மேம்.
கேப் வருது அதுதான்.
பரவாயில்ல இது போதும். மேம் உங்களுக்கு ஓகே?
எந்த ஓகேயா?
ஓகே மேம்.
இது கூட நல்லா புரியுது.
Tell you all is that we have that live YouTube streaming also has started because once we on the Zoom it will be live.
Oh, now we are live.
Yeah.
We are already live actually. Nobody will be watching. That's okay.
But we are on live.
Okay. Anyway, good luck.
All the
Smile while you are speaking. That's the only instruction. Okay. Anything else you need, ping my number so that I will be on the other desk.
Okay. Yeah.
Ma'am, I'm not going to admit everybody now. I'm going to admit only at 9:15 everybody. We will admit only our technical team. Okay, Adira.
Adira is there.
Adira, Neha, Mahesh, Ravi, Jagan. Anitama.
So actually இப்போ வீடியோ டிஸ்ப்ளே பண்ண போறோமா நம்ம டிபார்ட்மென்ட் வீடியோ?
ஐயோ அது நான் லாஸ்ட் பார்க்கவே கிடையாது என்னன்னு சொல்லிட்டு
கவின் வந்து அப்புறம் சொல்லிட்டு இருந்தா இது வெல்கம் ஸ்பீச்சுக்கு அடுத்து ஸ்கோப் ஆஃப் த கான்ஃபரன்ஸ்க்கு முன்னாடி வீடியோ வரும் அப்படின்னு அதை என்கிட்ட கேட்டான் ஸ்கோப்க்கு அப்புறம் போடலாமான்னு இல்லை அதுக்கு முன்னாடி போட்டா பெட்டர் அப்படின்னு சொன்னேன். அதான் அப்படி நம்ம இன்க்ளூட் பண்றதா இருந்தா ஆதிராக்கு சொல்லிடணும் ஏன்னா அவங்க
ஆதிரா are you here? Adira?
Yeah, unmute Adira.
Unmute and then talk.
Yeah.
Yeah. Adira and Neha, right? No. Yeah. In case if I am introducing, I myself will say introduce and then I will give over to Adira. Adira, you don't have to take any tension on that.
Okay, ma'am. So first, have you seen the script, ma'am?
I have not seen. You have sent it to me.
1 second, I'll just forward to you.
No need. I'm not going to check anything. You just do it now because it's already we are live, and so actually I don't think I have to correct. Neha is not in the waiting room also. Oh, what happened?
Yeah, she'll be joining soon, ma'am.
Okay, fine. So I will join. Raveen is there. Yeah, Raveen. I will admit him. Shall we admit everybody at around 9:15? Mahesh is our Mahesh only now. Then ma'am, Praveen Mahesh Satyava.
എനിക്ക് ഇപ്പൊ കണക്ട് ആയി ഇപ്പേ കണക്ട് ആയിട്ടുള്ളൂ. മാം എല്ലാവരോടും അന്വേഷിക്കുന്നുണ്ട്. നേഹ വന്നിട്ടില്ല. ജസ്റ്റ് മെസ്സേജ് വന്നു.
Hello.
Bala, are you sharing the screen? Why don't whoever is in the meeting, why don't you unmute and talk? Lakshmi, are you there?
Yes, ma'am.
Lakshmi, there is a lot of echo in your voice. Just find out, like, are you sitting with somebody else?
No, like almost all of us are together in the scholarship. 1 minute, ma'am.
Lakshmi,
Yes, ma'am.
till the inauguration session, each of you sit in different places because there will be disturbance.
Yeah,
Okay,
please.
ma'am.
Because
Yeah,
when all the systems are on at a time, definitely there will be disturbance.
So all of you sit at different places, or else mute your mic.
One sit in the classroom, one sit in your HD.
Can you check this video which Bala is? Bala, actually we could see now.
All of them can mute their mic. Right now, only Adira is comparing.
Yeah, then Nadira can do speak.
Adira, you can speak now.
Yes, ma'am.
Just say a few words.
Good morning, everyone. Is it fine, ma'am? Sorry, I can't hear you, ma'am. Rukmini, ma'am. Sorry,
Yes,
Sudharani, ma'am.
I just muted now. That's it.
Okay.
Yeah, we are able to hear you. Your voice is clear.
Okay.
My voice is clear.
Yes, ma'am, it's clear.
Yes, ma'am.
Thank you. Thank you.
We'll admit everybody in another 5 minutes, okay? And we have to keep searching whether our YCVC is
Yeah.
entering.
Yeah, we have to
He
keep
is
checking.
entering.
And Madhu said he's trying. He's not able to gain entry.
Now I'm not okay. Fine, I will admit our faculties.
Madhu, Lata, if they are all waiting, you can admit our own faculty.
Yeah, I'll do that.
AJ or somebody.
Trivaratnam sir is not there.
Rukmini, did you speak to KJ sir yesterday? No.
No, ma'am, I forgot.
Now can you call? Just call him.
Ma'am, you can also call, ma'am. Bala, we don't see the this one. We can hear only the voice. Bala, we don't see the video. Bala, can we play from here? Can you tell me where it is? Can we play from here? play பண்ணுங்க. Can you see all this with the music? Can you see the videos with the music?
No, ma'am.
No, ma'am, it's your desktop we are able to see.
We are seeing your desktop, yes. But we can hear the music.
Yeah, music is audible.
Hello, what do we do now?
What happened? It's not starting.
Okay, can you at least this 1 minute?
Play now. Play now.
Huh?
Play now.
Yeah.
Play now.
Shabba. Now can you see this?
Yes.
Yes, yes.
Yes.
Yes, ma'am.
University anthem.
அது எல்லாம் அந்த folder.
Folder குள்ள இருந்தா வரல. Open பண்ணி வச்சாதான் வருது. You have to keep it open or else nothing will come. Where is this one? Where is the university anthem? I have to open it and
முன்னாடி வந்து மல்டிபிள் ஷேர் இருந்ததுனால
இல்ல it is multiple share only. I have to do it right now.
Okay, எல்லாரையும் ஆன் பண்ணி
So join meeting or the Zoom.
ஜாயின் மீட்டிங்ல
So shall we admit everybody now? I will wait for 2 more minutes.
Our faculty, those who are waiting, have you admitted?
Yes, I have done it. Whoever I could see, I saw Asmita ma'am, I saw Lata ma'am. I will check whether Kitlangi sir is here. I think he is not there. Madhu sir, yeah, he's here and I'm admitting him. Jeevratnam sir is not here.
Yeah, Jeevratnam sir, I called. He said,
Yeah.
go ahead, and the pricings are there, he said.
Jeevratnam sir, and who is the other one? Tirunau. He's also not here. I don't see him. So whenever he comes, we'll admit him. So shall we admit everybody? I'll admit and then we will see.
Yes, maybe if VC is coming at 9:30, yeah, we can.
We should be there, no? All participants should be there. Already 97 participants are waiting. Yes.
But there's a problem with my internet connection.
Hello, good morning.
Good morning.
Good morning.
Yes.
Good morning.
Yeah, good morning. So when we are starting?
Our Vice Chancellor will be coming at 9:30. So we'll wait for him and then we will start.
Okay, good. One second, I'll just try to adjust my video.
I have to. Can you?
Dr. Rukmini, Professor Devi Prasad sir has entered.
Yeah, I just saw that, ma'am. I have to make him a post. God, it's moving like 1 minute.
One second, I'll just try to adjust my video.
I have to. Can you? Good morning, sir. Dean, sir.
Sir, you have to unmute, sir. Yes, sir.
Hello.
Morning, sir.
Good morning, sir.
Yeah.
Sir is here.
Yes, sir is here.
Yes, sir. Welcome, sir. Welcome to this conference. Hearty welcome, sir.
Hopefully my net won't fail me today.
Hopefully not. We wish not.
Yes.
I'll try to remind the Vice Chancellor. Yeah, may I know who's sharing the screen? Hello? Hello, sir. 9:30.
Divya, I think.
Divya Sangeeta, can you please stop sharing the screen? We will give you time during your presentation. I welcome all the participants. We are waiting for the chief guest to join us. So we'll start immediately as and when he joins. I request all the participants to mute your mic if possible, mute your cameras also. And please don't share the screen. You will be given enough time before the start of the sessions. Okay. Actually, whoever is sharing the video, I just want to tell them that we are not able to hear the voice. So I will share it from my end, if you're okay with that. Voilà.
Madam, the audio is still not audible. Hello. Rukmini madam.
Hello.
Welcome, sir.
Yeah, good morning. Good morning. I have
Welcome
joined.
to this conference. Thank you, sir. Thank you for your presence. Dr. Rukmini.
Good morning, everybody.
Good morning.
Morning.
Good morning.
Good morning, sir, and hearty welcome, sir, to the program. Adila, are you there? Shall we start the program, sir?
Yes, ma'am.
Actually, your voice is echoing, Adila. If you have multiple equipments, why don't you mute other equipments?
Yeah.
Hello. You know,
Yes,
after the introduction, you can allow me to say a few words because I have got to rush immediately.
Yes,
Yeah.
yes, sir. Thank you so much, sir. I'll be
Thank you.
Yes. Adila, can you start the program?
Yes, Adila.
Yeah. A very good morning to all.
Good morning.
I'm extremely glad and privileged to welcome all our dignitaries, distinguished speakers.
Okay, if there is a problem there, then we can start the program. I welcome all of you for this two-day international program on drug discovery and translational research. We'll start the program with the university anthem.
புதுமை புதுமை புதுமையே போற்றுவோம் புதுவை பல்கலைக்கழகமே உன்னை வாழ்த்துவோம் வங்க கடலனை தாராட்டும் ஞான சூரியன் உதிக்கும் இடம் இது எங்கள் புதுநெறி காட்டும் உலகம் எந்நாளும் அறிவு தேடல் தொடரும் அறிவற்றை பொழியும் ஆசிரியர் அது தினமும் நனையும் மாணவர்கள் புதுவை பல்கலைக்கழகம் அறிவினாதம் தண்ணீர் சோலை அகில போற்றும் கல்வி சாலை மனித பலமே என்று
Hello everyone.
Yeah, go ahead.
Now I would like to request our extremely dynamic and energetic head in charge of the department and the coordinator of this international conference, Dr. R. Rukmini, to welcome the virtual gathering.
Thank you, Adira.
Thank
Good
you.
morning, everybody. I extend a hearty welcome to all of you for this two-day international virtual conference on drug discovery and translational research. Eminent scientists from different countries are participating in this 2 days event and will be enriching our knowledge in the field of drug designing by their rich research experiences. First of all, I extend a hearty welcome to our Honorable Vice Chancellor, who has been the pillar of support for all the good initiatives of the university. He is the guiding force behind every program that involves knowledge sharing and intellectual development. We are indebted to you, sir, for your continuous support to our department. We are extremely thankful to you for being with us today amidst your busy schedule, and I extend a hearty welcome to you, sir. It's my pleasure to welcome our respected Dean, School of Life Sciences, Professor K.V. Devi Prasad, who handholds us in every new initiatives and enriches our path with his vast knowledge and wisdom. He has helped our school to achieve the goals on time. We are elated by your presence here, sir, and I extend a warm welcome to you. It gives me immense pleasure to welcome Dr. Ram Mani, faculty from University of Texas Medical Center, who is an expert scientist in the field of cancer and human molecular genetics. He has done excellent research in the field of epigenetic events associated with cancer development. We are extremely thankful for being with us, especially in this odd hours of your time, and I extend a hearty welcome to you, sir. I'm happy to welcome Dr. Vinod Skaria, Senior Scientist from IGIB, New Delhi. He is a clinician and computational biologist and has been involved in creating novel methods for gene analysis and understanding genome variation. He is a part of various pioneering projects in genome analysis. We are honored by your presence here, sir, and I extend a warm welcome to you. I wish to place on record my hearty welcome to various speakers of the events, Dr. Tabriz Siddiqui from University of Manitoba, Canada, an eminent researcher in the field of neuronal network, Dr. Vijayendran Chandran from School of Medicine, University of Florida, an expert researcher in the field of neuronal disorders, Dr. Malayalam Mariappan from Yale School of Medicine, an excellent scientist in the field of cell biology. I also extend a wholehearted welcome to all the dignitaries, scientists, faculty, research staffs, students from our university and other institutes. I thank the participants for their overwhelming support. There are about 500 participants from various institutes across India. I'm sure that these sessions will enrich our knowledge and expand our horizons of research. Once again, I welcome all of you and wish you all enlightening and enjoyable sessions ahead. Thank you. And before going to the next event, I would like to take this opportunity to play a small department video made by our own PhD students to bring in the rich history of our department. So I thank our research scholar, Mr. Praveen, Mr. Mahesh, Ms. Satya, Ms. Lakshmi, Mr. Arun, and Mr. Jagan for their efforts in making this video. Thank you very much. Now I kindly request our Honorable Vice Chancellor to deliver the inaugural address.
Thank you, Dr. Rukmani. My heartiest congratulations to everybody for organizing this very useful conference at a time when this is most needed, in my opinion. All those who are present, all the respected guests from all over the world, and our Dean, Dr. Professor Devi Prasad, Dr. Rukmani, and all others who have taken the lead in the department to organize this conference. I'm quite aware of the fact that organizing a conference, even if it is virtual, is not a very easy thing, and they have put in a grand effort. I must give my heartiest congratulations to all of them. This conference on drug discovery and translational research is a very important one, particularly at a time when we are all facing this very unfortunate pandemic. And all over the world, the whole scientific community is in a kind of depression. And but I must say that they have really taken up this challenge in a big way, and they have come out with certain protocols which are helping the mankind overcome this. Now, when we come down to drug discovery, it's an important old science. People are working on it. Let me tell you what way it has undergone a grand transition. From a stage when we were just administering the drugs, whatever way it was coming, wherever one needed 5 mg, we were giving 100 mgs, 500 mgs at a time, with the hope that it will reach the affected area. And though it will sound a little strange that you will say, how does it matter if it is 100 mg instead of 5 mg? If one calculates the drug which is being administered all over the world and the number of people who are taking, the way population is growing, if somebody calculates, you will find what a staggering amount of drug is being wasted all over. And not only that, the kind of overdose which is being given, in which, in my opinion, is not needed at all. So from that stage to a stage of nanoparticles discovery, and then gradually encapsulating the drugs in this, and then slowly again making a kind of a survey that what exact amount would be required, went on. And then from there we came over to nanobiomedicines, and the whole domain changed over to the nano domain. And we also know, which I'm sure you would have, many of you would have read, that the moment anything reaches nano domains, the set of properties also change. Now, this is where our biochemists and microbiologists and everybody have done a remarkable work, that from nanomedicines, they encapsulated drugs in nanoparticles, and we reached a scenario which is what is referred to as targeted drug delivery. We encapsulate in a nanoparticle and then inject. The drug goes straight away to the affected area, and here the amount that is needed is what is being administered. So extra drug is not being given. There is a saving. There is so-called, as we are all conscious of this, that there are many side effects. So all those are avoided with this approach of targeted drug delivery and going over to the nano domain by following an approach which is what is called the translational research, of course, but then giving the right dose for the right area, right affected area. Now that probably would not have been possible had it not been for the excellent research work being done by our wonderful biochemists and microbiologists all over the world. And this has led to a very fast discoveries and a quantum jump in the research in these areas. And what has it been done? How this has been done, at what rate is being done, what is the success rate. All this is before us, the way our vaccines have been developed. People thought India will never be able to come out with a vaccine and we'll have to depend upon all others. But then we have all known the way our researchers at ICMR and other agencies have come forward and the way they have done this, we have not only come out with 2 vaccines, the third is also around the corner. So we have shown the world that we are capable. Not only that, we are concerned about humanity. And that is what has led to India sending these drugs, the vaccines, to many neighboring countries just to help come out of this. And once the whole humanity comes out, I think the world will be safer. And that is what the approach of India has always been, to make humankind safer, to have a peaceful coexistence, to have a peaceful world. That is what our approach has been, not only here, in all the world bodies, particularly UNO. India has always followed that approach. And with that approach, I think this will also be a step in that direction. As I said, what has been done, how this has been done, what is the success rate, all these one can come to know only, I'm underlining, I'm emphasizing, only when there are conferences of this kind, where scientists from all over the world would present their work for, I would say particularly for the benefit of young researchers all over, and they will be able to take a lead that what area to go in for, what direction to give to our research. And once they come to know, probably they'll be able to do much better work. So I compliment my department that they have taken a lead at a time when it was most needed. So my heartiest compliments. And I wish all the researchers who are participating, I was told there are more than 500 who are participating. So I would urge all of them to interact effectively. And this is a message for the youngsters, to talk to them, interact very effectively, so that our research goes further, many folds, and our success rate becomes higher, and you achieve your goals faster. So I compliment and wish a very happy interactive session in the 2 days to come for everybody, including the teachers, researchers, and students in particular. All the very best, and God bless all of you. And I wish the conference a grand success. Thank you so very much.
Thank you so much, sir, for the extremely thoughtful and motivating words. We are profoundly grateful for you to spare the precious time amidst your busy schedule. Thank you once again, sir. And now I would like to welcome Dr. C. Sobalakshmi, the organizing chairman of the conference, to highlight the audience on the scope of the conference and how the young researchers and the student community will be benefited from this 2 days event. Please welcome, ma'am.
Yeah. Thank you, Athena. A very blissful good morning to everyone. I welcome you all to the International Conference on Drug Discovery and Translational Research 2021, called as DDTR 21. Every year, for celebrating National Science Day to commemorate the discovery of the Raman effect by Sir C.V. Raman, the Department of Biochemistry and Molecular Biology conducts a scientific event. The theme for this year, National Science Day, as given by the Government of India, is Future of Science, Technology and Innovation, Impact on Education, Skills and Work. In line with the theme, we are organizing the International Conference on Drug Discovery and Translational Research. We are having eminent speakers of Indian origin who are doing commendable work across the globe advancing science and technology. The sudden outbreak of SARS-CoV-2 has taught us the significance of drug discovery and translational research. The purpose of this conference is to create a platform for the young scientists and students from various institutes to gain knowledge about the recent trends in drug discovery process. The deliberations cover topics to understand how scientific research aims to overcome the drawbacks of conventional therapy and develop new molecules as well as diagnostic tools. The talks by the invited speakers will address the specific areas like cancer progression and diagnostics, diabetes, COVID-19 screening procedures and sequencing, epigenetic regulation, etc. The event also sets stage for the scholars to present their research findings to groom themselves as young scientists. We strongly hope that all the participants would get vivid exposure, knowledge, and career advice from this august gathering. So we greatly welcome and acknowledge the participation of around 500 attendees. On the first day of the conference, 2 invited talks followed by oral presentations along with e-poster sessions are scheduled. On the second day, there are 3 invited talks and continuation of oral and e-poster sessions accompanied with graphical abstract and photography contest will be organized. So let us gear up to celebrate, discuss, and experience the spirit of science with our renowned speakers and enthusiastic participants. Thank you all and have a spectacular day ahead.
Thank you so much, ma'am, for giving such a brief idea about how this conference will be helping us to upsurge our knowledge and knowledge in the recent research and development in the field of neuroscience, cancer biology, its translational and epigenetic regulation, and the most celebrated topic of the century in every field, COVID-19, from the exclusive work of our eminent speakers. Now, I would like to request our much respected patron, the Dean of School of Life Sciences, Professor K.V. Devi Prasad, to deliver the special address. Please welcome, sir.
Sir, I think the mic is muted, sir. Your mic is muted.
Now I'm online, I guess.
Yes, yes, sir, you're on.
Good.
Please, sir.
Thank you. Thank you. It's my good morning, everybody. Good morning, Professor Singh, who has just had to leave us because he had to. The Prime Minister is visiting Pondicherry today, and so I'm sure Professor Singh is extremely busy with that. Will be busy with that very soon. And it's my pleasure to be here in this department's international online meet on a very important topic on drug discovery and translational. There was a time when scientists used to look down upon translational research, but that has been in the past for quite some time. Now, translation research has become the need of the hour because the problems humanity faces are so diverse that we need to start looking for specific solutions, even though that should not be actually the real basis for doing science. But it has become necessary, and necessity forces us to adapt our thinking process, and that's why we have come to recognize translational research as a very important aspect, and drug discovery is the natural consequence of translational research industry. And I'm extremely glad that the department is doing this effort as a part of its Science Day celebration. Nothing can be more apt than this. And as the Vice Chancellor pointed out, it has become extremely important to do this kind of research to find solutions for many diseases. And of course, diseases that are high mortality attract a lot of funding, but there are many diseases which are high morbidity but low mortality, which also require drugs to cure them, and they are not very popular, so to say. I'm sure people will start looking at those also. And of course, as an environmentalist, my concern is that we also take care of where the drug ends up in the environment, how to make sure the drugs are made innocuous by the time they get the environment. That's also an issue. I'm sure that's also will be worth looking into by biochemists for the long-term stability of our ecosystem. I'm extremely glad that Department of Biochemistry and Molecular Biology is organizing this, and I wish the 2 days workshops all the success. Let me not be standing in the way of extremely interesting talks that are coming up now. So all the best again. Thank you so much for having me. Thank you very much.
Thank you so much, sir, for your generous presence and wise words. Now, may I request Dr. S. Sudharani, the co-chair of the conference, to propose the vote of thanks.
Thank you, Adira. Honorable Vice Chancellor, Professor Gurmeet Singh, respected Dean, School of Life Sciences, Professor K.V. Devi Prasad, distinguished speakers of this conference, faculty members, young researchers, and student participants. I deem it a great honor to propose the vote of thanks to all the dignitaries who have made it convenient to attend the inaugural session of this international conference, DDTR 2021. On behalf of the organizing committee, I express my
Dr. SS, your mic is muted automatically, I guess, somehow from the middle. Yeah.
Sorry. On behalf of the organizing committee, I express my sincere thanks to our respected Vice Chancellor, Professor Gurmeet Singh, who has been constantly extending his support and encouragement to our department. Thank you very much, sir, for gracing this occasion with your presence and delivering the presidential address. We are extremely grateful to you, sir, for your motivating address. I must express my sincere, deep sense of gratitude to our respected Dean, School of Life Sciences, Professor Devi Prasad, sir, for his unstinted support to our department and for his insightful address delivered today. Thank you very much, sir. I would like to express my sincere thanks to all the invited speakers of this conference who have readily accepted to our invitation. I thank all the speakers who have come forward to share their research findings and knowledge gained through their work with the participants and enlighten them during this conference. It is my duty to express my sincere thanks to all the participants comprising of faculty members, young researchers, students of UG/PG programs from various universities, research institutes from within and outside the country. The success of any conference is decided by the enthusiastic and interactive participants. And hence, I thank each and every participant of this conference, DDTR 2021. Thank you all. I must express my sincere thanks to all my faculty colleagues of our department, office staff, PhD students for their untiring efforts and hard work put in for this conference. Thank you all. I must also thank the Pondicherry University administration for their support. Thank you all.
Thank you so much, ma'am, for your wise words. And I thank everyone on my behalf. Our inaugural session is over now. Now we will be starting our technical session. So I gladly welcome Satyapriya and Lakshmi to take over the session. Thank you, guys.
Satyapriya, are you there? Okay, so if there is a technical problem, we will start the scientific session. And I welcome Dr. Ramani. Thank you very much, sir, for being with us today. And I request Dr. C. Subbalakshmi to introduce our honorable speaker.
Yeah. I'm very glad to introduce the inaugural speaker, Dr. Ram S. Mani, who is an assistant professor with joint appointment in the departments of pathology and urology at University of Texas, Southwestern Medical Center, Dallas, USA. He is also a member of the Harold Simons Comprehensive Cancer Center. He did his PhD at JNCSR Bangalore in the field of human molecular genetics. He pursued postdoctoral studies at the University of Michigan, investigating on prostate cancer genomics and epigenetics. Later, he also worked as a research investigator over there, and progressively, he established his independent research group at UT Southwestern Dallas. So he's a dynamic scientist in the field of cancer, and he also contributed a lot in the discovery of mechanisms underlying the formation of ETS gene fusions in prostate cancer. And his lab is mostly focusing on different epigenetic mechanisms, transcription regulation, and genomic rearrangements associated with cancer. And to his credits, he has obtained several awards and honors like NIH Pathway to Independence Award, American Cancer Society New Investigator Award in Cancer Research, and the Harold Simons Comprehensive Cancer Center Impact Award, Career Enhancement Program Award, and last year he received Breakthrough Award from the Department of Defense. And he is also part of many scientific review committees in American Association for Cancer Research and NIH. And his growing number of publications include several high-impact journal articles. Notably, he has 2 articles in the journal Science, which is supposedly a dream journal for all the researchers. And also he has, very good number of publications in Nature Medicine, Cancer Cell, Cancer, Cell Reports, etc. So today, he will be sharing with us his work regarding 3D genome architecture and transcriptional regulation in prostate cancer. So now I welcome Dr. Ram S. Mani to deliver the inaugural talk. Ram, unmute yourself.
Hi, Subbu. Can I share my screen?
Yes, yes, sir.
So thank you very much, Subbu, for the kind introduction. I wish to thank you and all the organizers for this exciting meeting and the invitation to present our work. So today I will be talking about 3D genome architecture and transcriptional regulation in prostate cancer. This is a relatively new area. It's a new research area, and it's also very new for us. I have not done this work much of 3D genome in my postdoc, so this is a new direction I started in my laboratory in UT Southwestern. So try to move the okay. Okay, yeah. So let me start by giving an introduction to prostate cancer, and much of this talk is about prostate cancer. So it is the second leading cause of cancer death in men, next only to lung cancer. There are about 240,000 new prostate cancer cases every year, and 30,000 men in the US die of prostate cancer each year. There are multiple causes for prostate cancer, some being inflammation of the prostate. Genetic ancestry is a risk factor. African American men have prostate cancer at higher incidence, and there is some amount of genetics, but aging is the single most significant risk factor for prostate cancer. More men die with than of prostate cancer. So determining the patient population at risk of dying from prostate cancer is a very important aspect of prostate cancer research. Let me elaborate this by saying that only 1 in 10,000 men under the age of 40 will be diagnosed with prostate cancer. This rate shoots up to 1 in 39 for ages 40 to 59. And 1 in 14 men aged 60 to 69 will be diagnosed of prostate cancer, and more than 65% of all prostate cancers are diagnosed in men over the age of 65. It is a disease that is strongly associated with aging. given that more men die with than because of prostate cancer, there is a lot of variability in disease progression. There is a critical need for the identification of biomarkers that distinguish between indolent or non-aggressive forms versus aggressive forms of prostate cancer. Now, let me quickly walk you through the various stages of prostate cancer development. PIA, or proliferative inflammatory atrophy, it's believed to be a precursor lesion for prostate cancer development. You see it towards the left. Can you all see my arrow pointer?
Not yet, so. Oh, yes, now we can see.
Okay, you can see the pointer moving, right?
Yes.
Okay, excellent. So PIA is believed to be the precursor lesion for prostate cancer, and it is suggested that that PIA gives rise to PIN, or prostatic intraepithelial neoplasia, which could be either low-grade PIN, and low-grade PIN can transition into a high-grade PIN. And this could also, many high-grade PINs can eventually transition into invasive carcinoma. While prostate cancer by itself does not cause lethality, it's the metastasis that contributes to cancer-associated death. So many men who are presented with prostate cancer, if they are treated early with radiation or surgery, it's kind of curative. But at the time of diagnosis, if it's already spread, it's metastatic, then the outcomes are not as good. So what's unique about prostate cancer? It's androgen signaling. Androgen signaling is a distinguishing feature of prostate cancer development. Charles Huggins received the 1966 Nobel Prize in Physiology or Medicine for his discoveries concerning hormonal treatment of prostate cancer. So it's been known for a while that androgen signaling is causally associated with prostate cancer. So what's androgen signaling? Briefly, androgen signaling involves testosterone, which is the male hormone, which gets converted by the enzyme 5-alpha reductase into DHT, or dihydrotestosterone. And DHT binds to AR, or androgen receptor, and there is a dimerization. The AR dimer, there is a lot of cofactor exchange, and the AR dimer eventually enters the nucleus from the cytoplasm. And upon entering the nucleus, AR binds to specific DNA elements called AREs, or androgen response elements. It does not bind to all AREs because there is some amount of chromatin remodeling. So it is known that most of the AR binding sites have another factor that binds, which is FOXA1. So there is a cooperative binding of AR and FOXA1. So FOXA1 is a pioneering factor, transcription factor, that binds and opens the chromatin. And those FOXA1 binding sites, which also have an AR binding site alongside, in those sites, AR will come and bind. And AR recruits a lot of coactivators, most notably P300. And these would lead to enhancer activation. These binds to, the point is, AREs are spread all across the genome, and these are not necessarily located in gene promoters or transcription start site. AREs can be located anywhere, anywhere in the genome. Most AREs are distal from transcription start site, and these are and most of these AR binding sites are outside coding regions, so AR binds to the non-coding region. But interestingly, most of these binding sites have this mark called acetylation of histone H3 at lysine 27, and these marks are basically enhancers. So AR binding leads to activation of enhancers, and these enhancers control transcription by long-range chromatin interactions. An enhancer can be right next to the gene, next to the transcription start site. It could be 10 kb from the transcription start site. It could be 100 kb from the transcription start site. It could be anywhere. And a given enhancer can regulate one gene or many genes. A given gene can be regulated by multiple enhancer, and an enhancer does not necessarily regulate the nearest gene. It could regulate a distal gene. So it's a big puzzle in the field. And it's the androgen signaling that drives prostate carcinogenesis, and there's a reprogramming that happens. So androgen receptor is important for the normal growth and development of the prostate gland, but in the presence of certain accompanying mutations, AR reprograms the prostate epithelial cells, leading to dedifferentiation program, and there is initiation of prostate cancer. So it is a hallmark of transcriptional dysregulation. Well, as I said, it's been known for many decades that androgen signaling is a key factor. So while men with metastatic prostate cancer initially respond to androgen deprivation therapy, nearly everyone develops resistance. And once this resistance develops, there is no good treatment. And the outcomes are very, very bad. So understanding androgen receptor signaling gives us new opportunities to find treatments and cures for prostate cancer. So one of the big puzzles in the field is, what does the androgen receptor do? When it binds to a distal region, what are the genes it regulates? And what is the cistrome? Cistrome means the combination of cis-acting elements, and that is not really known. Methods like ChIP-seq have identified binding sites. We still don't know the targets of AR. Some targets we know, but most targets are unknown. So these were some of the questions that we addressed in our laboratory. So in my talk, I'm going to cover 4 aspects. I will start with giving a brief outline of 3D genome architecture and transcription control. I will then focus on regulation of the regulators. I've already given an intro to AR and FOXA1. I'm going to talk about how these regions, AR and FOXA1 genomic regions, are regulated. This will be followed by a discussion on transcriptional regulation by AR and FOXA1. So the AR protein and the FOXA1 protein, how these 2 proteins combinatorially regulate transcription and drive prostate tumorigenesis. I will also talk about transcriptional dysregulation by PCA or prostate cancer germline risk alleles. It's the most genetic cancer of all cancers known. So ancestry is a strong risk factor, and there are about 180 risk alleles which have been identified by GWAS or genome-wide association studies. And if men have these risk alleles, they are at a much higher likelihood of getting prostate cancer than those men without the risk alleles. So let's begin with 3D genome architecture and transcription control. So as I've alluded before, the genome is, the linear genome is folded. Just the way proteins are folded, even the genome is folded. And the sequence by itself does not totally dictate the folding. It's the availability of transacting factors like transcription factors and other proteins like CTCF, which dictate the folding of the genome. And methods like Hi-C have identified regions like TADs, or topologically associating domains. In the top panel, you could see these triangles. These triangles are generally referred to as TADs, or topologically associating domains. And what these topologically associate, the key feature of TADs is that there is more interaction within a TAD than between TADs. So if you can see these triangles, the region within these triangles are interacting, but a region within a triangle will not, or is less likely to interact with the region in a different triangle. So TADs represent constraints within which chromatin interactions happen. A given TAD can have, if you look at the bottom panel, a given TAD can have a bunch of genes which are active in tissues in a tissue-specific manner, or a TAD can have heterochromatin or silent genes, and so on and so forth. Some TADs are really big, and it could have sub-TADs where you could have a few active TADs and inactive TADs, sub-TADs, or which are also called chromatin contact domains. And generally, these high-C determined TADs are about 1 MB in size. So much of this knowledge has come by the parallel development of methods. The most important method which led to the to increase in our knowledge on the genome architecture is a method called chromosome conformation capture, which was developed about 20 years back by Nancy Kleckner and Job Decker. And this method basically is a proximity ligation based method to look at the frequency of interaction between different loci in a chromosome or in different chromosomes. And a genome scale integration of chromosome conformation capture, or 3C, is called high-C. High-C is a method by which you could tell the frequency of contacts within any two region in the genome. Another parallel development is the CHIP methodology, or chromatin immunoprecipitation. And if you do a genome scale integration of CHIP, you get a CHIP-seq. You can also do fancy integration of CHIP and high-C. So if you if you start with a chromosome conformation capture and then follow it with a CHIP, it's called a high CHIP or a plaque-seq. You could do a CHIP, and then you could do a proximity ligation of the CHIP products, and you could get and then the method is called chia-PET, or a chromatin interaction analysis by paired and tag sequencing. So today I'll be talking most of my talk will be on chia-PET, or chromatin interaction analysis by paired and tag sequencing. So what do these methods tell us? These methods tell us how the genome is folded. And I would briefly give you an overview of these methods. In this case, chia-PET. So you take cells and cross-link cells, and when you cross-link the cells, the genes or the DNA and the proteins are all locked together. And sometimes a distal two DNA sequences which are far apart in the linear genome, if they come together, they would also get locked together. The cross-linked cells can be lysed, sonicated, and then you could chip purify your protein of interest. This is followed by proximity ligation. So in addition to regions which are close together, if a distal region comes if a distal region comes to near a gene or a region of interest, that also gets captured, and we follow it by a proximity ligation. And then we basically capture the biotin beads and then make a library and sequence the library, high throughput sequencing. We do hundreds of millions of reads capture, and then do computational analysis and develop new methods, and also use pre-existing methods, statistical analyses, to really understand how the genome folds. By using this kind of data, you can answer a lot of questions. For example, how are enhancers and promoters organized in the PCA, or prostate cancer 3D genome? What is the fundamental relationship between RNA polymerase associated chromatin interactions and the higher order genome architecture defined by cohesins, CTCF, or the TADs? How do master transcription factors like AR and FOXA1 regulate their own expression? And how does the binding of these factors to distant regulatory elements control polymerase activity in gene promoters or transcription start site, and govern transcription? And how do germline risk alleles, if someone has a cancer predisposing allele, how do these alleles contribute to cancer development? Does this impact the genome folding, and does it impact transcription regulation? So these are the kind of questions that you can answer by the kind of methods that I'm talking about. So we did the chia-PET pipeline. to understand the interactome of Hol2, RNA polymerase 2. So we sought to identify the global differences in transcription regulation by comparing RNA Hol2 ChIA-PET, along with we integrated ChIA-PET data with ChIP-seq, RNA-seq, and a lot of other data sets, and compared the genome folding of benign or normal looking prostate cells, RWP1 cells, and PCA cells, AR positive PCA cells like LNCAP, AR amplified PCA cells like VCAP, and also androgen independent metastatic cancer cells represented by DU145 cells. So what did we discover? We discovered complex networks of chromatin interactions and transcriptional hubs. We were surprised to see that in addition to enhancer-promoter interactions, we also saw promoter-promoter interactions. So by partitioning the genomes into promoters, gene bodies, and other regions, we saw thousands of interactions. And in addition to discovering interactions between enhancers and promoters, we also discovered enhancer-enhancer as well as promoter-promoter chromatin interactions. So there are thousands of interactions, and these are in the form of transcriptional hubs. So where does this lead us to? So we were able to pair all the enhancers to their target genes, at least the enhancers which were active, which were mappable in these cells. We were able to map them to their target genes, and likewise we could those genes which were expressed, those promoters, we could see what are the enhancers which these promoters were interacting with. So we saw that in general, in comparison to the benign cells, the cancer cells displayed an overall increase in enhancer usage per gene, as well as an accompanying increase in genes per enhancer. So there is definitely cancer development is associated with change in the 3D genome organization. In this case, we see definitely a change in the enhancer promoter connectome, enhancer connections. These connections are all changed. So when we got all this data, there were not reliable statistical approaches to really identify statistically significant gains in enhancer promoter contacts or losses in enhancer promoter contacts. So we basically developed a new model for the statistical comparison of chromatin interactions. And we noticed that the interaction strength between anchors are basically binding sites which are connected, and a binding site is a peak. Those peaks that interact with other peaks are also called anchor peaks. And we saw that the interaction strength between anchors followed a negative binomial distribution. So we developed a new model, which is called DNB, or difference of negative binomial distributions, to compare two different negative binomial distributions. By applying this model, we identified statistically significant gain and loss of chromatin interactions across all pairwise comparisons in the cell lines that we analyzed. So indeed, we were able to identify thousands of significantly gained interactions as well as lost interactions. And this kind of gave us a good pointer into understanding what are the interactions, how these interactions influence transcription, and how the downstream pathways are altered. So, so we saw that there is some kind of a correlation between enhancers per gene and transcript abundance. However, this correlation was not too strong. Rather, the more significant correlation was RNA Pol2 chromatin interaction strength per gene and transcript abundance. So we indeed observed that the RNA Pol2 interaction strength chromatin interaction strength in a promoter is way more better predictor of gene expression than the number of enhancers that a gene has. So it's the chromatin interactions that determine determine transcription. So the next question is to understand the relationship between changes in chromatin interaction and changes in gene expression. So what we did was we took all genes which were upregulated or downregulated and did the comparison. And if you see in the top left panel, this is all genes which are upregulated or downregulated. Then from that, we took the top 50% upregulated and downregulated, 40%, 30%, 20%, 10%, and so on and so forth. Remarkably, what we see is there is a correlation between change in chromatin interactions and change in transcription. So we see that gain of expression of a gene is associated with increase in chromatin interactions, and likewise loss of expression of a gene is associated with the loss of chromatin interactions. And as you go up the scale from all genes that are differentially regulated twofold or more to the top downregulated or the top upregulated genes, you indeed see the correlation constantly increases in all the comparisons. So in the top, you see the VCAP cells versus RWP1, LinCap, or RDU145. Any cancer cell you take with a normal cell or any two cell types, there is a strong correlation between changes in gene expression and changes in promoter-associated chromatin interactions. So we think that changes in RNA Pol2-associated chromatin interactions are likely to be drivers of gene expression changes. And this is particularly the case for the most dysregulated gene. So if a gene is really highly expressed in a cell type, it's almost always it's because of an enhancer or a chromatin interaction. So at this point, we have addressed how enhancers and promoters are organized in the 3D genome. So the next question is, why does an enhancer pair with a particular gene? Why does it pair with one promoter? Why not any promoter? So is there a framework? So that's what we plan to unravel in the subsequent analyses. So this is a global snapshot of I won't say it's a global snapshot. It's a snapshot of a chromosomal region, and we show integrative analysis of RNA Pol2 associated chromatin interactions. You see a lot of tracks here. So in the top, you could see DNA-seq, which is the equivalent of an ATAC-seq, which tells open chromatin. Then you have H3K4 trimethylation, which marks gene promoters, CTCF, which marks insulators, K27 acetylation, which is an enhancer mark, RNA-seq, which is a transcription mark, and these gray bars are TADs, topologically associating domains. And this is the RNA Pol2 chia-pet data, RAD21 chia-pet data. And RAD21 is a cohesin component. I will come to it later. What you see is this area is devoid of transcription. There is no RNA-seq signal. So this is a heterochromatin region. So you don't see Pol2 associated chromatin interactions. But in this region, this is euchromatin. There is a lot of transcription. You see a Pol2 associated chromatin interactions. And in general, there is more Pol2 associated chromatin interactions and RAD21 associated chromatin interactions in euchromatin regions or regions of active transcription, but not so much in heterochromatin or regions of inactive chromatin. And again, high C tells you these TADs or these triangles. So there's a lot of interaction within this triangle, but not so much between. Well, you do see some interaction, but much of it is within these triangles or TADs. We do see that indeed, RNA Pol2 peaks overlap with H3K27 acetylation peaks. So there is a strong overlap between Pol2 peaks and Pol2 interaction peaks with enhancers, but we did not really see an overlap with CTCF or insulator elements. Likewise, RAD21, which is a cohesin component, the RAD21 peaks or interaction anchor peaks overlapped with CTCF peaks, but not with Pol2 or K27 acetylation peaks. So this is a quantitative analysis, and we see that indeed RAD21 or cohesin associated chromatin interactions were significantly longer than Pol2 associated chromatin interactions. So blue is RAD21, these are long range interactions, and Pol2 are the short range interactions. So we immediately, based on our data and the emerging scientific literature, we hypothesized that indeed these are the RAD21 is the outer boundaries within which Pol2 associated chromatin interactions happen. So we hypothesize that indeed these are outer boundaries which provide insulation for relatively short range RNA Pol2 associated chromatin interactions. So what does this mean in terms of gene regulation? And that can be exemplified by the KRT8-18 gene cluster. Now, these are two genes, KRT8 and KRT18. These two genes are highly expressed in the prostate luminal cell types. And we see that these two genes have a promoter, and their promoter also has an accompanying, there's an enhancer nearby. And these two genes are highly connected by Pol2. So there is RNA Pol2 associated chromatin interactions between KRT8 and KRT18. And this interaction happens within the framework of an outer interaction, which is cohesin associated or RAD21 associated, these outer chromatin interactions. So you can see two tiers. The inner short range RNA Pol2 associated chromatin interactions are flanked by the outer long range RAD21 associated chromatin interactions, and these sites are marked by CTCF. So what does this mean? This basically means that these RAD21 are actually the boundaries. These boundaries prevent these enhancers from activating genes outside the boundaries. So this is a keratin gene cluster. You have keratin 8, 18, you see keratin 78 and 79, but these two genes are not expressed in luminal cells. So it was not known why keratin 78, which is right next to keratin 8, is not expressed, but this keratin 18 is expressed. And the reason is because these two are flanked by RAD21 associated chromatin interactions. Now, why is there a RAD21 associated chromatin interaction? Because there are CTCF binding sites. So CTCF binding sites create RAD21 associated chromatin interactions. And within these genes, near these genes, there are enhancers, which are presumably activated by binding of, in this case, FOXA1. So FOXA1 binding and presumably other factors, transcription factors, activate the enhancers. These interact, but these are prevented from interacting other genes because of this boundary wall, which is made by cohesin. So that, we believe, is the framework for gene regulation or transcription control, why some genes are active, how an enhancer chooses its transcriptional target, and how an enhancer is blocked from interacting with other genes. So enhancer promoter interactions direct the specificity of transcription regulation, whereas enhancer boundary interactions or boundary-boundary interactions serve as protective moats, walls, to regulate off-target chromatin interactions and also control transcriptional noise. So with this, we've kind of have some idea about the fundamental relationship between Pol2 associated chromatin interactions and the higher order chromatin genome architecture defined by cohesins and the insulator protein named CTCF. So the next question is, how does the master transcription factor, in this case AR and FOXA1, how do these factors get regulated? So the regulation of the regulators, AR and FOXA1 are the regulators. How does the gene loci coding for these proteins get regulated? And it's well known that AR amplification is common. AR amplification is seen in the majority of metastatic castration-resistant prostate cancer. Castration-resistant is people who fail these androgen hormonal therapy. And there was a group there were a couple of groups that reported an AR enhancer. And indeed, AR has an enhancer. And we noticed that this enhancer loops in and regulates the AR gene. So this blue shade in the right is the AR gene, and the promoter of AR gene interacts with the enhancer. And what happens is, in most men with castration-resistant prostate cancer, both AR and its enhancer get co-amplified, and as a result, there is massive chromatin interactions. So in the top is a cell line in which there is no AR amplification. So there is a single loop between the enhancer and the protein, and you can see the RNA in green. And VCAP cells have massive AR amplification. Both the AR and the enhancer is amplified. So you see massive chromatin interactions, and this presumably contributes to massive transcriptional upregulation of the AR gene, and which you could see by a really significant RNA-seq signal. So this is cell line data. So cell line data is always okay. We really need to see what we discover in cell lines. Is it holding up in patients? So we had a fantastic collaboration with the team of Johan de Bono and his amazing co-workers in Royal Marsden, London, and they did array CGH, high density array CGH analysis or copy number analysis in about 27 tumor specimens from tumor metastasis. As you could see, both the AR gene and the AR enhancers are co-amplified. Interestingly, there is more enhancer amplification in the left. Like these are the patients in which both the enhancer and the AR are amplified. Deep red is amplification, light red is copy gain. But there are many patients where there is a copy gain of the enhancer without really a change in AR. So we think that AR enhancer amplifications are indeed more prevalent than AR gene amplification. We also saw something really surprising here. Generally, copy gain gives you more copies of something, and deletion gives you a loss of something. So these are really mutually exclusive. So you should not expect a deletion when there is an amplification. But we were really surprised to see subclonal deletions in the context of amplification. So indeed, we saw some patient tumors in which you had massive amplification of this complete subchromosomal region. There was also deletion, subclonal deletions, but these were very targeted deletions between the enhancers and the AR gene. So copy gains increases the number of connections between the enhancer and the gene. We believe that in the context of copy gain, if there are targeted deletions between the enhancers and the gene, these could further increase the chromatin interaction strength, and chromatin interaction strength governs transcription. So you could have amplification and you could have microdeletion, but if it happens in the right direction, in the right location, these could act very synergistically and drive the overexpression of oncogenes. So copy gain increases the number of modules of interactions between the enhancers and the gene, and the deletion increases the interaction strength. And that's what we see in the next slide. We indeed see that there is an increase in RNA Pol II chromatin interaction strength between enhancers and promoters, and there is an association between, when there is a decrease in distance, there is an increase in chromatin interaction strength. So when there's an increase in chromatin interaction strength, there is an increase in transcription. So copy gains, deletions, these all tune in enhancer promoter connectome, and this could lead to changes in transcription of AR, which is the key master transcription factor that is involved in prostate carcinogenesis. So here is the proposed model of AR dysregulation in metastatic CRPC. in a normal cell, you have an enhancer of AR that drives AR, but in the setting of metastatic cancer and in the setting of amplification, you could see multiple modules. Here you see just a single module. It is in an X chromosome. If there is amplification, you could have 3, 4, 5, 6, or 20 copies, 20 modules of interaction. And in subset of those modules, if there is a deletion, the interaction strength goes up, so you see more transcripts. And we also see that this enhancer cluster acts as a landing site for AR, FOXA1, and ERG. So indeed, master transcription factors like AR and FOXA1, they govern their own expression. So they bind to their own enhancer and they control their own expression. So the other master transcription factor is FOXA1, which is also called a pioneering factor. And in this case, we saw that this is FOXA1, and we see that multiple enhancers located in the adjacent MyPol1 gene. So there's a gene called MyPol1, which is next to FOXA1. But this gene has a lot of enhancers. And in addition to being a gene, FOXA1 provides enhancers that connect to, in addition to being a gene, FOXA1 also provides sequences for enhancer sequences, which get activated and in those cells which have AR and FOXA1, and these enhancers connect to FOXA1 and drive the expression. So MyPol1 is both a gene and also an enhancer of FOXA1. And again, we see that FOXA1 is a master regulator. These enhancers are co-occupied by master regulators, which are ERG, FOXA1, and AR. So indeed, we are able to understand how master transcription factors like AR and FOXA1 regulate their own expression. This is because these factors bind to their own enhancer, so they control their expression. So the next question we addressed is, what is the nature of regulation, transcriptional regulation by AR and FOXA1? So before our study, people have done like the hundreds of ChIP-seqs for AR, and typically people think that the gene next to the AR binding site is the AR binding site. If it's an enhancer, the gene next to the AR binding site is the target of the enhancer. It's the gene to which the enhancer loops in. And we were really surprised to see that that's not the case most of the time. For example, let's look at the left plot. So this is the targets determined by RNA Pol2 chiapet, and we took a subset of those RNA Pol2 enhancers which had AR binding. And then we identified the genes that are regulated by RNA Pol2 chiapet, or our 3D genome pipeline. And this is the genes regulated, which are the nearest genes to AR binding site or FOXA1 binding site, and these are random genes. So we saw that the expression of the gene next to an AR or a FOXA1 binding site is the average expression is no different from random genes. So using an approach like the nearest gene is a target, you basically miss the gene, or maybe that enhancer was regulating many genes which were totally missed. And indeed, the true way to get the target of an enhancer is by using a 3D genome approach. And we see that the expression of target genes by our approach was significantly higher than the nearest neighbor genes. So indeed, you need 3D genomics to understand how the non-coding genome controls the coding genome in terms of transcription. So we use this approach to create virtual chromatin contact maps. I won't get into the details. So you have real chromatin contact maps by chiapet. Then you overlap that with AR or FOXA1 binding sites, and then you can create virtual contact maps of your factors of interest. And we were surprised to see that it's the EIF2 signaling, which is the top pathway that is regulated by AR and FOXA1 in LNCAP and VCAP cells. So the genes involved in EIF signaling or this EAF pathway regulates translational initiation. So you see 3D genome, non-coding genome enhancers, which get activated by combinatorial binding of transcription factors. These loop into and regulate genes within a framework guided by cohesins, CTCF, and this drives transcription. And you could basically control EIF2 or translation. Translation is like protein synthesis. So you could have a fine tuning of protein synthesis via 3D chromatin contact maps or via 3D chromatin interactions by these factors. So how does, so we've addressed how binding of AR and FOXA1 to distant regulatory regions control Pol2 activity in the 3D genome. So these are some representative examples. So in the middle panel, you see KLK3, which codes for PSA. PSA is the most common test. It's a biomarker for prostate cancer. It's a commonly used test. It's not a good biomarker, but it's commonly used biomarker. So we see that indeed in cell lines that express PSA, you have these enhancers active, and it's not just one enhancer and one gene. It's a hub. You have a hub of genes, KLK3, KLK2, and there's a bunch of KLK genes of the same family, and these are all interconnected. And in a cell line that is AR FOXA1 positive, these enhancers are activated by AR FOXA1 binding, and you have these connections. But in an AR negative cell, these enhancers are not active. You see different chromatin interactions. So the availability of master transcription factors dictate the fate of the cell by activating enhancers, and these enhancers drive transcription, drive genes by looping into transcription start sites in a framework which is determined by cohesins and CTCF. Again, this is another example. What we see is, in this case, in LinCap cells, you have the gene called ZBTB16, which is highly expressed. And that's because the enhancers are active. Why are the enhancers active? Because you have AR binding, you see in this panel, and there's combinatorial binding of AR and FOXA1 leading to enhancer activation. These enhancers interact with the promoter of this gene. This gene is active. This hub is within the RAD21. So you have the short loops of Pol2, which are within long loops, which is RAD21. And RAD21 blocks these enhancers from activating genes outside. Like these genes are not active. But in DU145, when you do not have AR or FOXA1, none of these enhancers are active because there's no AR FOXA1. So there's no Pol2 interaction. The gene is inactive, but you can see that the insulation is still there. The RAD21 interactions are still there. So enhancer activation and transcription regulation and CTCF binding and RAD21 looping are kind of not totally related. They're not related at all, although they work together. So while AR binding and enhancer activation and Pol2 interactions upregulate genes like ZBTB16, the cohesin associated chromatin interactions, these act as boundaries and prevent these enhancers from activating genes outside the neighborhood. Again, MYC, you see different cells, many cells have MYC. In the center, you see MYC, but the way MYC gets active is very different. In AR positive cells, you see interactions in the left. These are AR bound enhancers which loop in to regulate MYC, but MYC is also active in AR negative cells. That's because other transcription factors bind elsewhere, activate enhancers, and these enhancers loop in. So this complete space is non-coding genome, and there are some RNA which also facilitate chromatin interactions and help in MYC expression. Now, I just have a few more minutes. I will now go from, these are all somatic changes. So I talked about copy number changes, which happens during disease progression from clinically localized cancer to metastatic cancer. But now I'm talking about genetics, cancer predisposition, or how cancer really happens. So I will give a brief one example of transcriptional dysregulation by prostate cancer germline risk alleles. So this is a complex diagram. All that it says is there is an enrichment of risk alleles in enhancers. So GWAS, or genome-wide association studies, have identified about 180 risk alleles, and men with these risk alleles are at a higher likelihood of getting prostate cancer. These are associated with initiation of the disease. And RNA Pol2 bound regions and chromatin interactions are indeed enriched for PCA risk SNPs in benign as well as AR positive cells, but we do not see these kind of enrichment if a cell loses AR expression. So this is the expected distribution, and this is where you see the observed, which is way more significant. So a lot of these risk alleles happen within non-coding regions. These are not really non-coding, these are enhancers. So I'm just going to give an example of one particular risk allele. So this risk allele, which is RS684232, is upstream of this gene called VPS53 in a proximal enhancer element, which has FOXA1 binding, AR binding. This is an active enhancer. This also has ERG binding. ERG is ERG overexpression is commonly seen in prostate cancer because of a gene fusion. So one would hypothesize that although this is outside this gene, but because it's in an enhancer, maybe this risk allele regulates this gene. What we really saw, but why is this 3D genome study really helpful? Because when we have these 3D genome maps, we indeed see that the VPS53 promoter actually loops in and interacts with another gene called FAM57 and Gemin4. So RNA Pol2 chia-pet analysis revealed chromatin interactions between all the three genes. So now you could extend your hypothesis. You could extend and you can say that, well, this may regulate this gene, but because this gene loops and interacts with two other genes, maybe everything is co-regulated. And if you see something in a cell line, one could always call it as an artifact. It's not really meaningful. So you have to really see this in patient samples. So that's what we did. We took, and you can see a data in one clinical cohort, but then one could say, oh, you just see it in this clinical cohort. So you have to see it in a second clinical cohort. And sometimes you need to see it in multiple clinical cohorts. So we took the CPCG cohort, and we see that the presence of this risk allele, AA, AB, and BB. BB is the risk allele in homozygous fashion, AB is heterozygous, and AA is a wild type. The presence of this risk allele is associated with downregulation of not just VPS53, but also FAM57 and Gemin4. This is not just true in the CPCG cohort. We also see this in the TCGA cohort. Again, TCGA cohort is a cohort of it's a cohort where you have a lot of tumor specimens, and we could get to download this data, and we see that again the presence of the risk allele is associated with downregulation of the three genes VPS53, FAM57, and Gemin4 in patient tumors. Now that's not just it. We also see it in the third European cohort, the Porto cohort. So by stratifying the tumors into AA, AB, and BB genotypes, with A and B representing the wild type and the risk allele, we indeed see that the presence of the risk allele is associated with the downregulation of all the three genes in all the three cohorts. So indeed, gene regulation happens in 3D. It's not just a cell line phenomena. It happens in real patient tumors. So then if this really happens, the next hypothesis is maybe there is an enhancer and the enhancer activity is going down. So that's what we tested in the Porto cohort ChIP-seq analyses. So there is a fantastic Nature Communication paper available where ChIP-seq has been done for AR and a lot of other factors and enhancer marks in about 100 prostate tumors. So we reanalyzed that cohort, and we indeed see that there is an H3K27 acetylation mark in gene 1, VPS53, and this acetylation goes down not only in VPS53, also in FAM57, also in Gemin4. So indeed, the presence of one risk allele upstream of this gene in a non-coding region is associated not only with the downregulation of the transcription of these 3 genes, but also the enhancer activity. So the next question we had to address was, this is more genetics. I won't go into the details, but sometimes in genetics, there's something called linkage disequilibrium. Your allele can be tagged with something else, and maybe this allele is not doing it, something that's co-segregating with it. So we did some functional studies, and indeed we see that when you overexpress the wild type allele, you see increased transcription. If you put the risk allele, the transcription goes down. So it's really, it's functional. It's this allele, the presence of this allele is downregulating transcription. Now, why is the enhancer activity going down? And that's presumably because there is a reduction in AR binding. Again, when we analyze these tumors, we see that AA is associated with maximum AR binding, AB is associated with intermediate, and then BB is associated with low. So it's the transcription factor. And it's not only that, it's cooperative binding of transcription factor. So this enhancer also has ERG binding. So we were able to stratify patients based on patient data. This is not cell line, patient data based on ERG status. Indeed, we see that the downregulation was of all the 3 genes was way more significant in ERG positive cases in comparison to ERG negative cases. Now, what's the clinical significance of this? Indeed, these 3 genes go down as when you look at prostate cancer specimens by increasing grade. The increase in grade is associated with a decrease in expression of these genes, and there is also some association with biochemical recurrence-free survival. So what's the model? The proposed model is that these 3 genes are interacting. Because you have a risk allele, there is presumably a loss of chromatin interactions and downregulation. So this is really significant because when you have a protein coding gene which changes one amino acid, that one gene is changed. Here you have a non-coding variant that doesn't change just one gene, it changes 3 genes. And so we really addressed this question, like this is just one example. There are way more examples, which of course I can't cover because of lack of time. So indeed, germline risk alleles and somatic mutations interact we address this question, and we've shown that germline risk alleles, like these risk alleles, and somatic mutations like ERG, these interact. So there is a germline somatic interaction which modulates transcription which modulates the transcription of genes via long-range chromatin interactions. So this is like a work which is impossible to do by one single lab, and we had amazing collaborators in different countries. So in my laboratory, Susmita Ramanan did all the ChIP-seq and Chia PET analysis. She did the experimental part, and GIAPE UN did the bioinformatics part, and we were supported by Yong Chen in Michael Zhang's lab, who was a statistician. He developed the DNB model. So this is a work which is impossible by one lab. So my lab, mainly led by Susmita and GIAPE, and they were supported by Guem He, Yang Peng, Adam, and Nida. We collaborated with other labs in UT Southwestern. In urology department, we collaborated with Ganesh Raj. The bioinformatics core in our department with population scientists in UT Southwestern. We had a fantastic collaboration with UT Dallas, Michael Zhang, and Susmita, GIAPE, and Michael Zhang, Yong Chen are the co-first authors of the study. And me and Michael are the co-senior authors, which was published last year in JCI. Johan de Bono and his team helped us look at the significance of the study in metastatic tumors. The AR angle was solved with Johan de Bono's collaboration, and we collaborated with Paul Boutros and Katie Houlihan to analyze the germline part. And it's not possible without generous support by funding agencies, which involves NIH, CPRIT, and also the US Department of Defense. Thank you very much for listening to my work. I'm basically telling what my team did, and it's such an honor to present the work by this great team. I'm happy to answer any questions that you may have.
Yeah. So thank you, Ram, for the excellent talk. Now we look for some questions. I'll check the chat box. Yeah. So it was really a jam-packed slides that you had. And so what do you think the clinical application would be from your findings?
One application is clinical application of this kind of a work. Okay, yeah. So there are several applications. First application is understanding cancer incidence, because prostate cancer is a very it's got a very strong genetic component, and this is linked to risk alleles, and these risk alleles are in non-coding regions. Non-coding means there's no protein coding regions. To really understand the meaning of the non-coding region, you need to see what's going on. Is it an enhancer? Is it a boundary element? If it's a boundary element, you need to know what's happening. Is the boundary gained or lost? If the enhancer activity is gained or lost. So to understand the mechanism by which risk alleles contribute to cancer, that's one application. And then you have a lot of copy gains. It's not just gene copy number, you have enhancer copy number. Sometimes you have a gene rearrangement. That's because of an enhancer activating a gene. So by looking at the 3D genome, you can really understand how the non-coding genome controls the coding genome, and how somatic mutations, copy number changes, copy number losses, and germline variants impact the 3D genome to influence transcription and change the lineage, or change the predisposition for cancer.
Okay, yeah, thank you. So any questions? Any other questions from participants? Anybody would like to interact with the speaker?
So as of now, there's no question in this chat box, and I'm sure that speaker's time is also I think it's by midnight, I guess. So if the participants have any doubt, we can just post them through email, and so that he can answer whenever he finds time. Thank you so much, sir, for being with us, especially in these odd hours.
I like to express our sincere thanks to Dr. Ram Mani for giving an excellent coverage on the three-dimensional landscape of RNA-Pol2 associated chromatin interactions in prostate cancer. It is really interesting to know that his work discovered thousands of novel chromatin interactions, and his talk was packed with a lot of key findings on complex regulatory networks involved in prostate cancer. I think he's ready for next Science or Cell paper, and I wish best of luck. And I'm sure all the participants would have benefited by the talk. And later you can for all the participants, you can interact with Dr. Ram Mani through email, or you can also send us the questions, and you can later discuss. Okay. Yeah, thank you. Thanks a lot, Ram, for sharing your
very much,
work.
sir. Thank you very much.
You're very welcome. It was my pleasure to present.
Yeah. So just 1 second, Ram. I hope you can see my...
Definitely, yes, ma'am.
Yeah. So you'll be receiving this shortly. So this is a small gift from us. Thank you.
Thank you.
Thank you, ma'am.
Yeah, thank you. And so Adira.
Our next speaker is already here. I'm happy to welcome Dr. Vinod Skaria. He's from IGIB. I request Dr. CS ma'am to introduce the speaker.
Yeah, sure. So I'm very glad to introduce Dr. Vinod Skaria. Yeah. Okay, just 1 second. I stop my screen sharing. Okay. So I'm very glad to introduce Dr. Vinod Skaria, who's currently a principal scientist at CSIR Institute of Genomics and Integrative Biology. And he's also an adjunct faculty at Indraprastha Institute of Information Technology, Delhi. He's a clinician scientist, widely regarded for his pioneering work on human genome sequencing and significant contribution in the area of clinical genomics, allied with computational biology in India. He has many feathers to his cap, like the accomplishments in sequencing the first Indian genome, first Sri Lankan genome, and first Malaysian genome, to cite a few among many. And he's a Kavli Frontiers of Science Fellow of the US National Academy of Sciences and an elected fellow of the prestigious Royal Society of Biology, UK. And he's also associated with many network programs like Genomics for Understanding Rare Diseases India Alliance Network, which is abbreviated as Guardian, and then genomics and other omics technologies to enable medical decisions. It is called as GoMed. And also he's involved in Indigen program on population genomics for public health. And he's also a part of the program which is doing genomics of Arab and Middle Eastern populations. And obviously now he did a lot of sequencing work in COVID-19, and already he has a lot of reputed research papers. And now I'm very glad to invite Dr. Vinod Skaria to deliver his talk on what did we learn from the SARS-CoV-2 genomes from India.
Thank you, Dr. Subbalakshmi, for inviting me here, and it's a pleasure to be talking to you all. I hope my slides are visible.
Yes.
Yes, sir.
Yeah, so over the next 35 to 40 minutes or so, let me introduce what we have actually learned from sequencing a lot of SARS-CoV-2 genomes from India. But before I start my talk, let me introduce what my lab works on. We call ourselves genome informaticians. That necessarily means that we work on computers, trying to look at genomes and trying to make useful guesses from these genomes. The variety of things that we attempt to address using computers. We try to understand genomes of organisms. This includes human as well as model system genomes. Trying to understand how they are organized and how they are regulated, and also understand the functionality of non-coding RNAs. We work quite extensively with clinicians on understanding rare Mendelian genetic disorders, and also to understand the underpinnings of effective drug dosage and use, which is called pharmacogenomics. We quite extensively work with a lot of collaborators around this region, include Southeast Asia as well as the Middle East, trying to look at genetic epidemiology of Mendelian genetic disorders. And a small portion of my lab actually works in the area of pathogen genomes, where we used to work on tuberculosis, but of course, with the viral pandemic, which happened last year, we also ventured into working on SARS-CoV-2 genomes. And my today's talk is essentially the work from this small area of looking at viral epidemics. Now, for students out there, what has really changed in the present pandemic compared to what has happened previously was that we today have a better microscope. And to exemplify this point, what I would suggest is, if you have ever gone back to literature and try to understand how epidemics or for that matter, even pandemics have been investigated before, most of the previous pandemics or even outbreaks or epidemics have been investigated using traditional microbiology. And this includes culture, isolation, and observation of the microbes, and in many ways, assays to prove the pathogenicity of the pathogen. Now, what really happened in the last 1 decade was the drastic improvements in the throughputs for sequencing, nucleotides, and if you believe every organisms in the world would have a genome, that genome could now be sequenced at quite effective rates in terms of the throughput, at quite effective speed in terms of the time to sequence, and quite effective in terms of the analysis capabilities that we can today do. So in other words, what today has really changed is our ability to understand pathogens at much greater resolution that is not previously available. And that's essentially exemplified in the current pandemic. It didn't take months to actually make available the sequence of the organism that caused the pandemic. And as you see here, the first genome of SARS-CoV-2 was published in February 2020, very early in the pandemic, and the sequence was actually available even much earlier than the publication. Just to exemplify the point that it takes just days, if not a week, to actually be able to understand what really caused an epidemic. And this is a considerable departure from the traditional ways of doing microbiology. Now, if you really go back to the paper and try to understand how was this possible, you would really understand that traditional microbiology was never employed in this particular pandemic. So if you look at the methodology, you will see the samples of the bronchoalveolar lavage was obtained, RNA was extracted, the RNA was sequenced and analyzed to be able to identify the pathogen, not just identify the pathogen, but also put them in the context of phylogenetic tree to understand how similar or dissimilar are these organisms compared to the genomes that have been previously sequenced and deposited in public domain. Now, the other thing that you need to keep in mind is that all organisms evolve by accumulating genetic variations. A subset of these genetic variations could be functional or deleterious, but nevertheless, what really matters is the speed at which an organism can evolve. SARS-CoV-2, like all other RNA viruses, do have a fairly constant mutation rate, and this mutation rate is approximately estimated to be 1 in around 7 to 10 days. In other words, as a virus gets transmitted from individual to individual, over time, the mutations do accumulate in the genome of the virus. And I will come to this sort of central theme again and again, just to exemplify that this has a lot of applications, not just in understanding the evolution of the virus, but also in understanding the genetic epidemiology or the spread of the virus. Now, if you really go back to look at examples, so these are just 3 random genomes early in the pandemic, collected from December 26th, Jan 8th, and February 27th. All of them are available in public domain. And what you see here is over time, the virus has accumulated mutations, and this mutations is what we really go back and look at. Now, the other thing that has really changed in this particular pandemic was the wide availability of genome sequences and researchers willing to share the genome sequence in public and open databases. And today, 2 large databases, one is GSAID and the other one is the NIH SRA4O data and the GenPank for completed genomes, sort of become the center stage for this availability of genomes far and wide. And never ever before in the history of mankind, you had 590,000, almost becoming 600,000 genomes of SARS-CoV-2 available, or for that matter, any organism available in public domain, which essentially serves as a central point for all researchers who want to work and understand how the virus has evolved, or for that matter, any pathogen has evolved. The other point that I also need to exemplify in here is we need to keep in mind that virus doesn't really grow in a linear term. In many ways, it diverges out because there are individuals who could spread the virus to multiple individuals and so on and so forth. Now, as the virus gets transmitted from individual to individual, it accumulates mutations at a constant rate, and the mutations occur in the background of the virus that already was transmitted to that particular individual. Or in other words, you would have lineages or chains of events, each of this virus containing a unique barcode of genetic mutations, and these genetic mutations are dependent upon the ancestry of that particular genetic lineage or the genetic background. So just exemplified in here, like for example, if you count the mutations as 1, 2, 3, 4, 5, 6, the virus accumulates mutation to make 1, 2, and then at a point of time, it can accumulate a mutation which is 1, 2, 3, or it can accumulate another mutation which is 1, 2, 4, and so on and so forth. So just by looking at the barcodes of these genetic mutations, you can actually trace the ancestry or the origin of the virus. And if you have adequate genomes in time and space, you can actually calculate at what point in time that this infection happened or this divergence event occurred. Now, if you sample, ideally, a population of humans at any point of time who are infected with SARS-CoV-2, ideally, you should have equal proportions of all the different genetic lineages of the virus. But that's not what really happens. At some points in time, some genetic lineages are far more in number compared to other genetic lineages. As you see here in the second time point, 1, 2, 4 is far more in number compared to 1, 2, 3. And these are essentially what we call as emergence events. And this could be because of largely two factors. One, the host factor, which essentially means that an individual transmitted the virus to a large number of individuals, something what we call as super spreader events, or the virus which is gained a particular genetic mutation, which makes it more infective or that enables it to spread far and wide. So such emergence events are of interest to scientists because that allows us to understand both the host as well as the viral factors which come together to create such outbreaks and which allows a particular genetic lineage of the virus to spread much more than the other genetic lineages. Now, practically all these genomes could be put into a sort of a phylogenetic tree, and that is dependent upon the similarities of each of these viruses, or for that matter, the similarities in terms of the mutations. And if two viruses share the same set of mutations, then they would co-occur or occur closer in a particular phylogenetic tree. What we need to keep in mind is that when we draw these phylogenetic trees, we are essentially talking about genetic lineages because they are all different by just a few genetic mutations, which therefore would make them a genetic lineage and a clade and not really a strain of the virus. So you would have heard in newspapers that a new strain of virus is emerging, but they necessarily mean is that it's a new genetic lineage of the virus. Now, SARS-CoV-2 is a particular strain of the virus consistently different from the SARS-CoV, which was caused for the previous coronavirus pandemic, and of course the MERS-CoV or the Middle Eastern Respiratory Syndrome virus, which was cause of the outbreak in the Middle East. Now these are different strains of coronavirus, but what we're talking about in the press are essentially clades or genetic lineages of the virus, which are all SARS-CoV-2, but all distinctly different from each other by a few or a handful of genetic mutations. Now, there are, of course, resources, quite considerably large and curated resources like NextStrain, which has put up samples of genomes from across the world in terms of a phylogenetic tree. Now, as you see here, and as I stated before, a subset of viruses or some lineages do have a large preponderance in a phylogenetic tree compared to others. This could be because of multiple reasons, like I sort of stated before. But what I need to exemplify in here is that these lineages are all different by just a few genetic mutations. Otherwise, they're all SARS-CoV-2. Now, you have seen different colors in here, and these different colors essentially signify clusters or genetic lineages of the virus. very early in the pandemic, typically in the early months of the pandemic, these clusters or lineages roughly translated to the geographical origins of the virus, or the geographical spread of the virus rather. So as you would know, the B, which includes the B, B1, B2, and B4 are the genetic lineages which spread out of China into Southeast Asia. And each of these lineages have very specific geographical sort of preponderances. The A2A, which is a blue, what you see out here, is today the largest lineage, which is marked by a very specific genetic mutation, which is called a D614G. And this largely spread in Europe and further with the rest of the world, and today is the most dominant lineage of the virus present across the world. Now in India, to give a brief history, the first case was confirmed in January 2020, and of course it has been more than a year, and we have a considerable large number of cases, and of course a significant number of deaths, being a very large country with a very large population. Now put this in perspective, the genomes that we have available, there are around 10,000 odd genomes that is estimated to have been sequenced in the country. At least 6,900 of these genomes are available in public domain in multiple different resources. And this has come from the work of a large number of researchers from over 21 medical and research institutions spread across the country. Now, one of the resources that you can access the genetic variations and these frequencies is this database called INDICO, where we have made available the frequencies of genetic variations which are found in the genomes from India. Now, to do genomes and to be able to do it at scale that we require, you need to have tools and techniques and methodologies to be able to build high throughput genomics. And one of these tools is essentially an approach what we call as COVID-seq, which is essentially one of the first NGS tests approved by the US FDA and available for detection as well as surveillance in the US. And one of the first clinical validations or real-life applications of this approach, which is called COVID-seq, was actually undertaken in India in the lab of Dr. Sridhar Chivasubu, and with contribution to a fantastic set of young researchers in the lab during early pandemic. Now, in simplicity, this approach is quite flexible and modular, and you could do anything from 96 samples to approximately 3,000 samples in approximately a day's time to be able to sequence and analyze in terms of the detection of the virus, as well as to put them into the context of genetic lineages and also understand are there functional genetic variations in these samples. So in other words, such as high throughput approach, which also drives the cost significantly down, could enable a large number of genomes to be sequenced in a good turnaround time at an efficient cost to understand the spread of the virus across the country. Now, early in the pandemic, we have been fortunate to have worked with a large number of clinicians and collaborators from a number of southern states, including Tamil Nadu, Kerala, Karnataka, Andhra Pradesh, Orissa, UP, as well as Rajasthan, where we created this consortium called the INDICO Consortium, where we could sequence a significantly large number of SARS-CoV-2 isolates to understand how did the spread or how did the epidemiology of the spread occur in these states. Now, briefly putting things into perspective, what you see here is a phylogenetic tree. What you see here marked are the two genomes from the early pandemic sequenced by the National Institute of Virology, and this belong to the B clade, mostly out of China. The predominant isolates belong to the A2A clade, sort of mentioned that these are the genomes which have the D614G mutation, and predominantly early in the pandemic have come from European travelers. You have, of course, clades which have come from Iran. And apart from all this, you have a clade which doesn't find a significant map into any one of the global populations. Now, since this was very significantly different and has been found predominantly in India, we sort of named this lineage as the I or the A3I, I to signify that it is predominant in India, and A3I to signify that it at some point of time seems to have diverged from the A3 lineage, and the A3 lineage traces its origin to modern day Iran. Now, as I said before, each of these genetic lineages are only distinctly different from each other by a handful of genetic mutations. In the case of IA3I, it was marked by 4 genetic mutations which are not shared with the rest of the lineages or the clades out there. And of course, there's a 5th genetic mutation which seems to have been shared by the A3 lineage of the virus, and therefore the name A3I. Now, of course, you could look at the evolution of this clade in terms of how many number of mutations could be accumulated over time. And what we realize is that IA3I clade had a significantly lower mutation rate compared to all the other lineages which are prevalent out there. And the mutation rate was approximately 1.19 into 10 to the power minus 3 compared to 1.48 into 10 to the power minus 3 for the A2A and all the other lineages out there. So we sort of expected that at some point of time, this genetic mutation deficient virus, because it had some disadvantage in terms of accumulating mutations, therefore would die down eventually, because viruses evolve by accumulating mutations faster and more efficiently. You could also plot the kind of distribution of the IA3I, which is in purple, across the different states in the country. And what you also see is that different states have a distinctly different prevalence of this particular genetic lineage, with the majority in Delhi and southern states like Andhra Pradesh and Karnataka, but of course in very small proportions in states like Kerala, which had none of these samples of A3I clade. Now, as you see here, over time, the A3I clade has been gradually replaced by the more prevalent, the blue clade, or the A2A, which is a genetic mutation which contains the genetic mutation D614G. Now, it is very well understood that D614G containing viruses are much more infective than the viruses which don't have it, and then therefore they could have a selective advantage of transmitting the virus faster and more efficiently. And that's sort of exemplified in here, that today a significant large majority of genomes that have been sequenced late in 2020 has only the A2A clade or the mutation D614G. Now, I said before that looking at the genetic lineages and spacing them in time and space, you could also trace the origins and the spread of the virus as travelers spread the virus across different states and from different countries into India, and of course from India to outside. Now, that is one of the approaches that we call as molecular contact tracing, because contact tracing is typically effective only in small populations where you can trace each individual and ascertain the contact maps. But when individuals travel from port to port, or from, in many cases, across major cities of the country, it is very difficult to trace the ancestry of the lineages of the virus. And the only way today, which is sort of fail-proof, is to be able to sequence the genomes and understand how similar or dissimilar are these genomes compared to what has been sequenced in other parts of the country. Now, then therefore, you should be able to trace the interstate spread of the virus in space and in time, and also understand the dynamics of the spread. And this is very useful in policy building to contain the spread of the virus. You could also create contact maps, like I sort of mentioned before, and also identify hotspots or clusters of infections that have happened in different parts of the country. And each of these colors essentially signify a major state from which these outbreaks would have occurred. So if you have a very significant large cluster, that necessarily means that there was a very large outbreak in that particular state, and the diverging points essentially state how that particular cluster or outbreak have contributed to different outbreaks in different parts of the country. And as you see here, most of these clusters have contributed to significant interstate spread of the virus. And one of the major learnings from this analysis and from the genomes of SARS-CoV-2 in India is that a significant number of the spread in terms of SARS-CoV-2 infections have happened through interstate travel rather than by international introductions. Now, of course, by looking at genomes of viruses, we could understand how to make better diagnostics. I will delve a bit deeper into that. We could understand immune epitopes and the ways by which the viruses could escape such immune epitopes. And this is very useful to design probably better vaccines in the future. And of course, when we come to diagnostics, everybody in this room pretty much knows what real-time PCR is. And real-time PCR is today the gold standard for diagnosis of SARS-CoV-2, which offers a very high sensitivity as well as specificity for detection. But nevertheless, the real question remains that real-time PCR is based on primers or probes which can bind to the viral genomic material and amplify very specifically a stretch of the genome, which are essentially again probed by another set of oligonucleotides. Now, efficiency of binding of primers and probes are also dependent upon the genetic mutations at these sites, and then therefore genetic mutations in the virus could adversely affect the efficiency of the detection. Now, it's very simple to actually go forward. You could collect all the primers and probes from literature, and this was done by a fantastic set of colleagues and collaborators who volunteered from across different parts of the country. And of significant mention is that a significant large number of these information was put together by college students out of Bangalore. And of course, once you can map these probes into the viral genome, you could map the mutations from the viral genomes which are sequenced across the country and across the world and ask this question: are there frequent genetic mutations which occur in these sites? So the red dots that you see in the map are essentially variations which occur in the primer and probe binding sites. And the y-axis shows the frequency of these genetic mutations in the Indian population. So in other words, what you could see is that there are a subset of genetic mutations which would adversely affect the efficient binding of the primer and probes. And this information is very valuable because even without doing the first test, in many ways, you should be able to predict that a particular kit is going to be very effective or a particular kit is going to be less effective given the mutational landscape that is prevalent in a particular population or a particular state. The other aspect of genomics is related to the immune system. And for students, briefly, the immune system has two arms. The humoral immune response mediated by the B cells, which produce antibodies, and cell-mediated immune response, which is modulated by the T cells, which cause cytotoxicity. Now, both of these arms of the immune system are very important in our ability to contain the virus and also to prevent the adverse events, what we call as the COVID or the coronavirus disease. Now, in clinical settings, there are also some clinical conundrums that come across where individuals either could have persistent infection. That necessarily means that an individual cannot clear the virus very efficiently. Some individuals, at least a few of them, have been reported across the world to have a condition called reinfection, where an individual typically gets an infection, recovers from the infection, only to get a reinfection at a later point of time. And of course, at least in a much fewer number of cases, there will be a paradigm called reactivation, where an individual would have got an infection which has subsided at some point of time just to get reactivated or to get the disease worsened further after a period of time. Now, while these are clinical conundrums, the only way to prove whether it was a persistent infection, or a reactivation, or a reinfection is the ability to ascertain whether these were the same viruses or different viruses. Now, such studies are possible only if you have cohorts of individuals who have been followed up very consistently over time. And one of these cohorts were actually created by Dr. Vivek Gupta, a close colleague and collaborator at the JIMS Hospital in Noida, which is a medical college where all the people, or all the clinicians and healthcare workers on COVID duty were put on a constant surveillance with RT-PCRs every 15 days. Now, looking back at this cohort of individuals, what we realized is that a couple of healthcare workers had a condition where they had got an infection early in the pandemic, which they recovered subsequently, only to get another round of disease or another round of infection proven by RT-PCR, and again recover back. Now, incidentally, none of these individuals were symptomatic or had classic symptoms of COVID. In other words, they were asymptomatic for the COVID. Now, this is possible to ascertain only, as I said before, if you had a surveillance program put in place, because you would test for COVID only if you have symptoms for COVID, and without symptoms of COVID, typically none of you would go to a testing center to get tested. Now, in this particular case, we could access the samples from both episodes, and what we really show is that the viruses from both episodes, E1 and E2, for the two individuals, I1 and I2, were distinctly different from each other by genetic mutations. And this essentially, therefore, proves that these individuals have got reinfection with different isolate of SARS-CoV-2, different from each other by few genetic mutations. Now, of course, as I stated before, the other closely mimicking conundrum is a case of reactivation. And in a case of reactivation, we expect the genomes from episode 1 and episode 2 to be exactly the same, because it was the same virus which had got infected, which had infected this individual in the first place. And this is another case referred to as from Vedanta, a child with stage 4 neuroblastoma who was on anti-cancer therapy. The individual tested positive and again tested negative, only to test positive again because RT-PCR testing was done as part of a routine checkup because these individuals were on chemotherapy. And the virus from both episodes had exactly the same complement of genetic mutations, which suggests that this is possibly a reactivation of the virus. And this also becomes the first case of reactivation of SARS-CoV-2 published globally. And of course, as virus mutates, there are mutations which would have very significant functional impact on the virus. And one of these impacts is its ability to escape antibodies, and what we typically call as antibody escape or immune escape genetic mutations. And in the recent months, there have been a flurry of publications which sort of characterized genetic mutations with the ability to escape one or more monoclonal antibodies, or panels of convalescent plasma which sorts of mimic the human immune response to a particular virus. Now, individually, these mutations are not of really great concern because these could escape one or more of monoclonal antibodies, but multiple of such genetic mutations occurring together could escape a significant number of antibodies, and therefore that is something that we need to be really concerned about. And for students and researchers who are interested in this, this is probably one of the only compendium of escape mutations available today. We list around 146 genetic mutations and their annotations, and you could, of course, download these genetic variations for an analysis and interpretation. And what we also realize is that the virus has an Achilles heel, just also because that these mutations co-occur on the receptor binding domain. So therefore, the virus cannot really mutate at every single point on the RBD or the receptor binding domain, because it affects its ability to bind to the human receptors and viral entry into the cells. In other words, there are only very specific hotspots in the spike protein which can mutate and escape antibodies. And to our beliefs, we have pretty much identified all the more major escape mutations that could ever occur on the virus. And this is of great significance because this is the valuable resource that you will require to build the next generation of vaccines which can counter immune escape genetic variations. Now, if you look at these genetic variations across the globe, there are at least a handful of them, at least almost a dozen of genetic variations, which have now become quite prevalent in the population. And prevalent when I say more than 1% in one of the countries from which genome sequences are available. And some of these, like for example, 477 or E484, are significant also because they could escape one or more antibodies. In the case of 484, it could escape multiple antibodies as well as panels of convalescent plasma. And therefore, occurrence of these genetic mutations in genomes are of significant concern because it could affect the efficacy of the vaccine programs. And if you look at Indian map, there are also genetic variations which are very predominant in India. And one of these genetic mutations is N440K, which we found very predominant in the state of Andhra Pradesh sometime mid during the pandemic. And of course, very recently there have been report of E484K and E484Q, which are also genetic mutations which can escape antibody response. So you would all ask, of course, in many ways, the pandemic has been weaning down across the world, and vaccines have been increasingly being administered, not just in India, but also across the world. Now, how does genomics fit in this paradigm? And what I would suggest is that genomics becomes even more important in the current scenario because we need to identify and tackle outbreaks and emerging genetic lineages, especially the ones with immune escape phenomenon. And even more importantly, we also expect a very small number of individuals to get infections even after the complete dose of vaccines. And these would be called breakthrough infections. At least we have started getting samples of such breakthrough infections, and sequencing the viruses in such breakthrough infections would be extremely valuable resource to understand how does the vaccine modulated immune response is countered and escaped by the viruses. And I think I'll stop there and take questions. But before I do though, let me acknowledge a lot of people who made this possible. All the sequencing was done by my colleague, Dr. Sridhar and his lab with a fantastic set of young researchers, including Rahul, Imran, Gyan, Sumit, and Sanjay. Fantastic set of collaborators from across the country who are part of the consortium. Many students in the lab who worked day in, day out, even during the lockdown, to put together the resources and the analysis. Collaborators from across the spectrum of institutions, Rakesh Mishra and DDTR from CCMB, and of course, not least, all the researchers who wholeheartedly put together the data in public resources from over 21 research and medical institutions across the country. And it is not possible to do all this without considerable amount of funding, and the consistent funding support has come from CSIR through multiple different programs. I think I'll stop there and take questions, but before that, for young researchers out there, there are a lot of resources which are available where you could understand the genomics and epidemiology of SARS-CoV-2 in the country, and of course, could learn a lot through the protocols that we have put together and published. I think I will stop there and take questions. If there are interested students who are interested in volunteering to any one of these efforts, you're welcome to write to me. Thank you.
Thank you, sir, for the informative talk on the SARS-CoV-2 genomes. There are a few questions in the chat box.
Sure.
Okay. There is a question from Visha. She has asked, how often has the COVID vaccines to be administered?
No, that depends upon the vaccine type and the schedule for administration that is being optimized during the clinical trials. Now there are vaccines which require 2 doses of administration. There are also vaccines which require only 1 dose of administration. So that is dependent upon the type of the vaccine and the optimization that has been done during the clinical trials.
Thank you, sir. Another question from D. Abhirami. Is there any solid proof that China is origin for COVID-19?
Yeah, so the early infection seems to have spread out of the country and including travelers who have visited the country. And for that matter, there has not been any contradictory proof suggesting otherwise and validated. So therefore, we should tend to believe based on what evidence is available.
Dear Pat, okay, one more question, sir. Can you also please mention some characters about the new mutated new COVID-19 virus?
No, so like I said before, that mutations are commonplace. Mutations do occur, and that is essentially the way the virus would evolve. So just finding a mutation on the viral genome doesn't really mean anything unless there is something else to suggest in terms of the functionality of the variant or in terms of the epidemiology of the virus that it spreads faster or slower. So what's really more important is the variants which have some functional significance, and you could go back to the resources to understand such genetic variations, and very specific properties conferred by the genetic variant, like for example, immune escape or increased infectivity. And these are essentially the viral variants, the mutations, or the lineages that we need to be concerned about.
There is a question again from Osha. Is the origin due to zoonosis transmission?
At least there have been good publications where they have sort of tried to address this question in a very logical way, putting into perspective the genomes from other organisms, or coronaviruses from other organisms into perspective. So I don't think there is any contrary evidence as of now. So, I mean, science is essentially based on evidence. So until we have a contrary evidence, we should not really tend to believe anything else.
Okay. This is from Rajeshri. What mediates the rapid mutation of COVID-19? Is it geographical or is it based on host response, which causes rapid mutation?
Okay, so that's a very interesting question. So broadly speaking, the mutation rate of the virus is pretty much constant across geographies. It is not really something that is very specific to a particular geography. But now, having said that, you should also understand that the mutations occur while the virus is in the cell or the human cell. And these virus mutations are essentially because of the human machinery which replicates the viral genomic RNA. And therefore, these introductions are made because of inherent errors in the human system. Now, there have been publications where they have shown that significant patterns of enrichment or some genetic variations or patterns of genetic variations are enriched in some populations. Now, this could be because of two reasons, like I stated before. One reason could be that a particular population or a particular individuals would have a very specific error-making mechanism, which is very unlikely though. The second possibility is that a very specific introduction event and the dynamics of the spread, which includes super spreading events, would have contributed to an emergence of a particular lineage or a particular genetic variant, which is found predominantly in a particular population. So unless it is being followed up very systematically through studies, it is very difficult to ascertain whether it is a host factor or a viral factor. Now, having said also that there is a constant mutation rate, there are also genetic mutations in viral proteins which could make it much less mutable. And one of the examples that I've showed is the IA3I lineage. And one of the reasons why we believe that IA3I genetic lineage has a much lower mutation rate is not because of the human factors, because it is spread across different populations in the country, but rather a mutation in the RDRP, and of course there is no experimental proof to this, it's still a hypothesis, that the RDRP or the RNA-dependent RNA polymerase was possibly defective in this particular virus. And as suggested, because there was a genetic mutation in the RDRP protein for the virus. So it could be both human as well as the viral factors which could influence the mutation spectrum in a virus. But largely, the mutation rate has remained a constant.
Okay, few more questions are there, sir. This is from Sayanthini Chakraborty. Why does virus cause more PCOD casualties?
Well, I will not be able to comment on this because I don't have any data to suggest that these patients have more or less mortality. So I'm probably not the right person to answer that.
Okay. Another question from Ashika. Is there any point of care of diagnostic tool for COVID-19 as of now?
Yes, of course, there are. And in fact, there are many. So this includes the widely used rapid antigen test. So rapid antigen test essentially looks at viral proteins using an immunoassay. And these are essentially point of care and very rapid assays that are available and widely used.
Okay, another question from D. Abhirami. Are there any feedbacks about the Indian COVID-19 vaccine from other countries?
I didn't really understand that question because at present there are only 2 vaccines, I believe, which are widely being administered, and both of them have been manufactured in India.
Okay, sir. There is a question from Pandian. Please comment on the frequency of variations in SARS-CoV-2 compared to H1N1. Also, he has asked one more question. Is there any mathematical model to predict the virulence of different variants in comparison with the wild-type SARS-CoV-2 virus?
The answer to both of them are yes. There have been actually a large number of publications on at least the second part of it, but at least a couple of them on the first part of it, which I've come across. Since I have not personally worked on this topic, probably not the best person to comment on them, but of course, I've come across publications on both of them.
There is a question from Milan. Is there any change in the inflammatory marker and erythrocyte sedimentation rate in the new mutated virus infection?
I've not seen any evidence on this yet. I might be wrong because evidence has been emerging almost every second. But theoretically speaking, you should not expect that to happen because these are only very small changes in terms of the mutation in the virus. The rest of the 30,000 odd base pairs of the virus is pretty much the same. So theoretically, you should not find, except for very specific rare variants. And one thing that comes to my mind is a very specific genetic deletion in the ORF8 gene, which has been previously also documented in the previous coronavirus episodes, where deletions in ORF8 gene would cause much less symptoms and actually make the virus attenuated. And I've at least come across a preprint which has discussed the clinical features of ORF8 deletions. So I believe the fact is that you are right, that there are genetic mutations which could reduce inflammation in the individual. But I don't think much of the biology of this is really understood.
This is from Vaishali. Is the COVID vaccine safe for everyone?
At least the preliminary data that has been available from the massive administration across a subset of people, which is healthcare workers, you could fairly conclude that they're significantly safe.
There is a question from Kashi Vishwanath. Your work had been demonstrated well about the different variants of the SARS, but what is the significant cause that make these many mutations?
So the mutations occur at a constant rate, and this is also, as you'll see, a factor of time. So the more you prolong the SARS-CoV-2 spread or the pandemic, the more mutations that would occur on the virus. So that is a natural process of evolution of the virus. The faster we can sort of eliminate the virus, we would also be able to stop the virus from being mutated.
Okay, so there is one more question from Shankari. Currently, there is a sure surge increase in the number of cases of COVID-19 in few states of India. Can we attribute this to the reported individual mutation or combination of mutations?
Yeah, so the numbers have been increasing in some states at least. But to answer that question, what we also require is a significant large number of genomes from those regions where it has been increasing. And that really points to the fact that we need to have a much robust mechanism of surveillance where you could sort of sequence at a constant basis a subset of the viruses. Now, for most of the states where this emergence have been happening, there is very little data in public domain, which essentially restricts or limits the ability to answer the very pertinent kind of questions that you're trying to understand.
There is a question from Shushmita. Will the COVID-19 vaccine protect against the new strain?
Okay, so that's a very, very tricky question to answer. So briefly, what I can sort of describe is there are two kinds of vaccines. The first kind of vaccine, what I call, are based on the spike protein, which is the exposed protein which elicits antibody response. And the second set of vaccines are essentially whole virion or inactivated viral vaccines, which has a complete complementary, what I call, set of epitopes in a virus. Now, if your question specifically is, does a particular variant escape a particular kind of vaccine? The answer is yes. If you look at the specific lineages, which include the E484 genetic mutation, there have been studies which show that E484 can reduce efficacy for spike subunit vaccines. Now, does that mean that the whole virion vaccines are effective? The answer is we don't have evidence yet. So until the evidence comes in, we don't really have a clear-cut answer on whether it could affect those vaccines. So these are essentially also a property which is dependent upon the vaccine and the type of the proteins used in the vaccine. But if you're pretty much asking the question, are spike protein vaccines effective against these emerging variants? The answer is no.
This is from Shrikanta. The genome of the virus is changing constantly due to mutations. Then how a specific vaccine can be administered to all the strains? Is it safe to use for all the strains?
Okay, so that comes to my second part of the slides that I sort of mentioned. So there are basically two arms of the immune system, right? One, I said, is a humoral immune system, which makes antibodies. And the second is the T cells or the cellular immune system, which causes cytotoxic reaction. Now, the immune escape mechanisms, that's also why I qualified this statement, are largely antibody escape mutations, because these are mutations which can prevent a specific monoclonal or polyclonal antibodies to bind to the spike protein. And therefore, most of these genetic mutations are on the spike protein, because that's the exposed surface onto which antibodies can bind. Now, the other arm of the immune system, which I sort of stated about, is a T cell mediated immune system, which essentially means these are modulated by T cells, which the immunization happens by a very different and distinctly different mechanism, where the whole virus is chopped into peptides, presented on the MHC to T cell receptors. Right? So the repertoire of T cell antigens or epitopes are far larger in number and encompass almost the entire repertoire of proteins in SARS-CoV-2. Now, until now, as well as I know, I should always take this statement because evidence is always emerging. As far as I know, there have been no mutations which can actually escape the T cell mediated response in SARS-CoV-2. So that therefore means that an immune escape mutation is only going to prevent one arm of the immune response against the virus. So therefore, we don't need to really panic to say that nothing is going to happen. But having said that, at least one arm of the immune response, which is antibody-mediated immune response, is very important in preventing infection. Now, what you also need to keep in mind is that, and endpoints might vary depending upon how you look at it. In one case, you would want to prevent infections from occurring, and the other end, you might want to prevent the disease from occurring, because infections by itself is not lethal, right? 85% of the individuals would get SARS-CoV-2 and be perfectly asymptomatic, or would have very mild symptoms. What you're really scared about is to have COVID or coronavirus disease, which makes you severely diseased, which requires hospitalization, and which is also the cause of death. So depending upon the endpoints that you're trying to look at, you might want to say whether the glass is half full or the glass is half empty.
Okay, this question is from Ravi Varman. Is COVID a seasonal disease? Will they arise every year?
Very interesting question. So if you ask the question whether coronavirus is a seasonal disease, the answer is yes. So SARS coronavirus is just one of the types of many coronaviruses that occur, that infect humans. And coronaviruses have previously also occurred in humans. but they've never been cause of COVID. COVID is the disease which we are all scared about because of the mortality, but seasonal coronavirus infections are very rampant in human populations.
Ma'am, shall we conclude the discussion session or should we take more questions?
So it's enough actually. I think because of the want of time, we will stop with this.
Yes.
Yeah. So the participants can directly contact the speaker and get their doubts clarified.
Okay.
Thank you very much, sir.
Thank you. It's my pleasure.
Thank you very much, Dr. Vinod Skaria, for giving an excellent coverage on the genome of SARS-CoV isolates from India and its impact on diagnostics and identifying the immunity epitopes and eventually to develop effective vaccine. I actually appreciate your COVID-19 open research data and resources initiative. This has made his data available for anyone in an open format, and now it is widely used across the world to understand characteristics of COVID strains. So we should be very thankful to him. And also he's a part of many important national initiatives and consortiums. So the interested students can keep in contact with Dr. Vinod Skaria and do some work for his projects. So I believe all the participants would have benefited by his talk. And again, I thank him for his excellent work.
Thank you very much. And it's always my pleasure.
Thank you, sir. And thank you, ma'am.
Yeah, so we also have a small memento for Dr. Vinod Skaria, and we will be posting it to your address.
Thank you very much. Thank you.
Thank you for sparing your valuable time. Thank you.
Thank you very much again, sir.
Well, that's the wrap up of the invited talks for today's session. Moving on to the next agenda, I request all the participants of the oral presentation session 1 to get ready. For the first session, totally there are 8 participants: Dr. Nannu Shafakutullah, Vishnupriya, Soundarya, Divya, Sangeeta, Pon Saravanan, Atira Ramesh, Anuja, and Priyadarshini. Dear participants, we'll call your names in this order, so kindly raise hand in the Zoom space before 2 minutes of your allotted time. And I'm delighted to introduce our chairpersons, Dr. S. Sudharani and Dr. Madhu Dyavaya from the Department of Biochemistry and Molecular Biology, who will be judging this session. Now, I request Dr. Madhu Dyavaya to give the introductory remark to the presenters of this session.
Good morning, everyone. I hope you're enjoying the eminent talks from scientists. So now we'll start the session 1, oral presentation. As it is mentioned that the time given is for 7 minutes. I hope you try to stick to that 7 minutes, and I request you to focus more on your findings so that you could explain it to make it ready. So then you can start now. Thank you.
The first presenter is Dr. Nannu from Mangalore University.
Yes, ma'am. I'm here.
Sir, you can start your presentation.
Yeah, okay. Yeah, good afternoon, everyone. Myself, Dr. Nannu Shafakutullah from Mangalore University. here to present a small research on the topic anti-cancer effect of lactic acid bacteria in colorectal cancer mouse model.
Can you put your PowerPoint in the full screen mode, like PowerPoint mode? Thank you very much.
So the colorectal cancer is the third most common cancer in the incidence as well as the mortality rate, and it's accounting for around approximately 9% of all cancers and 8% of the cancers related death. So when you take the treatment available for this colorectal rectal cancer, we are having the chemotherapeutic drugs as well as the radiation, but there are more side effects and discomforts in the patients. So the prognosis for advanced colorectal cancer, it is very poor, and the prevention is required to control this incidence of the disease. And many studies have confirmed that there is the endogenous microflora are involved in the onset of this cancer. So this makes it reasonable to think that changing the intestinal microflora could do some changes in the environment where the formation of tumor development is taking place in the body. So several mechanisms are explained where the probiotic organism, that is the lactic acid bacteria, which are responsible to reduce the formation of colorectal cancer. It includes they are responsible for alteration of the intestinal microflora, inactivation of the carcinogenic compounds which are getting generated daily in the system, competition for the petrificative as well as the pathogenic microbiota, because this pathogenic microbiota, they're also involved in the formation of cancer. And this lactic acid bacteria which we are intaking, they can improve the host immune responses, anti-proliferative effect, as well as the fermentation of undigested food. And this undigested food also responsible for some generation of the toxic molecules which are involved in the formation of cancer. So in the present study, what we have done is we have isolated a lactobacillus. So that is a lactobacillus plantarum subspecies plantarum strain MF6, and this has been used in the treatment of NN dimethyl hydrazine induced colorectal cancer in the mice. So what we have done is we have isolated this lactobacillus, one organism, lactobacillus organism from the marine fish, that is from Sardinella, sardine, identified based on the morphology, physiological, as well as biochemical characteristics. Then the same organism has been used to identify its probiotic property according to the ICMR guidelines. And finally, after the complete identification, it has been subjected for 16S rRNA sequencing so that we can authenticate that this is the particular organism. And we have found that the organism belong to Lactobacillus plantarum subspecies plantarum strain MF6, and sequences have been deposited in the gen bank, and we have got the accession number. This experiment was carried out in the Swiss albino mice. We have taken 25 Swiss albino mice, and the experiment was carried out for 28 weeks according to the guidelines given by ethical committee. So the experiment was divided. The whole experiment was divided into 5 groups, and the first group contains a negative. It was a negative control. Second group was a positive control, where the positive control means we induce the organisms with the cancer-causing agent, that is the DMH. Third group was a pre-treatment. Fourth group was a post-treatment, and the fifth group was pre as well as the post-treatment. And each set was containing 5 mice. In the drug and doses preparation, here we're given a subcutaneous injection of the drug, cancer-causing drug, NN dimethyl hydrazine dihydrochloride, at a concentration of 20 mg per kg body weight. And the animals were fed with a probiotic, that is a lactobacillus plantarum, at a concentration of 2 into 10 raised to 8 colony forming unit twice in a week in the form of dissolved in the milk and given through oral garbages. And the following parameters have been studied. So that is the general health of the experimental mice, food intake, tumor formation, fecal microbial count, fecal pH, antioxidant activity, as well as the bacterial enzyme activity has been studied. So this is just a preliminary identification. So in the food intake, we have seen there is some changes have been found between the negative control, positive control, as well as the treatment groups. Because in the treatment group, there is increase in there is rise in the food intake as well as the body growth has been identified compared to the positive control organisms. Then we have seen about the formation of tumors. So we have dissected the lower intestine and we opened up the lower intestine and calculated the total number of tumors formed. So in the negative control, there were no any formation of tumors. In the positive control, we got approximately 10 tumors. Out of the 5 mice, we got approximately 10 tumors. And in the pre-treatment, post-treatment, and pre and post-treatment, so there is decline in the formation of the tumors. So this decline, we can say that it is maybe because of the functioning of the lactobacillus plantarum, which were given to the induced mice. So these are the structures of the presence of the tumors. Then we have studied about the histology to see what changes has been done because of the drug and what changes has been done because of because of this lactobacillus lactobacillus plantarum. So this is a picture of the normal mucosa. Then you have seen the formation of aberrant cryptic foci, that is the initiation or the generation of the tumor. So when in C and D, we can see there is decrease or reduction in the aberrant crypt formation because of treatment with the lactobacillus plantarum. So fecal microbial count you have seen, because the lactobacillus in the feces indicate the survival, because they have to survive inside the organism. Colonizing is required because this organism should be there inside the mice. So we have seen the lactobacillus count has increased in the mice compared to the coliformic organisms. And we studied about the pH changes which occur in the feces of the mice. So it has been hypothesized that the alkaline feces increases the risk of colon cancer. But in our treatment group, at the end of our experiment, that is after 28 weeks, we have seen that the pH of the feces has been drastically reduced to acidic. So that is 5.6, 5.7, and 5.5 compared to the normal pH. Then you've done the antioxidant study, because the antioxidant are the substances that can prevent and slow the damage to the cells caused by free radicals. That is the unstable molecules that the body produces as a reaction to the environmental stresses. So in that antioxidant study, we have studied about the GST, glutathione S transferase. So in the pre, post, as well as the pre and post treatment organism, the glutathione S transferase activity has increased. And we studied about the SOD, that is superoxide dismutase activity assay. So here also in the pre and post experimental group, the activity has increased, and in the post activity there is no it was not significant. And then we have done the GSH activity, and in the GSH activity also there is a rise in the GSH activity has been found in pre, post, as well as the pre and post organisms. Then we studied about the bacterial enzyme present in the feces. So the enzyme, that is beta glucuronidase as well as the beta glucosidase, have been implicated in converting, because they are responsible to convert procarcinogens into carcinogens in the lower intestine. So those carcinogens cause the mutation and leads to the formation of this colorectal cancer. So in our study, we have seen that there is reduction in the activity of the glucosidase, as well as the reduction in the activity of the glucuronidase functions in the system, that is in the feces of the mice. So in the conclusion, so this is just about the preclinical study. So we have not gone for further study, further identification of the markers and other things. It's still in the process. So in the conclusion, I can say that the probiotics confer anti-carcinogenic effect by excluding the pathogenic microorganism, and they're responsible to compete for nutrients as well as the receptor producing the antimicrobial metabolites, and in turn they are responsible to activate the immune system. And the present study, the lactobacillus plantarum, whatever we isolated and used here, could be used as a potential probiotic strain to control the colorectal cancer. However, it is just, since it is a preclinical study, we can say that, however, the successful translation of these preclinical strategies into clinical practice will depend upon the outcome of clinical trials. So therefore, this strain should be whatever strain we isolated and identified. This strain should be subjected for further tests so that finally the organism can be authenticated and it can be used in the treatment of colorectal cancer. So these are my references, and thank you.
Thank you, sir. That was a very good presentation.
Dr. Nanu,
Yes.
it was a good presentation.
Oh, thank you, ma'am.
Yeah. May I ask you one question?
Yes, ma'am.
So, how long did you treat DMH? Like, what was the treatment time to induce colon cancer?
Pardon, ma'am?
How long you had to administer DMH to develop the tumors?
So that we induced for 1 month, ma'am.
Is it? Okay.
Yes,
Okay.
ma'am.
Okay. We did in rats. It took almost 3 months for us to get the cancers in rat, colon cancers. That's why I just asked you.
Okay.
Thank you.
Yeah.
And also, you mentioned about antioxidants, beta glucuronidase enzymes, all those things.
Yes,
So,
ma'am.
what was the sample used? Whether it was the tissue sample or
Tissue sample, ma'am. Liver tissue.
liver you assessed, is
Yes,
it?
sir. Liver tissue has been used. Not the blood, liver tissue has been used here.
Oh, you didn't try anything on the intestinal part?
No, ma'am. No, not from the intestine. Only liver tissue has been used for this.
Okay, you did only histopathology of intestine.
Intestine, yes, ma'am. Yes, ma'am.
Okay. Okay, then thank you very much for your presentation.
Okay, thank you, ma'am.
The second presenter is Vishnupriya from Bharatiyar University. Ms. Vishnupriya,
Yes,
are you ready?
Yes, ma'am. I'm ready.
Please share your screen.
Good afternoon, ma'am. Shall I start now?
Yes, please.
Okay, the title of my research is Enhanced Therapeutic Efficacy of Sorafenib in Combination with Bacoside A in U87MG glioblastoma cell line. Glioblastoma multiforme is the most common and the aggressive disorder and which comes under the grade 4 of glioma, which is approximately 60% among the 17,000 cases diagnosed cases each year. However, the progression-free survival of glioblastoma is only 14 months. Surgery, radiation, chemotherapy are the major standard of care treatment for glioblastoma multiforme. The major drug that is used for the treatment of glioblastoma is temozolomide, but due to resistance and severe adverse effects, the use of temozolomide is not much preferred. And sorafenib is another oral multikinase inhibitor, which is also used for the management of glioblastoma. However, its limited efficacy owing to the drug resistance is a matter of concern. And the overexpression and mutation of EGFR play a critical role in tumorigenesis and resistance to treatment of glioblastoma. So in this study, we are taking Bacoside A, which is isolated from Bacopa monnieri, which shows anti-cancer activities in various cancer cell lines. The present study investigates the effectiveness of Bacoside A sorafenib combination on glioblastoma cell line U87MG and also the underlying mechanism. This is statement of problem. The main reasons for glioblastoma resistance include highly invasive nature of tumor cells, resistance to chemotherapy, and existence of blood-brain barrier. Despite some minor improvements in progression-free survival, no obvious increase in survival has been associated with any particular regimen, including temozolomide. Even the sorafenib is also used in combination with temozolomide. However, the sorafenib also, the complexity of angiogenesis suggests that sorafenib cannot block the formation of tumor microvessels completely. The combination of sorafenib with Bacoside A targeting a parallel or vertical signaling pathway has not been reported so far. And sorafenib, the systemic administration was well tolerated and crossed the blood-brain barrier effectively. Unfortunately, it's used in the clinical treatment of patients with malignant glioma.
Vishnupriya, may I interrupt now? Can you please present your results, objectives and results?
Okay, ma'am. scientific objectives: evaluation of cell viability in glioblastoma cell line U87 Mg, exclusively using bacoside A and in combination with sorafenib by SRB assay, then to determine the underlying molecular mechanism of the combination regimen in GBM cell lines. Coming to the results, we first check the cell viability of U87 Mg cell lines after treatment with different concentrations of sorafenib and bacoside for 24 hours. Here you can see that there is a dose response decrease in the cell viability when treated with sorafenib and bacoside. The IC50 of sorafenib was found to be 10 micromolar and the IC50 of bacoside A was found to be 22 micromolar. Then the experimental design, we have for further studies, we have divided the cells into 5 groups. Group 1, control without treatment. Group 2, bacoside A IC25. And group 3, sorafenib IC25. Group 4, combination of bacoside A and sorafenib. Group 5, sorafenib IC50. Here, there is almost a 50% of decrease in the cell viability in the combination treatment when compared to individual treatment groups alone. Individual treatment groups, you can see the inhibition is almost similar to the IC50 of sorafenib, and we then constructed the isobologram of bacoside sorafenib combination, and we have got a synergistic effect. Here the combination index value is less than 1, and we can say that bacoside induces has a synergistic effect in combination with sorafenib. And the morphological changes by light microscopy and SEM. And here the morphological changes indicates that combination exhibited significant cytotoxic changes, such as cell rounding and floating cells. Then the combined regimen of bacoside and sorafenib, it enhances the LDH release in U87 Mg cells. Then when we check the disruption of mitochondrial membrane potential using BDFAX flow cytometer, you can see there is a shift in the peak when compared to the control. In the combination is represented by a blue peak, and there is a much shift from the control. Control is represented by pink color, and the blue peak, which is almost, that is combination peak, is similar to the IC50 of sorafenib, which clearly indicates that there is a disruption of mitochondrial membrane potential during the treatment. And we also check the viability of the cells after treatment using PI uptake assay. And there is the percentage of dead cells in the combination group is almost 55%, and which is similar to that of IC50 dose of sorafenib. Then we analyzed the cell cycle, and the figures clearly mention that the arrest occurred at G2M phase of the cell cycle. Next, we analyzed the ROS generation in the presence and also in the presence or absence of NAC inhibitor, and the treatment. The ROS production was peaked at 45 minutes when peaked at 45 minutes and further declined at 1 hour. Then we used N-acetylcysteine, which is an ROS inhibitor, and in the presence of ROS inhibitor, the ROS production was significantly decreased. Next, we determined the apoptotic cells and the necrotic cells using annexin 5 FRTC PI staining using BDFAX flow cytometer. The combination treatment showed apoptotic cells which are almost similar to the IC50 of sorafenib. Then we performed a tunnel assay to confirm the apoptosis using ApoBRDU assay kit from BD Pharmingen, and the Thorafenib Bacuside A combination treatment showed 45% of tunnel positive cells when compared to individual treatment groups. Then coming to the conclusion, Bacuside A and Sorafenib synergistically inhibited glioblastoma cells in vitro, resulting in a more pronounced induction of cell death than monotreatment with either substance alone, as evident from cell morphology as well as SRB assay. Then cell cycle arrest occurred at G2M phase. The LDH MMP PI uptake assay, Axif5 FRTC PI staining, and tunnel assay further confirmed the results. ROS was more pronounced at 45 minutes and then declines. Treatment with NAC further inhibited the ROS generation. Then significance to the field of research, proposed regimen would increase the localization of sorafenib, thereby reduces the dose requirement, recurrence, and also help alleviate sorafenib associated toxicity. Development of targeted therapy using this combinatory regimen in animal models. Then these are the references. Thank you.
Thank you, Vishnupriya. Madhu sir, do you have any questions for the candidate? Madhu sir?
Yes. So just to get an idea, what is the incidence of this, glioblastoma in India? How severe this is in India?
Vishnupriya, are you answering the question?
I think she left.
Maybe
Is she
connectivity issues are there. I think she's there,
Okay.
but
Maybe you
she's
may
not.
go to next speaker then. Yeah, you can go to next speaker.
The next speaker is Soundarya R from Bharatiyar University. Soundarya, are you there?
I think we can move on to
there in the participant list. I have made her the co-host, but you just have to call her, right? Soundarya? Yeah.
She has started.
Soundarya, we can see your feet. You can put it on screen. Yeah, but it's not very clear. You can just go a bit near the mic and then you can talk. Very feeble, Ma. Your voice is very feeble. And put your presentation in the full screen mode, and then you can start.
Dr. Rukmini?
Yes, ma'am. Can we go to the next?
Can we? Yeah, I think
maybe
she's got
we'll
some.
give some time for Soundarya to get ready, and we'll go to the next speaker. I guess the next speaker is Divya Sangeeta. Divya, there are so many Divyas in the participants. Can you just show the raise hand option? Yeah, very good. Okay, I will just make you the co-host. Thank you, Divya.
Maybe.
Yeah. So the next participant will be Divya Rajkumar or Divya Sangeeta. Yes. And Soundarya, we can have your presentation later. Can you share the screen?
Ma'am, this is Divya. Am I audible?
Yes, you're audible. Fine.
Ma'am, is it in full screen?
Yes, it is.
Yeah. Shall I start, ma'am?
Yes.
Good afternoon. This is Dr. Divya from Madras University. My topic is on the interaction of enamel proteins with endocrine disrupting chemicals, leading to degeneration of tooth enamel and in silico approach. So basically, the crown portion of the tooth consists of three parts: enamel, dentin, and pulp. The process of this enamel formation is termed as amelogenesis, which basically occurs in the last third of the fetal life to up to 4 to 5 years of age after birth. And the cells which are involved in this amelogenesis is termed as ameloblasts. So basically, these ameloblasts secretes various proteins, which includes matrix proteins, which are amelogenin, enamelin, amylotin, ameloblastin, and tuftelin, and also proteases, which includes kallikrein-4 and MMP20. So basically, what is the process is these ameloblasts secretes the enamel matrix, and the proteases degrades this matrix, followed by crystal deposits. Thereby, there is enamel formation. Molar incisor hypomineralization is a developmental enamel defect where the mineralization content is reduced, leading to discoloration and fracture of the affected tooth. So clinically, it is seen as white opaque spots on the enamel of first permanent molars and incisors. So the prevalence rate is around 15 to 18% in children of 6 to 9 years of age. Next is endocrine disrupting chemicals. Basically, the endocrine system consists of various glands which produces hormones which reaches the bloodstream, and to the target organs, it binds with the receptors and there is cellular response. So what these chemicals does is it disrupts this endocrine system by mimicking the endocrine hormones. Therefore, the cellular response is disrupted. So the sources of these hormones common is pesticides, food additives, medical products, etc. And there is around 58,000 of these chemicals which has been identified. The common ones includes bisphenols and phthalates. So coming to the health effects, whenever these chemicals we are exposed, the human body is exposed through oral, inhalational or transdermal routes, there are various health effects which are seen, like metabolic disorders like diabetes or endocrine disorders like and cancer. Ma'am, can I continue?
Divya, actually, I can see the echo of you. Why don't you switch off or move another echo which is nearby?
Ma'am, I'm not able to hear you. Ma'am, shall I continue? Ma'am?
Yes, it is okay now. You can continue.
Yeah, okay, ma'am. So the common endocrine disrupting chemicals includes bisphenol A and phthalates. They are basically used as plasticizer in the production of plastics in order to increase the flexibility. So the tolerable daily intake for bisphenol is around 4 microgram and for phthalates is around 20 microgram per kg body weight per day. So what is the key problem which it has been addressed is these endocrine disrupting chemicals, they target the enamel proteins, mainly kallikrein-4 and enamelin. They modulate their expression, thereby leading to molar incisor hypomineralization. Since amyloblasts, the cells which are responsible for enamel formation, are also susceptible to these EDCs, the tooth itself can constitute as a novel marker for the exposure of these chemicals. So, though there are many in vitro studies and animal studies being performed, here we have done an in silico docking study in order to identify the interaction between the chemical and the enzyme. So the results of the docking after the protein and ligand preparation, this was done using Autodock software, where this was docking of callicarin 4 with bisphenol A. Here we have found there was an interaction between the ligand and glycine at 193rd position of the enzyme, and the hydrogen bond distance was around 3.1 angstrom. And on docking callicarin 4 with phthalate, we found the interaction was between the ligand and serine 195. So callicarin is basically a serine protease, and the catalytic triad at the active site of this enzyme includes serine 195, histidine at 57, and aspartate at 102 position. So what we have found is that these EDCs have a good binding energy and low inhibition constant. Thereby, it shows the effective inhibition of the enzyme at the active site, thereby leading to the hypomineralization of the tooth. So to conclude, this data has given an insight on the mechanism of the structure-activity relationship at the active site of the enzyme, and this can be exploited to design better therapeutics. Thank you.
Thank you, Dr. Divya Rajkumar.
Thank you, Divya, for a very nice presentation.
Thank you, ma'am.
I think we can move on to the next speaker.
Thank you, ma'am.
The next presenter is Dr. Pon Saravanan from Pondicherry University.
Yes, Dr. Pondicherry, I have made you the co-host. You can share your PowerPoint and then you can present.
Hello, am I audible? Can I start?
Yes, please. You're audible.
Yeah, thank you. Good afternoon, everybody. I'm Dr. Ponni, and for the next few minutes, I will be talking about my research work on KDM6B, which is a lysine-specific demethylase enzyme. So before moving into the results, I would like to give a brief introduction about the research area. So the research area is epigenetics. Epigenetics is inheritable changes in gene and protein expression without any changes in the DNA sequence. And the genes are turned on and off by modifications of the DNA and the histone tails. Since the function of every cell is determined by the genes and protein it expresses, epigenetic alteration can be considered as the kingmaker, because regulation of gene transcription is the key biological determinant for cellular differentiation and function. So moving on to the modification of the histone tails in DNA, there are certain set of proteins which create this modification called as the writers, and which are capable of removing this modification called as the erasers, and a set of proteins which can recognize the pattern and elicit further signaling. So there are many enzymes that are involved in the modification of the histone tails and DNA, and they have been attractive targets for the epigenetic therapy, especially cancer, and they have been altered in all disease conditions. So among these readers, writers, and erasers, there are one particular family of proteins which are least studied and is the interest of our study, is the Jumanji C family proteins. So I will be concentrating on KDM6 protein, which is a lysine-specific demethylase. The function of this protein is to remove the methylation marks and to enhance gene-related transcription and promote tumor pathology. So the need of the hour is to inhibit this demethylases protein so that the transcription is kept in an off position and so that the tumor is repressed.
So moving on to the work strategy. Initially, through structure-based virtual screening, we identified certain compounds and we ranked those compounds and took the top 5 compounds to biological characterization, which included cellular cytotoxicity, apoptosis, cell cycle, gene expression, and trimethylation status of the histone proteins. So as you could see here, the top ranked five structures are represented here. These had better binding score when compared to the reference compound, which is GSK J1, with minimal interacting residues than the reference compounds. However, these 5 compounds were selected for the study and deemed for further evaluation. In this slide, you can see the positioning of the compounds within the active site of the KDM6B protein. The red color is the reference compound and the blue colors are the candidate molecules. So moving on to the cellular cytotoxicity, we observed cytotoxicity of these molecules at higher concentration, and the IC50 were calculated respectively. Among the 5 molecules, we took 2 molecules with lower IC50 values and deemed them for further evaluation. As expected, these molecules induced apoptosis in HeLa cells at a concentration of IC50 concentration after 24 hours of exposure. So complementing this, we also observed an accumulation of cells in the G0, G1 phase of cell cycle, which is complementing with the apoptosis assay, indicating that these cells are retained in this phase without progression when treating with these compounds. And then we did a gene expression analysis where we found that these molecules increase the expression of P21 and P53, which inhibited cyclin B1 expression and blocked the cell cycle at G0 phase. We also checked the target genes HOXA5 and FOXO3, which were reduced compared to the control cells. And Western blotting detection of trimethylation levels of histone 3 lysine 27 revealed that these molecules demonstrated elevated trimethylation status of the histone proteins. So to consolidate the present research, in different environmental conditions, this upregulation of KDM6B protein is elicited due to several signaling pathways like MAPK, NFKB, STAT6 signaling pathway. And this upregulation leads to the initiation of transcription and tumor progression. So the objective of this study is to block this JMJD3 or the KDM6B proteins and to keep the transcription in the off status so that the tumor is being repressed. And to summarize the present research, through structure-based virtual screening, we identified 5 molecules, among which 2 molecules qualified as prospective candidates for anti-cancer therapy. And since these molecules are just fragments, these could be considered as novel scaffolds to design and develop future potent KDM6 inhibitors with implications in epigenetic therapy. And to conclude, this research strongly recommends to prioritize research on KDM6 inhibitors, as these are till date very less focused, and the usage of KDM6 inhibitors might be helpful to suppress the effects of KDM6 protein overexpression in clinical malignancies, especially in cervical cancer. So that's all. Thank you.
Thank you, ma'am. So the next presenter is Ms. Atira Ramesh from Pondicherry University.
Hello everyone, I hope I'm audible and my screen is shared properly.
Yes, Atira, you can proceed.
Thank you so much. So today I'll be discussing, good afternoon all. Today I will be discussing about circadian rhythm or the biological clock and its regulation with a small non-coding RNA binding protein, PV. So what is circadian rhythm? That is the 24-hour rhythm that is generated from the suprachiasmatic nucleus of our CNS that will be generally regulating the physical, mental, and behavioral changes as a daily cycle. And it is endogenously a regulated thing, but with the help of some external cues from the environment, especially sunlight and temperature. So the sunlight will be entered through the retina of our eyes and it will be directed to suprachiasmatic nucleus, that is CN, in our brain, and output will be the physiological and behavioral rhythm which is coordinated by our system. So it is modulated by some pacemakers, especially some proteins present in this clock neurons present in our SCN. So I'm going to explain the possibility of the regulation of the system with the help of PB protein. So what is PB protein now? It is first discovered in Drosophila, which is mainly present in germline cells that helps in the maintenance of germline cells and its self-renewal. In humans, there are 4 PB isoforms, L1, L2, L3, and L4. And the model system which I am going to present is Drosophila, and it is having 3 PB isoforms, PB, AGO3, and aubergine. So these PB proteins will be binding to a small non-coding RNA, piRNA. And our rationale is like we have seen PVL4 is getting downregulated when the timeless protein, which is a circadian protein, when the PVL4 is downregulated, circadian protein timeless is also getting downregulated in transcript level. So in 2017, you et al. Have represented that through GSK pathway, another circadian clock protein is getting ubiquitinated, and it is suppressed by the inactivation of PVL2. So what we are trying to answer is whether PV is having any role in the regulation of central circadian rhythm. And my approach is through Drosophila as a model. So this is the circadian circuit in Drosophila, where this 150 neurons are present, which is a stringent and closed circuit, and the proteins present in this will be regulating the entire system. And that regulation is through transcriptional translation loop of which of the gene expression of especially period gene, timeless gene, clock gene, and cycle gene within the nucleus and in the cytoplasm. So the materials and method I used for the study is like I used behavioral and locomotor assay using the splice strains, RNA lines, PV, AGO3 and aubergine, and driver lines clock 856 Gal4, which is procured from Bloomington Drosophila Stock Center, USA, and immunocytochemistry of Drosophila brain. So I will explain how the behavioral assay is executed. The 5 mm glass tubes will be taken in which I will be keeping food, and the Drosophila will be kept inside that, and I will be plugging in that, and that will be setting in a Drosophila activity monitor, DAM monitor, and in one monitor there will be 32 flies of same genotype, and that will be kept in either dark cycle where 24 hours darkness and light dark cycle where 12 hours darkness and 12 hours light will be provided. This system will be kept inside a perceval, which is kind of an incubator, and the activity will be monitored. So what happens inside is that, like in the middle of through the middle of the tube, IRA will be transmitted. So each time the flies will be passing this IRA, one bout activity is calculated, and it is monitored in the computer, and later on that is analyzed through MATLAB software. So we will be getting an actogram which represent each activity as an activity bout. So as I said, I used whole body mutants first for my study, which will be giving 3 main set of data. So power of rhythm, period, and rhythmicity I will be calculated. So in this case, the period is the important thing is period, which will be calculated by the periodogram represented, which I'll be getting through the MATLAB analysis. So this period length is the expression time of genes which is present in the transcription translation loop. So again, power of rhythm is the stability of the rhythmicity exerted by each genotype of the flies, and rhythmicity is the percentage of flies which will be in rhythm, in rhythm in the system. And that is for DD analysis where 24 hours darkness was given, and for LT analysis where 12 hours darkness and 12 hours light is given. We will be able to find normally in a wild type 2 peaks, that a morning peak and evening peak. But in the case of our mutant flies, PB, ago and aubergine mutants, that morning and evening peaks are not present. So that will be, and that was a interesting data. So we further did RNA-I lines, took RNA-I lines for our studies. So what I have done in this is, I have downregulated PB protein and AGO3 in 150 specific clock neurons in Drosophila brain with the help of clock 856 Galford driver. So these first two are PV RNAi controls, then these are driver controls, and this is the target. So we can clearly identify the arrhythmicity in the expression level, activity level of this flies. So normally it should be having a morning activity and evening activity, but it is totally arrhythmic in the case of mutants. This analysis is across 3 experiments, and PB is showing very least rhythmicity, and the power of rhythm, that is stability of the rhythm, is also very low in PB mutant flies, and also the period length is seen to be 17 to 26 hours, which is much lesser. And then it is LDSS. So in the LDSA also the same type of flies are used, where the PV and AGO3 is downregulated in 150 neurons. These are the experimental lines where we can see the PV mutant flies are having high activity throughout the day. So normally during the night the activity should be less, which is not happening in the case of PV mutant. So
Adira,
yeah.
sorry to interrupt you.
Yeah.
You are exceeding your time. It's going to be 8 minutes now. Could you please summarize?
Yeah, sure, ma'am. So here in the case, we can see that PV mutant flies are having high nighttime activity, and later on I checked the differential expression of PV protein in brain, where I could find in different time points the PV expression level is varying, and especially in a particular ZT14. The expression of PV is visualized or visible in one of the specific clock neuron, DLN, and also DNs, and some of the non-clock neurons. So to conclude, we could find PV rhythmicity in PV downregulated cells, flies, and also high nighttime activity in the LD condition. Also that represents there might be some role of PV in the sleep rhythm or sleep cycle. So our future perspective is to elucidate what is the molecular mechanism behind this regulatory action, and how peripheral and central circadian rhythm are interconnected. So I would like to thank my PI and Dr. Shiba Vasu, who helped me to do the experiment, and eminent scientists who helped me to get the antibodies for different experiments. Thank you.
Thank you, Ms. Adira.
Good. It was a good presentation, Adira.
Thank you, ma'am.
We can move on to the next speaker.
The next person is Anuja from JIPMER.
Adira, you can stop the screen share so that Anuja can share. Adira? Yeah, Anuja, just wait for a few minutes. Adira, are you there? Can you stop the screen share? Okay, thank you. Anuja, now you can share your screen and start presenting.
Yes, ma'am. Yes, ma'am. Ma'am, is it visible, ma'am?
Yes, ma'am. Now it's visible.
Okay. A very good afternoon to faculties and delegates online. So I'm Anuja Anandraj from PhD scholar from Department of Clinical Pharmacology, JIPMA. So working under the guidance of Dr. Sandhya, I today present a part of my thesis which is entitled Frequency of VDR Gene Polymorphism, especially the FOC1 polymorphism and its association with serum 25-OHD levels among South Indians. Serum 25-OHD is basically serum vitamin D levels. So at the outset, I'll be discussing a bit of background, the objectives of my study, materials, methods. Ma'am, just 1 second, ma'am. So at the outset, I'll be discussing about the background, the objectives of my study, materials and methods, results, discussion, and finally the conclusion. So basically, VDR gene is nothing but the vitamin D receptor, which is located in chromosome number 12, Q arm. So it spans approximately 11 exons and approximately 75 kb of genomic DNA. It's basically a nuclear membrane protein. Vitamin D, which is known as cholecalciferol or serum 25-OHD, binds to this particular VDR receptor and it proceeds with the downstream metabolic processes. So there are various studies. Basically, this vitamin D receptor, which is located in the gene, vitamin D receptor gene has polymorphisms like APA1, BSM1, TAC1, FOC1. So among these 4 polymorphisms, which I have focused to present here is FOC1 polymorphisms of the VDR gene as in the South Indian setting alone and how it is associated with the vitamin D level. So there are various studies globally which has been published with conflicting results saying that conflicting results that whether there is any association with this polymorphism with that of vitamin D levels. So that also will be discussed in the upcoming slides. So to begin with, the primary objective is to evaluate the association of the vitamin D receptor gene, that is the FOC1 polymorphism, with serum vitamin D level. So from the healthy volunteers from South Indian setting. And my secondary objective is to compare this data, the frequency of the vitamin D receptor gene polymorphism of the South Indian setting, to compare this data with that of the other ethnic populations. That is the other polymorphic frequency of the FOC1 polymorphism with the other ethnic populations. So basically it is a human study. So Institute Ethics Committee approval was taken and informed consent was taken from 199 participants, that is the healthy volunteers. And then 5 ml of blood was collected, among which 2 ml was used for the serum vitamin D evaluation and the remaining 3 ml was used for the genotyping by RT-PCR. And then the statistical analysis was done to find out the association between the serum 25D level with that of the frequency of FOC1 polymorphism. And then, as we all know, p-value less than 0.05 is considered to be statistically significant. So moving ahead to the results of the primary objective. So as I already mentioned, whether there is any relation or association between the FOC1 polymorphism and the levels of vitamin D in a South Indian setting was my primary objective. So we came out with the results like this table projects the baseline parameters out of 199 healthy volunteers here. So out of 199 healthy volunteers here, we have split accordingly like deficient, insufficient and normal. So vitamin D level less than 19.9 nanogram per ml is considered to be normal. 20 to 29.9 is considered to be insufficient and more than 30 is considered to be normal vitamin D. So among 199 healthy volunteers, only 62% were deficient with vitamin D, 27% of healthy volunteers had insufficient vitamin D levels, and only 10% among 199 healthy volunteers had normal vitamin D level. This is according to the South Indian scenario. And these are the other baseline parameters which we collected like age, gender, BMI, and the mean serum vitamin D levels. So what is the objective? So moving ahead to the results of the primary objective is whether there is any association between this VDR FOC1 polymorphism and vitamin D level, and this is what the results we came up with. So it is tabulated like this is the genotype of the FOC1 polymorphism, the AA, AG and GG. This is the wild genotype, and these both are the homozygous mutant and heterozygous mutant. So among this 16 number, that is 8% of the South Indian population of 199 had AA genotype, 26% had AG genotype and 65% had GG genotype. Among the 8%, 30 number of people were deficient with serum vitamin D, 1 had insufficient vitamin D and 2 were normal vitamin D, and those were of this particular AA genotype. So among the 26% of AG genotype, 32 numbers had deficient vitamin D levels, 19 had insufficient and only 2 had normal vitamin D levels. And among 65% of GG genotype of healthy volunteers, 79 numbers had deficient vitamin D level, 34 had insufficient and only 17 had normal vitamin D levels. So what could we conclude from the primary objective is that there was no significant association between the FOC1 polymorphism and the levels of serum vitamin D among the healthy volunteers in the South Indian setting. So thus we achieved the primary objective. So moving ahead to the secondary results of the secondary objective is whether from this data collected, that is the frequency of the FOC1 polymorphism from the South Indian setting, from that data which we collected, we compared to the frequency of this particular FOC1 gene in other ethnic populations. That is the South Indian VDR gene frequency comparing to that of the other ethnic populations. So how this worked was, we came up with the results, like this was a tabular column. So we found that the frequency of the FOC1 polymorphism in the South Indian setting is found to be significantly different when compared to other ethnic groups. So other ethnic groups which we included were the Northeast Indians, the Africans, Americans, Eastern Asians, Europeans and South Asians. These were the other ethnic groups to which we compared the frequency of the FOC1 polymorphism from the South Indian setting with them. And we found that there was a significant difference in the frequency of this particular polymorphism between the South Indians and the other other ethnic population. Thus, we achieved the secondary objective also. So, from the at the outset, like among 199 healthy South Indians, so we found that 61% of them had a deficient vitamin D level, 27 had insufficient vitamin D level, and only 10 of them had normal vitamin D levels. And the frequency of the VDR gene, that is especially the FOC1 polymorphism which we studied in healthy volunteers of South Indian setting, the wild type 65% had wild type genotype, AG, that is the heterozygous mutant, was 26% and homozygous mutant was 8%. And in the secondary objective, the frequency of the polymorphism, the FOC1 polymorphism, was found to be significantly different, in South Indians when it was compared to other ethnic. So to conclude, FOC1 polymorphism is not associated with the vitamin D level. This is in accordance to the South Indian setting, and the frequency of the polymorphism in South Indians has been established and is found to be significantly different when compared to the other ethnic groups. Thank you so much.
Thank you, Ms. Anuja.
Thank you, ma'am.
Yeah, we can move on to the next presenter.
The next presenter is Priyadarshini Santakumar from Bharatiyar University.
Ma'am, shall I start, ma'am? Hello?
Yes, yes, please start.
Good afternoon, myself Priyadarshini. I'm a PhD scholar in Bharatiyar University, Department of Biochemistry. I'm here to explain part of my work about liposomal gingival-induced apoptosis and G2 MPS arresting lung cancer cell line. The introduction. Lung cancer is one of the most leading cancer in the worldwide and in India. Smoking is a major cause of Ma'am,
Actually, not hearing your voice properly.
Ma'am, I can't hear you properly, ma'am.
You have switched on multiple devices.
Ma'am?
Have you switched on multiple devices? Because we can hear it.
Ma'am?
Yes, Priyadarshini, can you hear me?
Yes, ma'am.
Have you switched on multiple devices at your end?
No, ma'am.
Okay, now
lung cancer remains the leading cause of cancer.
Actually, it is not clear.
Priyadarshini,
yes, ma'am.
I think two candidates sitting in the same room, switching on your devices.
No, ma'am.
Yeah, we could hear a lot of noises, background noise.
Background noise.
If you have switched to two devices, mute one device.
Yes, ma'am.
Are you presenting from your mobile phone?
No, ma'am. In the system only, ma'am.
Okay. Your mic maybe. Okay, you go with your objectives.
Okay. My main, ma'am, since I chose gingerol to treat the lung cancer. It's a potent phytochemicals derived from ginger, and due to its antioxidant and anti-tumor promoting activity, I select this gingerol to treat lung cancer. But like paclitaxel, the commercial anti-cancer drug, the gingerol also insoluble in water, which limits its application. To facilitate the administration of 6-gingerol into the lung cancer cell line, I encapsulate the gingerol into a liposome formulation, which is a promising drug delivery system for nowadays. So, in my study, is the main aim of my study to encapsulate this gingerol into the liposomes to facilitate the lung cancer treatment. This is my workflow. It's a dilauryl phosphatidylcholine, the lipid I used. It is an amphithetic lipid which possesses both positive and negative charge. It is a neutral and it is mixed with cholesterol and gingerol. Using thin film hydration method, I prepared the liposomes and surface morphology and charge of the liposomes is determined by SEM and DLS, and the functional group of the liposomes and both the gingerol is determined by FTIR. And the in vitro release study also I performed, and the lung cancer activity effect of gingerol and liposomal gingerol on lung cancer treatment is determined by MTT for fixation of the dose, and to determine the mechanism of the gingerol for lung cancer treatment, I did apoptosis and cell cycle and new cell analyzer. So, preparation only I will tell in the decline, ma'am. Other procedures I will explain in while the results. For the preparations, the lipid and the drug ratio, I increase the lipid ratio to as the gingerol is lipophilic drug, it increases the as lipid increases. So I increase the lipid ratio, change the lipid ratio to optimize the liposome preparation, ratio for optimized liposome preparation, and vesicle entrapment of cisgingerol is determined by encapsulation efficiency. It is analyzing by using UV spectrophotometer at 282 nanometer, and same for morphology study, and size distribution was determined by DLS, and theta potential also determined in the DLS instrument itself, and Fourier transform infrared microscopy spectroscopy is to analyze the functional.
Please give your results only.
In the encapsulation efficiency, as I increase the drug lipid ratio, since my gingerol is a lipophilic drug, the entrapment efficiency, the entrapment of gingerol increases when I increase the lipid ratio. So I found 1:20 ratio will be the optimized liposome ratio preparation. It achieves 98% entrapment of gingerol. And upon same, the average size was found to be 166 nanometer at 1000x magnification. It's spherical shape, so which facilitates the EPR effect. The liposome can directly enter into the leaky nature of blood vessels, and it directly reaches the human microenvironment. And average size distribution was analyzed by DLS. The average size was found to be 189.4 nanometer. And zeta potential, the normal value of zeta potential for stability of nanoparticle is minus 30 MB to greater than 30 MB. The gingerol liposome versus minus 19.1 MB, so which indicates the stability of nanoparticles in optimized range only. And this is the peak for TIR got from for free liposome. The significant peak for C-double bond and overstretching and phosphate group for phosphatidylcholine and acyl chain, the tail of phosphatidylcholine liposomes for obtained for free liposomes. And this is the 6-gingerol peak. C-double bond on C-stretch was noted. And in the gingerol liposome loaded gingerol, the both free liposome and the significant peak of the 6-gingerol also appear. So it is indicated that the 6-gingerol is entered in the liposomes. And release profile, it is compared with free gingerol and liposome. Both are mixed with methanol and put in the dialysis bag, and in the methanol medium, the release is noted. For the free drug, it shows the burst release. Upon 10 hours, it is fully released, but liposome shows the sustained release, which indicates it increases the
Priyadarshini, can you please conclude, because you are exceeding your time?
Okay, okay. In the apoptosis also, it shows when compared to control, the lysis population is very less, and compared to free drug, it is late apoptosis is increased, but in the liposome drug, early and late apoptosis will indicate the sustained release. In the cell cycle also, G2 M-phase substrate was noted. I'm concluding that the liposomal gingerol is more efficient when compared with free drug. It could be a promising drug for lung cancer treatment. Thank you, ma'am. Thank you, ma'am.
Thank you,
Thank
Priya.
you.
Yeah, I think we have one more participant. Soundarya.
Miss Soundarya, are you there?
Her name is there, but it's not showing the mic or the camera. So
maybe we can make her present in the next session.
Maybe, yeah. So in case if she comes back, then we can ask her to present in the second session at the end.
We would like to thank all the participants for their enthusiastic participation in the oral presentation session 1. The results for the oral presentation of various sessions will be announced during the valedictory ceremony. Now it's time for lunch break. We'll resume here at 2 p.m. And the participants of e-poster presentation are asked to log in 10 minutes before the start of the session. Thank you.
அதுக்கப்புறம் நான் இன்னொரு வீடியோ போட்டு 2 மணி வரைக்கும் ஃப்ரீ ஏதாவது பேசணும்னு கேட்க இந்த இமெயில் செக் பண்ணிட்டு எதாவது பண்ணுறதுனா பண்ணுறேன்.
What if you could have a flow cytometer, but without having to worry about waiting for warm-up, calibration or cleaning? Meet the nuclear counter NC3000 from Kilometer. From single cell counting to cell cycle, apoptosis and custom designed assays, the NC3000 does it all in less time and with a small footprint. For high-speed cell counting, the Bio1 cassette provides a one-step cell count without pre-treatment. The slides give you the flexibility to perform up to 8 cell counts in less than 3 minutes. The list of advanced assays includes cell cycle, vitality, GFP transfection, mitochondrial potential, DNA fragmentation, and XM5 and caspase assays. If that's not enough, there's FlexiCyte. FlexiCyte is a flexible cytometry platform that lets you build your own customized assay from the ground up, using the cell markers and fluorophores you want. Install the software on as many computers as you like and do your analysis from anywhere. In addition, the NC3000 is 21 CFR Part 11 ready and can be validated easily with the included IQ and OQ assays felt at home in both academic and R&D labs. If you need a powerful, flexible cytometer that offers high speed, precise cell counting, predefined ready-to-use assays, custom assays, and unlimited software licenses, all with no maintenance on a footprint smaller than your centrifuge, look no further than the Nuclear Counter NC3000.
Capture publication quality images without the hassle of a manual microscope. The cell imager you've been waiting for is finally within reach. Introducing the Image Express Pico automated cell imaging system from Molecular Devices. Automated acquisition plus intelligent analysis integrated into a single configurable system. Whether running single slides or up to 384 well plates, the Image Express Pico is designed to minimize manual effort and increase efficiency. Capable of bright field and fluorescence imaging, the Image Express Pico comes standard with temperature control and optional environmental control for live cell imaging. Unlike other imaging solutions, the Image Express Pico automates both acquisition and analysis of cell images. With pre-configured cell imaging protocols optimized for specific assays, ranging from simple cell counting to sophisticated neurite tracing, getting started is a snap. Each pre-configured protocol is optimized to collect the most pertinent results and to ensure accurate segmentation. The Image Express Pico automatically adjusts focus and exposure for consistently brilliant images. From a whole plate overview to an individual cell, you can visualize your images across multiple scales to gain key insights. Now you can use a single software package to acquire and distill images into data on the fly with our powerful Cell Recorder Express software. Generate heat maps, scatter plots, tables, bar graphs, and even movies with just a few clicks. And because access is the heart of functionality, our user-friendly Cell Recorder Express software is browser-based, so you can operate the system anywhere, from any computer with a network connection. The Image Express Pico is the intelligent answer to image acquisition and analysis. Are you ready to give up guesswork and tedious manual labor? Ready to embrace increased throughput and unmatched efficiency? Now you can. Image Express Pico by Molecular Devices.
So we had two community colleges. One was in Pondicherry itself. Second,
Dear Sir, on 29th November 2017, you took charge as the Vice Chancellor of Pondicherry University. As you complete 2 years of your tenure in Pondicherry University, you have taken the university in new directions of higher education and shaping the academic and research ecosystem of the university. The university is now reaching higher levels of ranking on the world global plane, and I'm sure that after the last documentary that we had, in which we documented on your achievements in the first year as Vice Chancellor, we are keen to know on where the university is headed to in the second year.
well, I'm glad you have said that we have already documented the first year. If you remember, last year we had focused on three things when I joined. And it was a very thrilling experience joining Pondicherry University. I had said that my initial thrust will be on three main factors. One was that promotions to be given to all teachers, because if something is not given at the right time, that leads to a lot of depression in one's mind. So that was the first priority I started with. Second one was to streamline the problems of research scholars. They would submit their theses and they will not know what is the outcome of this submission. So I discussed with them and we have drawn a program under which the total period from submission to the bibliography shall be completed in 5 months. And I'm very happy to report that this has taken off very well. People are getting completed their research work, research results within the stipulated period. The third problem was regarding recruitment. The university hasn't seen any kind of recruitment in the last about 7-8 years. So we had these are the three aspects which I plan to look at in the first year. And I'm happy to report that most of the promotions pertaining to teachers we have been able to carry out. The research scholars problems have been looked into. They're very happy, satisfied now. And the working ambience is of course improved a lot. The third, the data pertaining to the recruitment processes was also completed in the first year. And now we are almost ready to go in for the third area where we will be inducting a lot of new faculties. This was what I promised in the first year. And I'm happy to report more or less we have been able to achieve this. When we started rolling into second year, my thirst was on internationalization of the university. Idea was that we should open up our university to various other good universities across the globe. And we did go talk to a lot of other universities. And I'm happy to report that about 45 MOUs we could enter into across the globe. It had universities from all continents. And that has put Pondicherry on an international level in the true sense of the word. So this was one which I thought I would focus and I could do it. I'm happy to report about that. The next important part was there was, I will call it that, there was lack of research enthusiasm. And as you know that we have been having periodic meetings with all stakeholders, which includes teachers, students and non-teaching staff as well. I think once in a period of 2 months, I do call them and have a one-to-one open discussion. In that, I found that the research was not at the right level. So in one of the meetings that I had with all the teachers, I did motivate them. In fact, I encouraged them. I said every faculty member should go in for a project, and I encourage them that they are all brilliant enough. They can, the moment they write one, I'm sure they would be getting one of these projects from any of the funding agencies. Initially, the youngsters in particular were very hesitant. They said that when they gave these projects in the initial years of their journey, their projects would not go to the funding agencies for good 2 years and plus, which was rather disturbing. And I said, no, once you give it, I will ensure that within a span of about 2 weeks, the project shall reach the funding agency. And I am very happy to report that about 90% of the faculty could submit projects, and about 70% out of those were sanctioned also. Of course, the quantum of research projects, the amount of research projects which they got from various funding agencies obviously vary. It is from any level to projects running into fairly high amount. So I'll say from major projects to very major projects they came in our university. And the most satisfying factor in this is that a project called Spark, which people initially were very hesitant to apply because it involved the collaboration of other countries as well. That also I could infuse enough interest in them. And a fair number of people did apply, and I'm very happy to report that about 7 or 8 of the Spark projects were sanctioned to our university. This had put a fresh blood into every faculty member. There is one could get it, why not others? Now the research ambience has improved a great deal. People are very keen to start their work on. And overall, in cutting across the department, if you go anywhere, there is a fairly good level of research enthusiasm, and teachers are working very well. This gives you an immense sense of satisfaction in this area. Because I always believe that unless there is a serious research being carried out in a university, that university shall not progress to a desired level. And the result of this has been that overall, all rating agencies, almost all, have placed our university very high. And Pondicherry University has had a quantum jump in terms of ranking from all agencies. So this gives you a bit of satisfaction. And I think that has been the salient feature of my second year in office. The third thing which I did discuss with everybody was that we need to improve our infrastructure. I could not focus much on this side of it. Then we sat down together, designed what all is required. We made a program of what will be required in which particular faculty, and we designed the needs, and we applied to MHRD. And a lot of projects have been sanctioned, and that focus which was to be on infrastructure is also taking place. So these three things which are added to our corporate life of the university, I think that's very, very nice.
Sir, like you rightly said, that without the right conducive research atmosphere, a university cannot grow. And all the stakeholders, the students, the faculty, the research scholars, all of us who are here, we want a vibrant atmosphere. So how easy or difficult was it for you to put all of this into place and to encourage people, like you said, to write projects on an international level and get collaborations through the Spark, so that we could get more insights and the best of talent from across the world into a university?
As I said, initially, the faculty was not very enthusiastic. The vibrancy was lacking to a fair extent. One of the reasons which I could analyze was that earlier on, whenever they would have written any project in their initial phases of the career, the projects would not have reached the concerned agency. And when a project doesn't reach for a good 2 years, you lose the novelty of whatever agency was putting there. I did ensure that this happened. The project reaches the funding agency, and I had ensured this to all the faculty members that you leave this on me, and we will together as a good team will improve these things, and this ultimately did happen.
And so the MOUs that the university has signed, you said about 45 MOUs with different universities across the globe. How is that going to benefit, again, the students, the faculty, and the university?
I am always of the view that unless you look at the best, you will never endeavor to reach that particular post. So I did talk to students, I did talk to a number of faculty members, and I encouraged them that in case they wish to go abroad for any kind of training or any kind of hands-on experience, or in order to adopt the best practices that the other universities are following, I said that we must interact with them. And interacting with the other universities is not very difficult these days because we have all kinds of media at your disposal. So through email and through other means, people did start talking to other universities. And some of them went abroad also under this. And whenever we talked about MOUs, it had primarily three phases. One was that the MOUs, wherever we signed, it had an encouragement for the students in terms of the students can go there and the students from that university can come there to understand what is the best which is happening there and what we can start there, what can be initiated in terms of that. Second one was faculty exchange, which people have been having earlier also, but this was now introduced on a higher level, on a much better level, I'm called. And third was that when we are interacting with them, it should fructify into something useful. And that useful entity was that they would be able to organize joint conferences. Many departments did after we signed these MOUs. Many departments organized wonderful, useful, meaningful conferences, which resulted in a lot of students going there and students from those countries coming in there. Third part was this joint research project, the way we can put all these things together. It ultimately culminated into a kind of academic exchange, I'll say, on a wider net. Our faculty and the other universities which signed the MOU, they started exchanging ideas in terms of what equipment can be jointly used. These are very high-ended equipment which we do not have it here. And out of these, they could use those also. And this started resulting in something more useful than what is anticipated. In fact, Chandrayaan 2, which was 90% successful, some of our faculty from physics and mathematics were also involved in this. And this has resulted from an opening up of research engines in our university. It's really heartening to see.
That's very wonderful, because I think that's what all of us are looking forward to. An ambience that is encouraging, and where you know that whatever you dream of, it is possible. There's nothing that can stop you.
I'll add 2-3 more things in this. When we were discussing our academic endeavors, there were a lot of courses suggested. So we could introduce some new courses which have been duly approved by all our statutory bodies like the Academic Council and all relevant boards. One course we have started in Physics Department. Another course is started in Earth Science. And the third is on Surface Science in our Nanoscience Department. So it is not that all the conventional courses which are being followed in our university, they are the ones which are being focused on. We have added something new courses also. Some new courses will be more relevant to these subjects, to the growth of these subjects. One more area which I would consider it very much worth mentioning is the kind of courses we started in our community college. All over the country now, there is a feeling that community colleges are not a successful experiment. Where I am one of those who have been emphasizing that community colleges, if given right kind of encouragement, can be a big force in solving our society level problems in terms of creating Skill India resource. And the courses like lab technician, radiologist, upper shoemaking, for example, if I take, there is a shortage in Pondicherry in and around. And these need-based courses of the community, we started in community college. The response has been excellent, and they have made good progress in this. So through this, I have, wherever I could, I have emphasized that community college is a great experiment. And if you look at some of the countries who have done very well in terms of resource generation, in creating skill force. Canada is one, United States is another one, even Singapore is another one where community college experiments have been a great success. So our university had 2 community colleges. One was in Pondicherry itself, second one was Mahe, and they were, I will say, they were on the verge of dying down. I said, no, this is a great thing for the society. So we revived those, and they have started doing very well now. And soon enough, I'll be writing to EDC to give us some special help so that we can take it to a desired level.
I believe the Pondicherry Community College has completed 25 years.
25 years this year. Yes, we are going to celebrate that in a big way, and we want to send a message across that this is one of the best ways to create a force of Skilled India strength. Skilled India force, which is the dream of our Prime Minister. There is no need for everybody to have higher level degrees. In fact, a diploma which can be carried out at community college is the right kind of a thing to develop a force which is required for the society.
So I would like to ask you here that, like what you just pointed out, are there any such ideas in the university that looking at employability and looking at the fact that a large number of young people, they get degrees, but then once they step out of the portals of a university, they may not find placements, or they may not find the niche where they can be, productive and constructive and contribute to the country as a productive and strong force. So is the university going to look at creating new courses for that?
This is what I said. I am of the view that everybody doesn't need to go in for higher degrees. In fact, if somebody has a special talent for doing something useful one can always do a diploma, and this is where our community colleges are going to be coming in handy for the society. They can do, I'll give you an example. There's no need to be an electrical engineer. You can be an electrician, and that diploma can be done in our community college. So once you have that, you are on your own. You can have your own market created, and you can open your own venture. So instead of running around for seeking jobs, one can be a job giver when you have this kind of skill in your hand. And this is where community college is going to be a big force to reckon with. I'm very hopeful that we'll be able to create a huge force which will be serving all sets of all sides of society. This is this results will be visible very soon. In fact, within 2 years, one would be able to know this. In fact, when I started my tenure here, I visited community colleges, and they said this is the way we have been running. Some of the courses that we designed have really been a great help to the society. One I can mention is that these biochemical lab technicians, there was a shortage. Now we are able to send our people to the other states also. So such things can be of a great help. The other area which I'm going to focus this year is going to be in the area of sports. Sports in our university, I don't know for whatever reason, though we have good stadiums, good area, good grounds, but somehow it has been a neglected entity, and sports has been on a very low level. In fact, when I took round, I found that the grounds were in very bad shape. Our stadiums were not being looked after very well. So this is another area where I'm going to focus on. I've requested the Sports Ministry, the Ministry of Youth Affairs, to help us out in improving the conditions of these stadiums and grounds. And then we'll launch certain new programs. I recently discussed with the Secretary of Sports about these aspects of our university sports side. And he immediately came with an idea that we can create a center for excellence for training in athletics. So a team already has come, visited our university for 2 months back, and they are giving a shape to this. Under this program, the coaches will come from the ministry. The coaches will come, they design the program, they will train our university students. The food will be provided by them, the kind of special food which is needed for becoming a good athlete. And the idea is that first year training they will be given, and in second year, in the competitive world, when they enter, they'll become good athletes. And eventually they'll be able to participate at a state and national level. And we hope that some enthusiastic persons out of this will be able to reach at international level also. This is a good scheme of the ministry, and we are going to encash on this. We should be able to do very well because we have the right kind of atmosphere for providing right sort of training here. We have good ground, good area, pollution-free atmosphere, which other cities lack. So we must take advantage of this. I hope we'll be able to move in this direction also.
So along with sports, there's another area which needs a lot of emphasis, which is soft skills training. That the personality of the learner needs to be honed in a holistic manner, so they can face the challenges of the world, as it were. So is the university doing anything in that direction?
Well, in social sciences, I had suggested something like this, which will help in augmenting the personality of a student. So one of the things which has resulted out of that is, and UGCA also started encouraging this, that is that a subject like political science is going to be taught everywhere. It will start creating awareness among students. Similarly, I have told them that we design courses on public speaking, art of interview, art of preparing for competitive examinations. In fact, there is a light thought which is on this year that we should hold special sessions for the less privileged ones so that they can be trained for higher level competitive exams like civil services, banking, and defense area. I think this will also enhance the employability, image of the university, and overall personality of the student also. So this is going to be prepared soon enough, which will help students grow up in the right direction.
That's very wonderful. And another thing, sir, that this year the university also come out and made initiatives for the mental health of the students, because that is again a problem that a lot of young people are facing in terms of anxiety, low self-esteem, depression, and the university has come out. So is this going to be taken on a larger level to address mental health issues of students?
If you remember, a few months back, we had organized a good seminar for these challenge zones. And our applied psychology department did a wonderful work. I told them that they should come out with programs. Second, I suggested was that they should have their own team of counselors, which could be research scholars of the department. I have encouraged them to go to various hospitals to help them. I'm sure we will be able to contribute to this also. One more area which has attracted everybody's attention is the area of distance education. Somehow, again, I am not able to explain the reasons why there was no interest in the growth of this. People were somehow feeling that this is a thing of the past and it is not what we need in the present day. But I have always been maintaining that in our society there is a large section which for a variety of reasons could not get to the colleges or regular schools, and they could not get the right kind of education. That has led to this class to a great sense of dissatisfaction or depression, that they were very keen but somehow could not complete it. For them, I think this is a great thing. They can enroll here and fulfill their dreams, whatever they have at that time. So this is what we are revamping now, and we are advertising, we are popularizing this. And I'm sure this is also going to result in some good focus on education through distance mode. And in the same line, we have added many other colleges under our training program. So this will also augment the functioning of distance education. Yes.
Sir, on the cultural front also, the last year has seen a lot of vibrancy and new activities beginning. So can you tell us about that?
You see, whenever I look at a university, I always feel like encouraging our students to the Indian culture, Indian classical music, Indian forms of art. And one of the best ways with an organization which has done a human service in this area is our Speak Method. Started long time back, it started in 79, and it so happened that I was a young lecturer at that time. When they started, we were very enthusiastic and we started helping them. We never imagined that this Speak Method will grow to this status, that it will be probably everywhere. When I joined here, they said Speak Method used to participate, but for the last 7-8 years, it never came in. So we revived those legs and we organized excellent programs of Carnatic music and Indian classical music. It was on for 2 days. People from outside also attended this. I think that gave me immense sense of satisfaction that our students, young minds, are going to be drawn into our Indian culture also. They have a good place which they organize. So I am of the view that Pondicherry University is one where our students are so very intelligent, so very brilliant, so very enthusiastic about these things, that they are to be given little encouragement. I told them, whenever you want to do any play, if there is any problem, please get back to us. With this encouragement, they, I think, organized about 6 plays last year. And I don't know, those you have seen, they would bear me out that this was really of a wonderful standard. So the cultural life of the students, which is, in my opinion, a very important requirement, has started moving in the right direction. And this positivity should be there in the mind of students. This is taking a good shape.
Good afternoon, everybody. Welcome back to the second session of this first day event. Shashwat, are you there?
Yes, ma'am. I'm audible, ma'am.
Start the e-poster session.
Yes, ma'am. I'm audible, ma'am.
Yes,
Now?
you're audible. Fine.
Okay. So, yes, ma'am.
Yes, you can
Will you
start.
start, ma'am? Okay. So, very good afternoon to all. Myself, Shashwat. I am hosting your e-poster session 1. So, we have here our chair, Dr. Kitlangi Suchiang, Assistant Professor from Department of Biochemistry and Molecular Biology, and Dr. Asmita Das Gupta, Assistant Professor from Department of Biochemistry and Molecular Biology. So, well, I welcome you, sir and ma'am.
Yes.
So, should we start, sir? I'll start. I'll call this participant.
Yeah, just 1 minute. Good afternoon, everyone. To all the contestants, I'd just like to say, so you can stick to the allotted time of 5 minutes. And remember to put your cursor in the correct section of e-poster while you're explaining. All right. So with that said, all the best. You can start, Saswat.
Okay, sir. So I want to request all the participants of e-poster session 1 to raise your hand so that I'll make them a co-host and then they can share their screens. So the first participant is Pemula Gautam. Are you there? Pemula Gautam.
Yeah, Pamela, I have made him a co-host, Shashwat.
Okay, ma'am. Pemula Gautam from Chettinad Academy of Research and Education, and his topic is recent trends in nanomedicine using 3D organoid. So, Pemula Gautam, are you there?
Oh God, now I think he has a connection problem. He left, I guess. He's not there now. I just now made him a co-host.
Okay, so should we call the second participant, sir?
Yes, please.
Okay. So the second participant is Anil Kumar Vaidya from Pondicherry University, Department of Bioinformatics. So Anil, are you there? Anil, I think he also not there, ma'am.
Yeah, yes, I guess. I don't see his name here in the participants list.
Yes, ma'am.
Then we'll go to the third participant, Harini.
So the next, the third participant is R. Harini. R. Harini, are you there?
Yes,
Harini.
There. I'm making
Okay, Harini. Yes, sir. Okay, ma'am. So I'm from Mother Teresa Postgraduate and Research Institute and Health Science. Harini, you can share your screen and start the presentation.
Yes, sir. My voice is audible, sir?
Yes, voice is
Hello,
audible.
sir?
Yes, your voice is audible. Okay.
My screen is shared, sir.
Yes, it's visible. Oh, you can start.
Yes, sir. Good afternoon, everyone. Myself, Harini, from Mother Teresa Postgraduate and Research Institute of Health Sciences, Pondicherry. Today, my topic I'm going to present is that molecular docking studies on promising novel marine drug for mosquito larvae inhibitory effect. When we speak about vector-borne diseases, the first thing that comes to our mind is that mosquito-borne diseases, because they ravage health and well-being of human beings by spreading diseases like malaria, dengue, and filariasis. On the other hand, when we use excessive chemical insecticide in the management of mosquitoes, this may lead to development of it's a need of an hour to develop a bioinsecticide in the management of mosquitoes. Marine untapped resource for new drug development. So I have worked on Sargassum whitey, a brown seaweed, in the management of mosquitoes. Figure 1 represents the Sargassum whitey, a brown seaweed. And aim of my work is that to evaluate the constituents present in the Sargassum whitey in a methanolic extract by GCMS, and also to carry out docking studies. Materials and method. This Sargassum whitey was collected from the Mandapam coast, Rameshwaram, Tamil Nadu, by hand picking method with the help of a marine biologist, and it was authenticated, and the collected samples were cleaned, washed, and shade dried, and they are comminuted into fine powders. This fine powder had been extracted by acetone by maceration process for 72 hours, followed by extraction with methanol, because this acetone will denature the cell and causes the liberation of secondary metabolite, and this liberated secondary metabolite will be extracted by methanol. And this methanolic extract was concentrated under reduced pressure at 40 degrees Celsius to get a crude drug. This crude drug has been evaluated by GCMS analysis, and these are the parameters involved in GCMS analysis. And the constituent which has been identified by GCMS analysis, Lipinski rule of 5, drug likeness score, and molecular docking studies were performed. This Lipinski rule of 5 will characterize the ADME properties of the drug, whereas drug likeness score will determine whether a compound will fit to be a drug or not. These two parameters have carried out by using a software, Mole Inspiration software. Next is molecular docking. I have studied molecular docking using a software, Autodoc version 4.2, and the supporting softwares were MLG tool 1.5.6 and Python 2.5. These are the two proteins I have targeted. One is aryl alkylamine N-acetyl transferase 5B, which is present in Aedes species mosquito. The reason for selection of this protein is that this protein involves in skeletonization, that is, it protects the insects from physical injury and also infection. And the control and I've used is Temifos. And next protein is epsilon class glutathione S transferase. This protein is present in Anopheles species mosquito, and the control I use is delta methrin. And the reason for selection of this protein is that this protein is associated with developing resistance to a chemical insecticide like organochlorine insecticide. And the result I have got is table 2 represents the GCMS chromatogram of methanolic extract of Sargassum IT, which indicates that there are 18 phytoconstituents were identified by GCMS analysis. And table 3 represents the interaction of compounds with the protein aryl alkylamine in acetyl transferase 5B. All the constituents showed negative binding energy, which indicates that they have negative inhibitory effect on this protein. And the one compound, 1,2 benzene dicarboxylic acid and di-undecyl ester, showed the binding energy closer to that of control. And the figure 2 and 3 represents the docking image of control and the sample which had binding energy closer to that of control. Next, table 4. This represents the interaction of constituents with the protein, second protein, that is epsilon class glutathione S transferase. All the constituents showed negative binding energy towards this protein, which indicates that they have inhibitory effect on this protein. And the constituent diisooctyl phthalate showed the binding energy closer to that of control. And the figure 4 and 5 represents the docking image of the control and the constituent with the protein. Sir?
Harini, your time is up. Now it's already 5 minutes. So I'll ask our chair, Harini Swan, to ask question
yes.
for now.
So you can conclude, just conclude, keep a conclusion, right? Give your conclusion.
Okay.
Yes, sir. This GCMS analysis indicated the presence of 18 bioconstituents, and Lipinski rule of 5, which has been carried out by using a software, mole inspiration software. The constituent phenol 2,4 bis 1, ethyl obeys the Lipinski rule of 5, which shows that it have ADME property, good ADME. And whereas in drug likeness score, the constituent which I've highlighted in table 6 shows that they are fit to be a drug. That is, they all show negative charges, which indicates that they have.
Hello. Yes. So what is your next plan?
Actually, next plan is to carry out in vitro activity followed by formulation, sir.
Okay. All right. Dr. Ashmita, if you have any.
Harini, do you think this phthalate could be an environmental poison if you use it as a drug?
No, actually, I didn't carry out this toxicity study, ma'am, for this constituents. Okay. After carrying out in vitro, I will be going for toxicity study. In that case, I will be finding it whether it will be toxic to environment or not, ma'am.
Okay. All right. Okay. All right. Yeah. Yes. Thank you.
Thank you, ma'am.
Call the next one, Saswat. Unmute your mic, Saswat. Unmute your mic.
Shashwat, Anil Kumar Vaidya is here, I guess.
Okay. So Anil Kumar Vaidya, are you there?
Yeah, I'm
Anil?
here. Yeah,
Okay.
I'm here.
Okay, so you can start. You can share your screen.
Hello, everyone. I'm Anil Kumar Vaidya from Center for Bioinformatics, Pondicherry University. My topic is on identification of T-long non-coding RNA, which associated with the multistage long adenocarcinoma using competing endogenous RNA network. So here, competing endogenous RNA network is the interaction between coding and non-coding RNA. So generally, oncogenesis is a multi-step process where early stage is asymptomatic in nature. So usually cancer doesn't appear until the disease is an advanced stage. So early detection of cancer can to reduce cancer mortality. So among cancer, lung cancer is a leading cause of cancer death, in which lung adenocarcinoma having overall survival rate is less than 20%. CERN hypothesis is highlighted that the expression of a long non-coding RNA can regulate the expression of other target genes by competitively interacting with miRNA. And long non-coding RNA possesses a significant regulatory effect on cancer carcinogenesis and tumor progression. So we used an integrative computational approach to identify potential stage-specific LNC RNA to distinguish different stages of lung adenocarcinoma. So my objectives are identification of significant mRNA and LNC RNA interactions, and identification of differentially expressed LNC RNA, miRNA, and mRNA. Then identification of stage-specific differentially expressed LNC RNA. Then construction of stage-specific cRNA network. So for my methodology, we collected miRNA, mRNA interaction and LNC RNA, miRNA interaction from different databases. Then we filtered and we have only considered a non-redundant interaction information for my further analysis. Then we identified significant interaction between LNC RNA and mRNA by using hypergeometric tests by developed in-house Python programming language. So for differential expression analysis, we collected more than 1,000 samples, which include like tumor and normal sample from TCGA for stage 1, and more than 500 samples for stage 2, and more than 300 samples for stage 3 collected from TCGA database. TCGA database is like a repository of RNA sequencing data. So we identified a differentially expressed miRNA, mRNA, LNC RNA using our language. Then we went for Pearson correlation analysis between LNC RNA, mRNA to get positively regulated interaction. Then we for stage-specific differential analysis, we took only top 100 off-regulated LNC RNA for further to construct cRNA specific RNA network. For we did Pearson correlation analysis because study have found that if expression of one gene increased, then the interacting partner of that gene also increased. So, in result, in this Venn diagram analysis have shown that a total of 73 LNC RNA were shared among all the stages of lung adenocarcinoma. So a total of 10 LNC RNA shared between stage 1, stage 2, and 6 LNC RNA shared between stage 1 and stage 3, and 7 LNC RNA shared between stage 2 and stage 3. So, and we consider only 11 LNC RNA which is specific for stage 1, and 10 LNC RNA which is specific for stage 2, and 14 LNC RNA which is specific for stage 4. And these stage specific LNC RNA were considered for further regulatory analysis. So in this network, all network consist of nodes and edges. So here yellow that red color indicates LNC RNA, and this yellow color indicates mRNA, and the link between all the nodes are called edges. So for stage 1, we have found that there is only one LNC RNA, which is a hop LNC RNA. Hop generally having a higher degree is means like the connection between nodes.
Excuse me, Anil. Anil, your time, you already exceeded your time. So, sir, can you ask question? Sir, ma'am.
Yeah, let him conclude one point. What is the finding? What is interesting finding from this document?
Okay, sir.
Tell me.
Okay, so after constructing that cRNA network, we performed a functional enrichment analysis. So that in functional enrichment analysis was for differentially expressed genes that are involved in that cRNA network. So from that we concluded that hop genes are involved in cell proliferation and cell division and replication. So from that we conclude that there is a one stage specific LNC RNA for stage 1 and 3 LNC RNA for stage 2 and 6 LNC RNA for stage 3. So overall, we concluded that these 10 LNC RNA are identified as biomarker to distinguish different stages of lung adenocarcinoma.
Good. So Ashmita, madam. Yeah.
Hello. Yeah. Anil, can you hear me?
Yeah.
I mean, you were saying that you found like 3 stage specific LNC RNAs for each of the stages, right?
Not only 3. Yeah,
no,
not only
3
3 10.
markers. That's what you're saying. So what are these? What are the key functions?
For for stage 1, stage 2, you're talking about, right?
No. Finally, you said that for LNC, like stage 1, 2, and 3, there are 1 marker each that you found. No, or all are.
Yes, ma'am. Not only like 10 marker I have found, and this 10 marker, 10 marker like regulating. Ma'am.
What are the key functions?
Okay, these 10 markers are involved for regulating that mRNA. So that mRNA involved in cell proliferation, cell division, replication. So
What is that mRNA? What is that mRNA?
What is the mRNA? What is the protein?
That the coding gene. Coding genes.
Coding gene. What is the protein?
Okay. One I got, like there are lots of genes. So
okay. All right. I think he doesn't want to repeat. Okay. All right.
I especially focus on LNC RNA.
Okay. All right. Yeah. Thank you. No problem. Yeah.
Okay. Thank you, Anil.
Yeah.
So should I go for next participant, sir?
Yes, please.
Okay. So Anil, can you stop your sharing? Okay. So the next participant is Saraswati Nagaraj from Chettinad Academy of Research and Education. Saraswati, can you share your screen?
Yes, sir, I'll do it.
Okay.
Can you see my screen, sir?
Yeah, it's visible. Yes.
Yeah. Shall I start?
Yes, please start.
Good afternoon to all the panel members and one and all. Thank you for giving me the opportunity to present at the international conference. I'm Saraswati Nagaraj from Chettinad Academy of Research and Education, pursuing my PhD. The title is Taking Tradition to Microbial Infection Management Through the Nano Route. My topic of research for PhD is about how nanoparticles being used for functionalization with biological molecules. So in order to tune them into particles that can be used for localization as well as for the accumulation, and in order to prevent the microbial growth as well for the microbiocidal activity, which means a killing of microbes. Based on this, my presentation brings out how the antimicrobial natural compounds, let me say for instance, chitosan, which has got amine groups, which is positively charged, which binds with the negative charge of the microbe. Alginate possesses negative charge, which can bind to the positively charged biomacromolecules of the microbes. We can also prepare hybrid nanoparticles based on this, chitosan and alginate, which can suppress the bacteria by binding either to their positive region or to the negative region, allowing them to allowing the entry of the drug in a sustained manner, so that before the bacteria can develop resistance towards the drug or kill the microbes. Under this heading, we have different molecules like hyaluronic acid, pectin, which can be used in general for killing the bacteria, or in case can be prepared in such a way that it can be encapsulated the drug onto the active principles from plants. So our particles which has formed nanoformulations can be used for killing the microbes as well as the drug inside it. It's like a double-edged sword where you can kill the bacteria by A mechanism or a B mechanism. So when we talk about active principle, which can be encapsulated inside the nanoparticles, we have a curcumin, which is from turmeric extract, allicin from garlic extract, capsaicin from green tea extract, all these things which has been traditionally accepted long way. But by along the journey, it has lost its significance because of their incapability and low bioavailability nature as to accumulate in the target site. So now with the advent of this nanotechnology, these active principles have been attained and gained a tremendous importance. For instance, let's take an example of a current pandemic situation, COVID. We Indian, by virtue, we have an habit of taking these natural active principles as a part of our day-to-day healthy lifestyle. Here, these nanoparticles probably would have boosted in developing immunity against these kind of microbes. Maybe this would have not acted, or maybe not as a vaccine, but as an adjuvant to stimulate the immunity inside the other living systems. So if such kind of active principles can be encapsulated into nanoparticles, and these particles will have a different method by killing the bacteria. But again, the approach of using these nanoparticles is based on nanotechnology. We have to remember that the nanobio interface is the very important thing which have to be understood before we generally synthesize our particle. We have to understand the lipid membrane, what being added onto the particle, like functional moieties or ligands, and their functionality of our nanoparticles that can exactly happen when it go to the system. Because once we put it into the biological system, will the size will be retained or will it form aggregations? So that to consider this will be considered as a boon or a bane. In order to overcome this, we have to look at their different physiochemical properties. We have different formulations of nanomaterials like liposomes, polymeric, co-shell based, where these biological materials can be synthesized in right chemistry in order to conjugate or to encapsulate or to entrap or to absorb our drug. The mechanism may differ because directly the drug will not bind to the bacterial surface as to develop resistance towards it. So what happens is our shell or a core, each moieties of multifunctional nanoparticles will be designed, helps in localizing our drugs in a targeted site. These strategies are devised nowadays to develop with the diagnostic as well as the therapeutic potentials. For example, on one which I am researching on is against the Candida albicans. Here, Candida albicans are specific biomarkers which can selectively bind to an antimicrobial peptide. So when we look at the previous literature, we analyze that n number of antifungal drugs that are in present, but still amphotericin B is a gold standard drug in preventing the growth of the microbe. So what are we trying to is to encapsulate a biopolymeric shell with the antimicrobial peptide as a selectively localize their nanoparticles to a specific site. So whatever it is a drug. It doesn't know where it goes to, whether it goes to a normal site or to a disease site. So we are struggling in such a way in order to find the drug is being localized. So we'll be adding some functional bioligands like fluorescent moieties to understand the localization of drug. That is a kind of strategies being used Just look at the picture of a liposome, which are not stable. So we add a polymeric coat on top of it, which in overall comes as a biopolymeric nanoparticles. It is important to look at a way how it can go inside a biological system, or it can kill or prevent the growth of microbes either by attracting the nanoparticles, or by the production of free radicals, or either by modulating or through the interaction with the DNA, or by inhibiting the protein synthesis. And coming to the biomedical application part, if you look at the biomedical application part for managing any infection, here nanoparticles have got both diagnostic and therapeutic role. Nowadays, people are looking at low-cost paper-based devices, biosensors, and biomems device where we can detect, and which is being a relevant topic during pandemic situations. The nanotechnology have gained tremendous importance in this field, especially green technology. By nowadays, it is a sustainable technology. So researchers have driven nowadays to see whatever we have designed in drug formulation is sustainable. By 2030, it is expected in day-to-day life, even research is based on this sustainable goals. So to conclude, we can very well say that the nanoparticles are independently or in combination provides the antimicrobial properties, which can be used against the drug resistance bacteria or a virus. And it can also be tuned either through their size, shape, as well through the surface modification or through the conjugation. Thus, the nanoparticle-based antimicrobial agent have been explored only to a small extent, and there is a larger scope for furthering research in this area. Thank you all for giving me this opportunity to present at your prestigious institution. Thank you, one and all.
All right. So just one question. So what do you people talk about nanoparticles toxicity? So what is your view on that? Can you hear me? I think she's. Hello.
Yes,
Hello,
sir. Yes, sir.
Can you hear me?
Yes, sir. Yes, sir.
So what about the side effect of nano drugs and all those things? People talk about nanotoxicity also. So what is your
size-based, sir?
Huh?
Size-based.
All right.
Aggregations happen, sir.
Okay, Ashmita.
Yeah, so this is just a discussion that you plan to work on, right?
Yes, ma'am.
Okay. So you have not started any work yet, right?
No, ma'am, I have started. Synthesis part is done, and we are looking forward for the in vivo models, ma'am.
Okay. Okay. All right. Yeah. Thank you.
Thank you.
Next one.
Okay, thank you, Saraswati. So our next participant is Dr. Sumitha Gopalakrishnan. So, ma'am, are you there?
Yes, I'm there. I'm here. Yes.
Okay, ma'am. You can share your screen, ma'am.
Yeah, one second. Am I audible?
Yeah, it's audible, ma'am.
Yeah. Good afternoon. I'm Sumitha Gopalakrishnan from Calcutta University. My work is based on purification and characterization of lipase enzyme from Streptomyces albus ST04. Microbial enzymes have vivid uses in industries and medicine medicinal purposes also, and are more active and stable than plant and animal enzymes. In addition, the easiness of production of enzymes and microbial sources by fermentation leads to application in biochemical diversity and susceptibility to genetic engineering. Lipase enzyme characterization of Streptomyces albus ST04 strain isolated from coastal waters of Andaman Nicobar Islands and was carried out during the present study. And mass production and extraction are done by using standard methodologies, and lipase activity measured by titometric method as olive oil as substrate. The structure of protein was predicted in UniProt D6B385 from Protein Data Bank in the Swiss modeling tool. The substrate specificity of the enzyme to various substrates, pH, temperature, and various metal ion concentrations was evaluated. Kinetics of lipase enzymes and constants Km and Vmax were determined. Initial velocity was plotted against concentration of substrate by means of linear transformation of Michaelis-Menden's equation using linear Lineweaver workflow. Crude enzyme has showed specific lipase activity of 2.81 unit per mg. And ammonium sulfate precipitation, this desalting process, and final purification using Cephedex G100 column showed specific activity of 238.89 unit per mg. Final purified enzyme have exhibited 84.94 purification and 41.9 yield enzyme. SDS page analysis of purified enzyme yield single band of 47 kilodalton. And the enzyme showed highest activity at 9 pH 9 and 45 degrees Celsius and at 6% of substrate concentration in the presence of calcium ions. And these are the and this is the predicted structure, tertiary structure of the enzyme. And conclusion of the work, present study showed that lipase enzyme from Streptomyces albus STO4 have molecular mass of 47 kilodalton and enzyme is highly stable at a pH 9 and 45 degrees Celsius in presence of calcium and exhibited Km value of 4.56% with 23.6 unit per ml Vmax. Thank you. Hello.
Okay.
Sir.
Thank you, ma'am.
Yeah.
Sir?
Okay, what is your next plan for this enzyme? What will you do with this?
I have to do NMR spectra and other characterization processes.
And the ultimate one?
I have to do NMR spectra.
Applications, you think of any application, something like that?
Since the enzyme is very stable, sir, so we are in industrial purpose and other purpose, we are just planning to check, sir.
All right. Ashmita?
What was the optimal pH? So I was just wondering if you could use it with detergents.
Yes, sir. Yes, madam. The optimal pH is 9.
Yes, it's interesting.
Yeah, you could actually use it with detergents.
Sorry, ma'am?
You could use it with detergents.
Yes, sir. Yes, madam. Yes, madam.
Yeah, right. Yeah. Okay. All right, madam. Thank you.
Okay, ma'am.
Thank you for the nice presentation.
Thank you. Thank you, sir. Thank you.
So, ma'am, can you stop your screen sharing? Hello, ma'am. Can you stop your screen sharing?
Yeah. 1 second, sir. Yes.
Okay, thank you, ma'am. So, our next participant is Ankit Biswas from Regional Center of Biotechnology. So, Ankit, are you there?
Yes, I'm there. I'm just sharing the screen.
Okay. Thank you, Ankit.
So my screen is visible.
Yes, it is visible.
So hello everyone, myself Ankit Biswas. Today I'm going to present the topic titled using the combination of both probiotics and prebiotics to treat neurodegenerative disease progression. So what is neurodegenerative disease? So neurodegenerative diseases occur due to dysfunction or death of the cells in the central nervous system, and among them Alzheimer's and Parkinson's are the most common one. So in the literature, it is already evident that in Alzheimer's patient, the Firmicutes to bacteroids ratio decreases significantly. And in case of Parkinson's, there is an increase in LSC species, and decrease of Helicobacter pylori and E. coli and bacteroids in the non-normal gut microflora. So there is a gut dysbiosis that might be regulating the disease pathology of Parkinson's and Alzheimer's. So how might the gut dysbiosis regulating the disease pathology? So the gut dysbiosis can increase the metabolite secretion, the dysfunction of endocrine regulation, neural transmission of the secreted metabolite, and immunomodulation could lead to the beta-amyloid deposition, insulin resistance, and neuroinflammation in case of Alzheimer's. And in case of Parkinson's, alpha-synuclein aggregation oxidative stress can be there. So as the microbiota gut-brain axis is directly related to the neurodegenerative disease progression, so using prebiotics and probiotics could be a helpful way to stop the disease progression. So fructooligosaccharides, resistant starch, xylooligosaccharides, such kind of prebiotics have been already tried to stabilize the microbial gut flora and maintaining the diversity. And it has been shown that in Alzheimer's disease pathology, it is helpful and beneficial. But in case of probiotics, there is only one probiotic for preparation that is used in case of mice, but no such clinical trial has been done broadly. Only one clinical trial has been conducted to assess the effect of probiotic supplementation on Alzheimer's disease. But the use of both probiotic and prebiotic side by side to treat the disease progression is not yet used. So my proposed model is to use both probiotic and prebiotic and orally administered it so that it can maintain the diversity and stability of the microflora and maintain the homeostasis of the microbiota gut-brain axis. That could in turn, using this mechanism, could improve the neurodegenerative disease pathology. So the future direction of this hypothesis would be identifying the key microbe that are useful in neurodegenerative disease, identifying the key metabolites, elucidating the role, elucidating the synergistic and antagonistic effect of different pre and probiotic regimes, and after getting a significant hit, systemic study to get the molecular insights of the effect. So here are my references, and thank you all to
Sir, any questions?
All right. So nowadays people talk about this topic, right? It's a very hot topic for research, especially. So is there any finding related to inflammation? What do you think, according to you, say just from your point of view, that probiotics secrete that lead to the ameliorations of this neurodegenerative disease? What do you think? Can you predict something?
So, sir, there are already reports that it are showing that it could be helpful, but
in
mice models,
they reduce metabolites, or what? In what way they decrease inflammation?
So the molecular insights is not yet elucidated, so we can't say anything right now. But my prediction would be that metabolites and amyloidogenic proteins might be a
anti-amyloid. You mean to say?
Yeah,
right?
Yeah. Might be a key factors for regulating this.
Ashmita.
Hello, Ankit.
Yes,
any particular class of bacteria you are thinking would be good, like a lactobacillus bacillus or something like that?
Ma'am, using lactobacillus is already have been tried, but not good result have been found. So I might use a different regime of different microbiota that are present in healthy microflora, but not present in Alzheimer's disease condition.
So is it known that the gut microbiota is different in case of?
Yes, ma'am.
That's what
It
you're
is
saying in the
totally
introduction?
different.
Yeah. Okay. All right. Yeah. Thank you. I think, yeah.
Thank you, Ankit. So our next participant is Srividya Subramaniam. So Srividya, you can share your screen. Srividya?
Yes. Just a moment. Am I audible?
Yeah, you're audible, Srividya.
Yeah, just a moment.
Okay.
Good afternoon, everyone. Thank you for giving me this opportunity to present my work at the International Conference on Drug Discovery and Translational Research. This work is actually related to the the identification and validation of the anti-cancer potential of a compound of an alkaloid called harmalin. And this is actually derived from a plant. It's originating from a plant called Peganum harmala. And there are different forms of this alkaloid called harmine and tetrahydroharmine also. But in this case, we have studied the action of harmalin. And what we have done is, first of all, the reason that we have chosen this is because when we are looking at drug discovery and chemotherapy, we are always in need of newer lead compounds. So when we look at lead compounds, by doing chemical modifications and by finding the derivatives, we can modulate their chemical activity, we can increase the cytotoxicity, we can improve their specificity. So to carry out such work, first we have to understand what is the basis of their action in the in silico and in vitro studies. So what we have done is we have carried out some basic in vitro studies, which the results of which I will be sharing now. Now in this, this is the structure of harmalin, as you can see, and this is the plant from which it is derived. Now, as you can see, this has a pyrrole ring. This is actually an indole functional group containing beta carbolin structure. So if we are understanding the mechanism of action, for example, in the literature, very recently only some work has been done, and they are found by substituting various functional groups, some increase in the cytotoxicity, some difference in the specificity has been found for this structure. So actually our future plan also involves understanding the basis of how ROS generated generation or ROS involving apoptosis can be like what is the mechanism involved in such apoptosis, and how we can modulate the structure or modify the structure to improve the cytotoxicity. So in this case, what we have done is we have analyzed the we analyzed the cytotoxicity in both lung cancer and triple negative breast cancer cells, which is MD-MB231, which is the breast cancer cells, and AF9 lung cancer cells via MTD assay. And we have also analyzed among these two, the lung cancer cells showed a higher cytotoxicity. So we have gone further and we have analyzed the morphological feature changes by phase contrast imaging, and we have evaluated the mode of cell death by acridine orange yttrium bromide staining method, and also we have evaluated for the presence of ROS generation in the apoptotic mode of cell death by DCF-DA fluorescence staining method. So now you can, I'll just show the results here. We have found that harmalin showed a cytotoxicity at 24 hours in lung cancer cells, and we have found the IC50 value to be 56.2 micromolar. And we found that in the MD-MB231 cells, when we have carried out the similar experiment, the IC50 was very high and it was greater than 100 micromolar and could not be calculated because less than 50% of the cells had died. So we also know one fact that the triple negative breast cancer cells, they lack certain receptors, characteristic receptors, which which certain drugs will target, and therefore they can kill the TNB cells. But similarly, this does not seem to work on the TNB cells, but it is able to work on the lung cancer cells. So here we can see the graph for the percentage cytotoxicity from which we have calculated our IC50. And we can now see the next result, which is the phase contrast imaging, which is showing the proof of the decrease in the number of viable cells. And looking at the AO ETBR staining, we can see that over here, there has been a breakdown of the cells upon apoptosis, and the red color indicates the late apoptosis. And as we can see, since it is the IC50 treated cells, not all the cells have died, because we are trying to study the mechanism of the action. So we would like to understand what happens in the mediation of this action. And as you can see in the untreated cells, the cells are completely all right, and there is no change in the fluorescence. Now coming to the DCF-DA staining, when DCF-DA is converted to DCF by the cells, and if you have ROS generation, what happens is the DCF-DA is converted to a more fluorescent DCF form. So this is why you can see that compared to the untreated, where you can clearly see the features of the cells, the fluorescence in the IC50 treated cells is quite high. So this shows that ROS generation, which is nothing but the oxidative stress, which is usually induced by many of these plant phytochemicals, which are leads and also potential anti-cancer agents. This is what actually the mode of action involves, the ROS generation, and that is indicated by this DCF-DA to DCF conversion. And this is clearly visible in the IC50 treated cells. Now to conclude, we can say that what we understood is that in MD-MB231 cells, the action itself or the stress itself, maybe there are also some other targets in addition to the ROS generation, which we will have to analyze by the molecular mechanisms of gene and protein expression studies. But in A549 cells, it is more susceptible to this compound, and therefore it is able to show us a measurable IC50. And another reason is, of course, as we said, the direct mechanism, which is the increased ROS levels induced by harmaline in the A549. These are also, usually the ROS will induce the intrinsic mode of apoptotic, the apoptotic pathway, the intrinsic type. So this is also one of the reasons why it might be killing the A549 cells. However, this plant compounds usually have multiple actions, or they are multi-targeting in nature. So, and also in the other literature, recent literature shows that there are some proteins, 1 or 2 proteins have been discovered which could be targeted by this drug. However, the validation is not complete. So there is a lot of scope in understanding the mechanism of action and in creating the derivatives of this plant compound, which we will be studying further in which I'll be studying further in the coming months. So this is the work which I have done till now. So,
sir. All right, good. So this plant is eatable, Sri Divya?
Yes, sir. Actually, this one, this Peganum harmala, it is used as a psychoactive inducer. Some of the tribes in Africa, in the deserts of Asia, and also deserts of Africa and Central Asia, some of the villages and tribes, they actually use this in their traditional medicine they have tried. And also we talk about shamans and about witch doctors and such people. So they have used this to induce a kind of psychoactive trance, or some kind of, because it inhibits the monoamine oxidase, it inhibits serotonin uptake. Normally it has been found. This is also one of the, it is a reversible monoamine oxidase inhibitor. So it has some psychoactive activities. So by preparing derivatives, we will try to actually prevent this kind of side effects or other kind of effects. And it can focus more on the cytotoxicity aspect or the anti-cancer aspect. So this is actually what is mainly used for.
So that ROS mechanism, which mechanism do you think that this particular compound can induce ROS production?
Yes, sir. Actually, in this, if you see, I mean, according to the literature also, and also from our lab, the previous work done in our lab and in general scientific literature, it shows, we see that intrinsic apoptosis is induced when there is a lot of oxidative stress in the cells. In very rare cases, there is extrinsic pathway also, which is which has been induced, I mean, as the main pathway. But we can, it is, I mean, I can theorize it could be intrinsic, but of course I will have to verify it with the proper gene and protein expression studies before I can say that for sure.
All right. Good.
But it might be intrinsic.
All right, Ashmita?
You tested with 2 cell lines, I understood, right?
Yes, ma'am.
So why do you think it didn't show much activity in one and it showed more activity in the other? What are the specific differences that you are aware of?
Yes, ma'am. Ma'am, actually, now this MD-MB231, it is derived from the breast cancer gland, and also it lacks many of the receptors For example, the HER2 receptor, the estrogen receptor, progesterone receptor, many of the drug molecules which are made up of complex structures, including even this harmaline or any other drugs, which the PARP inhibitors, for example, they all have complex heterocyclic and larger structures. So those structures are only able to particularly and specifically act on the triple negative breast cancer. And because of, and in general, even not in our case alone, but in general, it has been found that many of the drugs which are acting on the other cancers are unable to act on MDMB231 because probably the drugs may be having multi-targeting effect, but they are unable to bind because of the lack of these receptors in the triple negative breast cancer cells, for which MDMB231 is a model system. So whereas in A549, we have many other targetable proteins and targetable genes, and also harmaline is something which binds to the DNA. Of course, we are also planning DNA binding studies, but harmaline in general has been found to bind to DNA. However, we feel that the ROS generated one seems to be a dependable strategy, and also in targeting a particular protein, because DNA binding, again, harmine is the isoform which is more active with the DNA binding. And DNA binding also, sometimes it is not very specific, and it will just induce a generalized cytotoxicity, which may result in the death of normal cells. So these are some of the reasons why MDMB, for example, not expressing the targetable proteins is unable, cannot be targeted by conventional drugs or naturally available pharmaceutical as they are. Without, I mean, only with derivatives, it might be possible.
Okay. Yeah. Thank you,
Yes. Thank you, ma'am.
Thank you, Srividya.
Thank you. Thank you. Thank you, everyone. Thanks for the opportunity.
So, Smita, can you stop your sharing?
Yeah. Sorry, I'm unable to access this.
Okay. So, our next participant is A. Vijayalakshmi.
Shashwat, I guess she is not
Yes.
here. I made her as a co-host, but still
Okay.
she's not seen in the list now. And
Okay,
Vijaytan
ma'am.
is also not seen. Can you please check again?
Okay, ma'am.
If Vijaytan has entered in another name, so can you please raise your hand so that I can make you a co-host?
Also, Pamela, are we looking for
Pamela is here. Yeah, Pamela Gautam is here, but I don't see his
Okay.
mic. Anyway, you can just call Pamela Gautam. He's here, but mic is not showing against his name. Pamela Gautam, can you open your camera so that we'll see you? I think there is no mic. Yes. So in that case, I guess that we can wind up the session.
Yeah.
Yes, madam.
Yeah. So we may take break for we will have a small break until like 3:05 for 10 minutes.
10 minutes.
And we'll start the next session. Let me check whether it is Vijay Lakshmi here. Somebody is connecting to the audio, and it is Pamela Gautam. No, it's Speak Alive one. Okay, fine, no problem. So we can break now, and we'll maybe join exactly at 3:05, so that we'll start our oral sessions early.
Yeah,
All right.
sure. Yeah.
All right.
Thank you, all of you.
Yeah.
Thank you, ma'am.
What if you could have a flow cytometer, but without having to worry about waiting for warm-up, calibration, or cleaning? Meet the nuclear counter NC3000 from Chemometric. From single cell counting to cell cycle, apoptosis, and custom designed assays, the NC3000 does it all in less time and with a small footprint. For high-speed cell counting, the VIA 1 cassette provides a one-step cell count without pre-treatment. The slides give you the flexibility to perform up to 8 cell counts in less than 3 minutes. The list of advanced assays includes cell cycle, vitality, GFP transfection, mitochondrial potential, DNA fragmentation, and Nexon-5 and caspase assays. If that's not enough, there's FlexiSite. FlexiSite is a flexible cytometry platform that lets you build your own customized assays from the ground up, using the cell markers and fluorophores you want. Install the software on as many computers as you like and do your analysis from anywhere. In addition, the NC3000 is 21 CFR Part 11 ready and can be validated easily with the included IQ and OQ assays, though it's at home in both academic and R&D labs. If you need a powerful, flexible cytometer that offers high-speed, precise cell counting, predefined ready-to-use assays, custom assays, and unlimited software licenses, all with no maintenance and a footprint smaller than the centrifuge, look no further than the nuclear counter NC3000.
Capture publication quality images without the hassle of a manual microscope. The cell imager you've been waiting for is finally within reach. Introducing the Image Express Pico automated cell imaging system from Molecular Devices. Automated acquisition plus intelligent analysis integrated into a single configurable system. Whether running single slides or up to 384 well plates, the Image Express Pico is designed to minimize manual effort and increase efficiency. Capable of bright field and fluorescence imaging, the Image Express Pico comes standard with temperature control and optional environmental control for live cell imaging. Unlike other imaging solutions, the Image Express Pico automates both acquisition and analysis of cell images. With pre-configured cell imaging protocols optimized for specific assays, ranging from simple cell counting to sophisticated murite tracing, getting started is a snap. Each pre-configured protocol is optimized to collect the most pertinent results and to ensure accurate segmentation. The Image Express Pico automatically adjusts focus and exposure for consistently brilliant images. From a whole plate overview to an individual cell, you can visualize your images across multiple scales to gain key insights. Now you can use a single software package to acquire and distill images into data on the fly with our powerful Cell Reporter Express software. Generate heat maps, scatter plots, tables, bar graphs, and even movies with just a few clicks. And because access is the heart of functionality, our user-friendly Cell Reporter Express software is browser-based, so you can operate the system anywhere, from any computer with a network connection. The Image Express Pico is the intelligent answer to image acquisition and analysis. Are you ready to give up guesswork and tedious manual labor? Ready to embrace increased throughput and unmatched efficiency? Now you can. Image Express Pico by Molecular Devices.
Excuse me, ma'am. Can I start the session?
Yes, Neha, are you there? Can we start? Yes, welcome back. Before that, who are the chairs for the session, Neha? I'll make them the co-host.
Ma'am, Asmita ma'am and Dr. Asmita ma'am and Dr. Latha ma'am.
Yeah, Latha ma'am I've already made. Asmita ma'am.
Okay, hello, can you listen to me? Am I audible?
Yes, yeah, you're audible. Neha, you can start the session.
Okay, ma'am. Yeah. Welcome back all after the break. Let's start the final event of today's virtual conference, that is oral presentation, which will be chaired by our two judges from Department of Biotechnology, Biochemistry and Molecular Biology, Dr. P. Latha and Dr. Asmita Dasgupta. Before starting the event, let's go through the rule and guidelines of the event. Each presenter is allotted 7 minutes, with about 5 minutes for the presentation and the remaining 2 minutes for the question and answers following the presentation. As we are having limited amount of time, I request all the presenters to remain stick to the allotted time. So the list of participants in the order are Purnima, Kavya, Kanaga Lakshmi, Arya Kannan, Desi Priscilla, Shafia Zabin Mubarak, Aruna Devi Selvraj, Gopinath Murugan, Dr. Sujatha, Murugan S, Vignesh Mariappan, Dr. Latha I. Shukla, P. Kalaivani.
Okay, thank you, Neha. Asmita ma'am, you are here?
Okay, now I would like to invite our judges, Dr. P. Latha and Dr. Asmita Dasgupta. Ma'am, please join.
Yeah, can you hear me?
Yes, ma'am.
Yeah, is Asmita ma'am here?
Ma'am, I think she has a connectivity issue. She'll rejoin soon.
Okay, then who will be the first presenter today? Do you have the list or should I call?
Yes, ma'am.
Neha has the list.
I would like to begin this event by calling upon our first presenter, Purnima from Chettinad Academy of Research and Education. Purnima, you can start your presentation.
Yes, ma'am. I'm ready. Please introduce yourself and stick to the guidelines. Proceed. Share your screen.
Am I visible?
Purnima has started sharing.
Excuse me, ma'am. Am I visible?
Now it's okay.
Yes, your screen is visible.
A very good afternoon to everybody. Myself, Purnima, pursuing MSc Medical Biotechnology at Chettinad Academy of Research and Education. Here is my presentation on nano-formulated naringenin for improved solubility and ROS scavenging. Let me start my presentation with introduction of what is naringenin. A naringenin is a widely studied flavonoid reported to be an antioxidant. It is found in various fruits and herbs, predominantly in citrus fruits. Other names for this naringenin is naringetal, salpipupol, 5,4,7 trihydroxyflavonol. This flavonoid is found to have antioxidant property, free radical scavenging property, anti-inflammatory property, and also found to have carbohydrate metabolism promoter and immune system modulator. It also blocks the lipopolysaccharide induced transcriptional activity to tumor necrosis factor alpha. Followed by the applications of naringenin. It has a wide range of pharmacological properties, including anti-dyslipidemic, anti-obesity, and anti-fibrotic. It also helps preventing and treating metabolic syndrome, reducing oxidation to have an inhibitor on human cytochrome P450 and cytochrome P450 isochrome cytochrome 1A2, which can change the pharmacological properties in humans. Naringenin is being researched as potential treatment for eczema. Naringenin has been demonstrated to improve memory and to reduce amyloid protein and tau protein. It also said to have antibacterial effect, antifungal, anti-inflammatory effect. So, why this naringenin is so studied? We all see that. But what are the issues with naringenin? The major physicochemicals of this naringenin suggest that it is a crystalline compound and has low release in oral dosage, thereby resisting Hello, am I visible?
Yeah.
Thereby resisting therapeutic dosage. Thereby resisting its therapeutic dosage. Moreover, this naringenin is a hydrophobic compound with poor solubility of approximately 5.81%. Naranjan is said to have low bioavailability due to poor solubility. Nanoformulation can resolve these issues. Due to their amplex property and diversity in design, composition, and construction, offer a dynamic and adaptable technology for enhancing drug solubility. Starting with equimolar egg phosphatidyl cholesterol in liposomes that influences the liposomal composition and surface charge, and the incorporation and retention enhances the solubility of the model. The major aim of a study is to prepare nanoformulated naringenin for improved solubility and ROS scavenging. The objective of the study is to synthesize the liposome from phospholipid isolated from egg yolk to prepare nanoformulated naringenin with improved solubility and ROS scavenging property using liposome. The methods which we followed in this synthesis was isolation of phospholipid from egg yolk, preparation of liposome, enchylation of naringenin with liposome, and test the effect of hydrophobic agents on solubility of naringenin. Here we have used urea, and to detect the ROS scavenging property using DPPH assay. Yes, we have taken egg yolk and have prepared phospholipids using egg yolk, and here we have used various amount of cholesterol in it in the preparation of thin film, and we have hydrated using PBS, and the liposome were formed. Here to the prepared liposome, naringenin was encapsulated using sonication, and thus liposome encapsulated naringenin was formed. DLS characterization of liposomes with different concentration of cholesterol, namely 1 mg, 2 mg, and 5 mg, given as lipo1, lipo2, and lipo3 were produced. The significant decrease in the size of liposome was noticed with increasing concentration of cholesterol, where the amount of cholesterol can be considered as packaging parameter. Then the increase in zeta, which indicates the stability of liposome, was noticed. Encapsulation of naringenin. To the prepared liposome, naringenin was encapsulated by ultrasonic, and spectrophotometric characterization was done to confirm the encapsulation of naringenin with liposome. Spectrophotometric naringenin in liposome. Here, the increment indicates the entrapment of naringenin in liposome. Lipo2 or liposome with 2 mg of cholesterol has better ability in encapsulation. Further increment cholesterol concentration produces similar effect as liposome 2. Therefore, 2 mg of cholesterol can be considered as optimistic character to prepare liposome for the encapsulation of naringenin. Spectral behavior of naringenin with hydrophobic agent. Here, urea is a well-known hydrophobic agent which we have been used. Increasing urea concentration has the ability to increase the absorbance of naringenin in water due to the solubility enhancement. Therefore, it can be concluded that the increment in the absorbance of liposomal encapsulation naringenin is due to the increase in solubility. Then for ROS scanning, we have used DPPH assay. DPPH 2,2 diphenyl 1-picoyl hydroxyhydride is a well-known antioxidant assay. Here, the 3 different samples of naringenin loaded liposome were mixed with freshly prepared DPPH. The ascorbic acts as the standard positive control, and DPPH in ethanol acts as a negative control. The free radicals in naringenin react with DPPH ions producing.
Sorry to disturb you. Your allotted time is over. Would you like to conclude it?
Yes, ma'am. Just 1 minute. I'm at the last.
Can you show the concluding slide?
Yeah, directly she can go to the conclusions. Please, Purnima, are you there? Purnima.
Hello, Purnima?
Yeah, she's not here.
Neha, you try calling Purnima.
Purnima, can you hear us?
Purnima.
I think there is some network issue.
Yeah, let us move on to the next speaker then.
Okay, ma'am. Thank you, Purnima. So our next candidate is Kavya from Bhartiya University. Kavya, you can share your screen.
Just stop them 30 seconds before their time and ask them to conclude. Okay,
oh, okay, ma'am.
Yeah.
Kavya, is she here?
Kavya, are we audible?
Kavya.
Kavya?
You can see her slide, but her voice is not audible.
Kavya, can you unmute your mic? It is muted.
Good afternoon, ma'am.
Yes.
Good afternoon, ma'am.
Yeah, Kavya.
Good afternoon, ma'am. Can you hear me, ma'am?
Kavya.
Ma'am, can you hear me, ma'am?
Yeah.
Yeah. Okay, good afternoon, all present here. My name is Kavya. I'm from Bardha University. And today I'm going to present about apoptosis inducing effect of diallyl disulfide on human breast cancer cell line. So, breast cancer is one of the most common forms of cancer that occur in women. So, it is a global public health concern, as many women with breast cancer, cancers are affected by breast cancer. So, therefore, treatment for breast cancer is inevitable. So, the plant compound said to possess various properties which can inhibit the tumor growth. So among them, an organosulfur compound disulfide seems to be a promising anti-cancer drug which can halt cancer cell progression and bring apoptosis in cancer cells. So my present study and objective was to analyze the cytotoxicity and apoptotic effect of DAS on MDMB231 breast cancer cell lines. So my materials are diallyl disulfide and MDT, DMAM, trypsin, EDTA, penicillin, streptomycin, and there are things which are used for cell maintenance. So coming to cell viability assay, I have an MDT assay which measures the cell viability based on redox potential of the cell. So MDMB231 cell lines were seeded with density of 10,000 cells per well, 96 plate per well plates, and it was allowed to grow for 24 hours. And different concentrations of the DAX are treated, administered to the cells. So after the medium was replaced with 100 ml of MTT reagent, 5 mg per ml of concentration in each well, and the cells were incubated at the degree Celsius for 4 to 5 hours. So after incubation, the 100 ml of BMS2 was added, which solidifies the fluorescent crystals, and absorbance was recorded at 570 nanometer using multiple reader. And the viability was calculated using the following formula. The apoptosis assay, the percentage of apoptotic cells were found using the multifunctional Muse annexin 5 Dexel kit, which was got from Merck millipore, and 1 into 10 power 5, that is 1 lakh cells were seeded into media, 6 plate media, containing media, and after incubation for 24 hours, the same media was discarded and the cells were treated with gas, that is 1.5 micromolar, which was got from the MTT assay. So the cells were then incubated for 12 and 24 hours respectively. The assay was performed following the reagent protocol, with addition of 100 ml of cells in suspension to each tube and 100 ml of the heat reagent. Samples were incubated for 20 minutes at room temperature in dark, and the percentage of apoptotic cells were analyzed using the Muse cell analyzer. And the cell cycle analysis, 1 into 10 power 6 of MDMB231 cell lines were seeded in 6 well plates. After incubation for 24 hours, the cell media was discarded and the cells were treated with DAX, and the cells were incubated for 12 and 24 hours. The pellets were obtained and were fixed with chill ethanol, 70% ethanol, and the same cell cycle reagent was procured from Merck millipore, and the protocol was followed. And coming to results and discussion, my MBT assay was based on time-defined manner. So at 24 hours, the percentage of cell inhibition was more compared to that of the control and 12 hours. And apoptosis assay, the dot plot represents the stage of apoptosis by the diallyl disulfide in time-defined manner. So my lower left quadrant in control has the more live cells compared to the right top of the control cell. That is the apoptosis of dead cells. So To conclude this apoptosis assay, my gas gave a better apoptosis spelling 24 hours. That is that it killed about 19.47% of cells. And in cell cycle analysis, this histogram represents the disulfide. It asks the cell cycle in G2M phase. So the more cells were answered at G2M phase in 24 hours compared to that of the control. To conclude, apoptosis results show 24-hour treatment with DANS to be more effective, and the cell cycle results shows DANS is a potent anti-cancer agent which can inhibit cancer cell progression via cell cycle arrest in G2M phase. In future, this formulation can be combined with other therapeutic targets to give a better outcome.
Thank you. Your allotted ma'am, is there any question?
Yeah, come. You have done from which department you are going?
I'm from Biochemistry department, ma'am.
Okay. So, what type of cells you have used?
Mine is breast cancer cells from MDMV2J1.
Your voice is not clear.
Yeah, they're taking the order.
Is it for me or for others?
Can you hear me?
Yeah, we can hear you, but your audio is fine.
I will.
Two instruments.
Yes, ma'am. Yes, ma'am. Yeah, that is. This is the cell analyzer, ma'am. Instead of flow cytometer, we can use the cell analyzer. We can just view the cell cycle, cell apoptosis, and much more in this instrument, ma'am. But we can't add the dyes here. We can't see the dyes here. But still, we can conclude that we have conclude the apoptosis cell cycle and all. It's a kit method, ma'am. With an instrument.
Oh.
Thank you.
Yeah,
our next candidate is Kanaga from government.
Kanaga Lakshmi doesn't have an internet connectivity and she wants to present tomorrow. She just called me.
Okay,
ma'am. I don't see the next candidate also. Can you just announce whether Arya Kannan is here in another name? Arya Kannan is here. Okay, if she is also not there, then we'll go to the next candidate. It is Daisy Priscilla. I have already made her the co-host. Yeah, Daisy Priscilla. Daisy Priscilla, you can.
Good evening, ma'am. Can you hear me, ma'am?
Yes, we can hear you.
Yes, ma'am. Is my PPT visible, ma'am?
Yes, it is visible.
Yes, ma'am.
So, good evening, ma'am. Thank you for giving me this opportunity to present my work. So, I am Daisy Priscilla, pursuing my PhD under the guidance of Dr. Anita T.S. From CADRF, Sri Balaji Vidyapith. So, my research topic is a cocktail of marine derivative and bioactive flavonoid potentiates the anti-tumor activity of temozolomide via PI3K-AKT-mTOR pathway in vivo study. So coming to the study background, glioblastoma multiforme is one of the most aggressive and invasive brain tumor, which constitute about 60% of the primary brain tumors. And according to World Health Organization, it is categorized as grade 4 glioma, which is the most aggressive and malignant grade. So the epidemiology is every 100,000 people in each and every year, 2 to 3 cases have been reported as GBM. And preferably, adults are more often affected by this particular disorder, and the median overall survival is following initial prognosis, it is just 12 to 15 months. So the most common diagnostic technique is CT followed by MRI and PET scanning. So the only like standard drug for GBM till date is temozolomide, which is a alkylating agent basically. So it crosses the blood-brain barrier and it is converted into active metabolite called MTIC, where it induces its effect by methylating the DNA base pairs, leading to apoptosis. But currently the cells, GBM cells, have developed resistance towards this drug, and these are all the major agents which is contributing to this temozolomide resistance. So this is just a pictorial representation of the current state of art treatment. So initially followed by surgery, chemo and radiotherapy is given. However, even after these treatment strategies, there is a relapse of tumor, and this is where there is a need for the new emerging strategies. And CRISPR-based knockout, immunotherapy, microRNA technology, and virotherapy have been tried, like new therapeutic strategies. And one among those is a combinatory therapy, which I'm going to discuss today. So I have chosen naringenin and chloroglucinol for to synergize with temozolomide, because naringenin has been already reported to inhibit this PI3K-AKT pathway in CT colon cancer cells, and likewise chloroglucinol is also reported to reduce PI3K-AKT expression levels and mTOR levels. So the broad objective of my study was to explore the synergistic effect of naringenin and chloroglucinol along with temozolomide by modulating PI3K-AKT mTOR pathway in glioma model. So we have chosen this particular pathway because in a majority of GBM cases, PI3K-AKT mTOR pathway is mutated, and this pathway is mainly involved in the regulation of tumor cell growth, proliferation, and survival. So targeting this pathway would improve the patient outcome and reduce the pathogenesis of GBM. So this is my research approach. In case of GBM tumor, the PI3K phosphorylates and activates AKT, which in turn activates mTOR. So this prevents apoptosis by inhibiting BAX or the pro-apoptotic effectors, and thereby preventing apoptosis and inducing tumor cell survival. So the proposed mechanism of action of our combinatory treatment would be to inhibit either PI3K, AKT, or mTOR expression levels, and thereby induce apoptosis. So this is the tumor engraftment overview. So using a stereotactic apparatus, 1 × 10⁶ rat glioma cells will have been implanted into the right striatum. Points were with reference to Bregma point, and following injection, tumor induction, after 20 days of tumor induction, the animals were exposed to the treatment groups temozolomide, naringenin, chloroglucinol, and a combination of all the three. And following 20 days of treatment, we performed hematoxin eosin staining, IHC, QRT-PCR, and Western blotting. So the results of our hematoxin eosin staining is displayed here. So when compared with, this is a normal control rat, when compared with the normal control rat, the brain of tumor control rat has a dense nuclear distribution with tumor regions highlighted here. And when treated with temozolomide alone and naringenin alone, chloroglucinol alone, there was a reduction in this nuclear condensation, and very tiny areas of gliosis was observed in the triple drug combination treated group, which is TNP, treated with temozolomide, naringenin, chloroglucinol together. And likewise, so IHC for Ka67 was performed. Ka67 is a nuclear stain. So you can see in this tumor control group, the nuclear positivity for Ka67 is about 90%, because all the cells are actively proliferating. When compared with the tumor control group, the triple drug combination has shown a least nuclear positivity. Likewise, for angiogenesis marker, we have for VEGF marker, we have performed IHC staining, and here it can be seen in the tumor control that there is high neovascularization and hello.
Daisy, your time is almost over. Would you like to conclude?
Okay. So, and similarly, we have carried out gene and protein expression levels for the markers of PI3K pathway, and in this it can be seen that the levels of PTEN has been increased when compared with the other markers like AKT, PDK1, mTOR has gradually decreased in all the treatment groups, and it is statistically significant in the triple drug combination in both gene and protein expression. Likewise, the levels of apoptotic markers were also assessed. In this, the levels of caspase-9 has been increased in the triple drug treatment group, which suggests that apoptosis has been undergone in these cells. So in summary, this study has shown that along with temozolomide, naringenin, and chloroglucinol has not only regulated PI3K, AKT, mTOR signaling pathway, but it has also induced apoptosis in GBM, as shown by elevated levels of caspase-9, BAX, and BAT. So to take it forward, clinical trials investigating the safety and therapeutic effect of this combination approach is a serious priority in future. So we would like to.
Ma'am, is there any question?
So Daisy, so
yes,
you want to propose that both, so right after the radiation therapy and resection, you want to propose that both the drugs will be given together.
Yes,
right?
ma'am. Yes, ma'am.
Okay. Yeah, nice. You have nice data actually. Yeah.
Thank you. Thank you, ma'am.
Have you published this data, ma'am?
Only with Naranjan alone I have published, ma'am.
Okay.
So this
Which
work
journal?
it is in Research Journal of Biotechnology.
Okay.
Yes, ma'am.
Yeah, good.
Thank you.
Yeah.
Thank you, Daisy.
Thank
Now I would like to call upon our next candidate, Shafia.
Neha, 1 minute. Before that, Kanaga Lakshmi is available now. So can we call her first?
Okay, ma'am.
Yeah. So I have made her the co-host, so we can just call
Kanaga Lakshmi.
So candidate is Kanaga Lakshmi from Government Arts College, Krishnagiri.
Yeah.
From Krishnagiri. Okay.
Kanaga Lakshmi, you can share your screen. Kanaga Lakshmi, please unmute yourself.
Yes, ma'am. Now I'm going to share my PPT. Madam, my PPTs are visible?
Yes, ma'am. Yes. Please proceed. I think you can put it in screen mode, full screen.
Full screen. Okay. Okay, ma'am?
Yeah, yes, ma'am.
Okay, ma'am. I am Kanaga Lakshmi from Krishnagiri Government Arts College for Women. Today I am going to defend gallic acid induces apoptosis and inhibits cyclin B1 gene expression on skin carcinoma human keratinocyte cells. Skin cancer is one of the majorities of universal cancers in the earth. Melanoma and non-melanoma are two foremost divisions of skin cancer. In non-melanoma tumors, basal carcinoma and squamous cell carcinoma are the most common type. In men and women, 19th most frequently occurring skin carcinoma is melanoma. It is 300,000 latest cases in 2018 by means of more than 1 million examination throughout the international level in 2018. Non-melanoma skin cancer is the fifth most generally arising cancer in men and women. The uppermost incidence of melanoma and non-melanoma in 2018 were found in Australia, after that New Zealand. According to Cancer Facts and Figures, 2020 report states that in US, 100,350 novel types of melanoma were discovered and 6,850 populations were passed away by this disorder. Annually, several additional 5 million skin cancer cases that are identified might be treated by shielding skin from extreme solar exposure and avoiding interior tanning strategy. Globacan 2018 reported in India, new cases of melanoma of skin is 3,048, death risk is 2,053, and 5-year prevalence of all age group is 7,331. Ultraviolet radiation from sun exposure, utilization of synthetic UV tanning beds and lamps, exposure to individual or familial genetic syndromes of melanoma, occurrence of several, at least more than 50 moles, people who are sun responsive, who have extreme sunburns or skin carcinoma, then individual with a very low resistance system and some environmental pollutants are also major risk factors for melanoma. The natural remedy is one of the mainly used balancing and substitute medicine to treat many different health conditions. Therapeutic herbs and their secondary metabolites are being gradually more acknowledged and valuable predictable cancer treatments. Phenolic acids are potent secondary metabolites, mainly spread in grains, vegetables, legumes, fruits, and cereals. One of the most important phenolic acid, gallic acid, It is also recognized as 3,4,5-hydroxybenzoic acid, and mainly tea leaves, leaves of coca, black radish, and some other fruits and vegetables contains gallic acid. Sun protection factor of gallic acid was determined by UV spectrophotometer. The SPF value of gallic acid was found to be 22.49. Scavenging ability of gallic acid on hydroxy radical, superoxide anion radical, and DDTH and ABTS radical was enhanced in concentration dependent manner. Here, the IC50 value of gallic acid on hydroxy radical scavenging activity was found to be 40 microgram per ml. It was found in superoxide preventing activity was 38.5 microgram per ml. And in DPPH radical scavenging activity, IC50 value of gallic acid was found to be 40 microgram per ml. In ABTS scavenging activity, IC50 value of gallic acid was found to be 43.5 microgram per ml. Then all the free radical scavenging ability of gallic acid was compared with standard ascorbic acid. Cytotoxicity effect of gallic acid on skin carcinoma, Hecat cell line was analyzed by MTT assay. Then percentage of cytotoxicity of gallic acid was increased in a concentration dependent manner. Then 50% of cytotoxic concentration of gallic acid was found to be 75 microgram per ml. Compared to control cells, treatment of 75 microgram per ml and 150 microgram per ml of gallic acid induced necrotic, early apoptotic, and apoptotic Hecat cells. Then apoptosis study was expressed by annexin 5 and propylium iodide dye. Then gallic acid on cyclin B1 mRNA expression was studied by RT-PCR. Here, the 75 microgram per ml of gallic acid showed downregulation of the cyclin B1 gene expression, and this result was compared to control and positive control H2O2 treated cells. Our results summarize that gallic acid exhibits better sun protection factor, inhibits free radical production, increasing cytotoxicity in skin carcinoma, Hecat cell line, downregulates the cyclin B1 gene expression in Hecat cells. Hence, gallic acid could be considered as a photoprotector and anti-neoplastic agent. However, to study the protective effect of gallic acid on oxidative damages, inflammatory and apoptotic signaling in experimental animal model are warranted. My suggestions and recommendation about these results in scientific level, GI exhibits various radical formation and downregulates the cyclin B1 gene expression. It is necessary to study its influence on some other molecular events such as apoptotic, inflammatory, and photoaging signaling markers before claiming this compound as a potential anti-cancer agent. In commercial level, GA containing sunscreen lotion may be prepared and employed against UV radiation-induced sunburn, erythema, edema, photoaging. In societal level, peoples may consume fruits and vegetables rich gallic acid in order to protect from environmental mediated pathological conditions. Thank you, ma'am.
Thank you, madam. Ma'am, does gallic acid have any effect on melanin production in our skin?
Madam, that was I'm not studied, madam, but it is a very good antioxidant agent, madam. So it is prevent the free radical formation in our skin, ma'am.
So you can apply it directly onto the skin externally.
Ma'am, for that we want to prepare that SPF based chemical formulation, ma'am, with some other high sun protective effect compound we want to prepare.
Okay, ma'am.
Otherwise it can't work, ma'am.
Okay, ma'am. Good. Thank you. Any other questions?
Thank you,
Asmita, ma'am?
No, madam. No questions. Yeah.
Okay, then thank you, ma'am.
Thank you, ma'am.
Another Lakshmi. Now,
thank you, ma'am.
I would like to call upon
before that, Arya Kannan. Yes, Arya Kannan is there.
I would like to call upon Arya Kannan from Bhattiyar University.
Yes, Arya Kannan.
afternoon, ma'am. Hello.
Yes, you can hear
Good
us.
afternoon. Am I audible?
You're audible, but the voice is not very clear.
Okay, ma'am. Now that is clear, ma'am.
Dialogue raised.
Yeah, dialogue raised.
Some background noise is there. Yeah.
Hello.
Audio.
By any chance, have you switched on two devices, Arya, in the same room? Only both get all the problem.
Can we move on to the next speaker and give her a chance later?
Okay, ma'am.
Arya Has she left the meeting?
No, she's there.
Maybe you can ask her to, sign out and then sign back in. That might help.
Yeah.
Okay, so can we call the next presenter then?
Yes, ma'am.
She can present maybe next.
No.
Yeah, it is Shafia. I think he's here. I will just.
Shafia Zabin Mubarak from Bhartiya University.
Again, please pronounce, ma'am.
Shafia Zabin Mubarak from Bhartiya University.
Okay. Yeah. Is she there?
Yeah. I made her a co-host. Okay. Not her a co-host.
Shafia Zabin.
Yeah, she's there.
Shafia Zabin, you're there?
Please unmute yourself, Shafia.
Shafia Zabin?
Yes, ma'am. The presentation is visible to all, ma'am?
Not yet, but we are waiting. Now we see some. Yeah, now it is visible.
So good afternoon to all. The title is Identification of Novel Scaffold to Activate GRP94 and HSP90 chaperone based on virtual screening, pharmacophore hypothesis, and QSAR model. So ER stress is a major pathological condition which involved in various disease conditions such as diabetics, preeclampsia, inflammation, mood degenerative disorder, cancer, etc. So the stress which occurs due to protein misfolding and aggregation. It also activates the UPR pathway, that is unfolded protein response pathway, and it increase or induce certain chaperones such as IRE1 ATF6 work. So ATF6C is one of a transcription factor, also increases other chaperones, ER chaperones like GRP78 and 94. So most inducible ER proteins are GRP94, so which are the hallmarks of physiological and pathological role for this UPR pathway. So UPR is a signaling pathway which acts as a reflex in ER stress and restores the homeostasis. And the most well-known protein is a glucose regulator protein that is GRP. So with 94 kilodalton, GRP94, which together enhances the protein folding machine. So otherwise the UPR will induce the ER stress and at last it will result in cell death or apoptosis. So it also results in activation of certain chaperones apart. GRP94 is 78, ATF6, IRE1, 1 alpha, HSP70.
So this is the overview of the activation of the UPR. So, so this is the ER lumen in which BIP will be dissociated from the IR when PERC ATF6. ATF6 will be cleaved out and it will result in GRP94 as well as in IRE1. It results in unconventional splicing variant and it results in activation of several chaperones, and on the upper it also results in endoplasmic reticulum associated degradation, that is a protein degradation. So GRP94 is a glucose regulator protein pathway. It is present in ER lumen and it is with the heat shock protein 90 family members. And ATP dependent molecular chaperones that regulate the stability and function of the client proteins involved in growth, survival, adaptation of the cancer cells to the cellular stress. So GRP94 is the key downstream chaperone in the UPR. So the main objective is to screen or identify the potential small molecules as novel agonists for this particular protein and to increase the efficiency of the protein folding and to elevate the cellular ER stress through approaches namely virtual screening and quasar prediction. So the well-known compound NECA, ethyl carboxyadenosine. So taken chosen as an agonist for the GRP94 for making a model, and it was done using the core hopping. So the workflow is under this. The core hopping, the compounds were collected and it was modeled using the core hopping model, and at last the pharmacophore hypothesis were validated, and also QSAR, quantitative structure activity relationship, also validated. And at last, the screening were done using the MASTO, that is virtual screening, using an extra precision XP. And at last, drug likeness were predicted using the software of ADMET properties. So the results are from the co-hopping scaffolds, we yielded 35 compounds and it composed of both the constraint, that is Ligon-based constraint as well as isosteric constraint. So from this we modeled a pharmacophore. So these are the list of the pharmacophore we identified, but we chosen the hypothesis as ADHR2, that is acceptor, donor, hydrophobic groups and alkaline groups based on survival score, site, vector, volume and matches. So this is the overall active superposition of ADHR_2, that is a pharmacophore hypothesis we built towards this. Next is QSR validation. The QSR validation is we have done using the standard deviation R square, F value, P, and root mean square error value, and Pearson. So based on the score also we validated that this ID will be a good for using as a model. So this is a 3D QSR model, and these are some of the parameters which we have approached towards for building this hypothesis. And virtual screening analysis were done for the compounds. The most top lead compounds are the zinc 6765024, and then
you have 30 seconds to conclude.
Okay. So these are the compounds we have selected, and with this, these are the 2D and 3D interactions. And we have also checked for the drug availability. So it has come under the criteria. So from this, based on core hopping, ADHR2 were yielded and it was selected. So among the screen components, the top 3 were identified as a potent lead molecules. It showed the hydrogen bond interaction with these residues, and further in vitro validation will be executed to evaluate the efficiency of small molecules in reducing the protein misfolding associated with ER stress. Thank you.
Thank you, Ms. Shafia. Ma'am, is there any question?
Hi, Shafia. So what will be the kind of in vitro studies that you will do after this?
We can approach towards the cell line validation. And we have to approach towards the PCR work, and we can validate through the in vitro studies also, through the use of a mouse model, we can validate it.
Would you want to check direct binding, like whatever bindings you are finding here? Would you check for direct binding of the your ligands with the protein?
Ma'am, it is based on their active site residues. We selected those compounds.
Right. That is, would you try to experimentally validate that?
Ma'am, that is based on because the known compound is a necamine. So the active site residues is ASL 162 and tyrosine 200 and tryptophan. So, but we have modeled from, we have acted this as a template, and we modeled more than 35 scaffold. So out of this, we have put the docking, and exactly it had binded to the particular residues which was already deposited in the databases. But we have modeled using the co-working scaffold.
Okay. Okay. All right. Yeah. Thank you.
Thank you. Thank you.
Thank you, Ms. Shafia. So
thank you.
Now I would like to call upon Arya Kannan from Bhartiya University. Arya, are we audible?
Arya is here. Arya, can you unmute your mic? Yeah, she's presenting.
Good afternoon, ma'am. Hello.
Good afternoon.
Good afternoon. Shall I start?
Yes, please.
Okay, myself, Arya Kannan, I'm Department of Biochemistry, Biochemistry University. Here I am presenting one of the research work, apoptosis reduction in human line adenocarcinoma cells, especially in AA549 cell line by the dialyl disulfide. Introduction. Cells is the second cancer is the second leading cause of death globally, accounting for an estimated 9.6 million deaths in 2018. Lung cancer is the prime cause of cancer death around the world. Epidemiological studies shows that the incidence of lung cancer increases and that lead to more death than breast, prostate, and colon cancer combined. So here I'm focusing on the phytochemical products, how it affects the cancer treatment, or what is the effect of the materials, phytomedicine for the future studies. So here I'm looking with the garlic product. So here the garlic possesses cancer preventive potential and significant enhancing effect on human immune system. So diaryl disulfide, it's an organosulfur compound and it has the ability to cure and prevent the growth and division of cancerous cells. So my main objective here I am presenting is to evaluate the cytotoxicity effect of diaryl sulfide on A549 cell line and evaluation of apoptosis and cell cycle arrest. So yeah, materials here I'm using for mainly focusing on the A549 cell lines. It's purchased from the NCCS Pune, and cells are cultured in BMEM with 10% FPS and grow at 37 degrees Celsius in humidified air with 5% carbon dioxide. So first we check with the ABT assay. So here we placed 10,000 cells per well in 96 well plate and look for the MTT assay. So anti-cancer activity of the gas was tested with MTT in A549 cell line. The percentage of ability was calculated using the following formula. That is the mean OD of the individual test group divided mean OD of the control into 100. Then we are looking for the cell apoptosis assay. So apoptosis assay is analyzed using a multifunctional new cell analyzer. So according to this, there are user's guide and manufacturer's instruction. So here the cells are seeded in a density of 1 into 10 to 6 cells per well into 6 well plate and treated with gas at various concentration 25, 50, 75 millimolar for 24 hours at 37 degrees Celsius. For the apoptosis, the floating and crystallized cells were collected and washed once with the cold TDS centrifuged. Centrifuged at a reduction of 5 minutes at room temperature. The pellets were suspended in 1 ml of medium and 100 ml of suspension was transferred to new tube, and then 100 ml of new initial analyzer kit reagent was added to each tube and incubated for 20 minutes. Dark and stained samples are analyzed with mini cytometry analyzers. Then the cell cycle analysis is carried out. The same amount of cells, that is a 1 into 10⁶ cells per well for the 6 well plate, and treated with the 25, 50, 75 millimolar DATs for the 24 hours, and the floating or adherent cells were collected and washed twice with PTS and 300 centrifugation at room temperature, and cells fixed at 70% ethanol for 3 hours, and the cells were washed with PTS, and 200 ml of suspension was transferred to a test tube, and next cycle reagent kit was added to the each tube and incubated for 30 minutes at room temperature in dark. So result from our first MTT assay, we focus on the 25, 50, 70, 100 concentration. So a gradual increasing of the percentage of inhibition we get. So according to our study, the triplicates, so we get a mean value of each one. For example, for 25 we get a 33.2, and then for 40 we get 45.7. So in 50% of inhibition was occurs at the 82.3
Arya, you have 2 seconds to conclude.
Concluding, ma'am. So next our apoptosis result. So apoptosis result, we focus on the 25, 50, 75. So as per the concentration increases, the apoptosis range. So here is an early apoptosis. So in 25 there is a late apoptosis is very less, but as compared with the 75 millimolar, the late apoptosis is gradually increases. So the apoptosis range increases as depending upon the concentration depending manner. So in 50 millimolar it is increasing, and for the 75 millimolar it is gradually increases, and it obtains and 19.56% late apoptosis occurs. Okay, for the cell cycle analysis, in concentration dependent manner, for the 25, 50, 75, a gradual decreasing of the G0, G1 phase, and a gradual increasing of G2, EM phase. So here the
Miss Arya,
yes, ma'am. So to sum up, the DAX shows a potential anti-cancer effect in several tumors. As our investigation proved that DAX induced apoptosis in A. pylori and leg cancer in a concentration-dependent manner. So DAX induces a significant apoptosis induction, and the cell cycle analysis shows that the significant cell arrest at G2M phase, and it causes a 75 millimolar concentration gives maximum cell cycle arrest in our cell cycle analysis. So further we are concluding, further we elaborate our studies in the time dependent manner. And we are focusing for the targeted drug delivery system with the nano carriers for our future studies.
Thank you, Ms.
Thank you.
Ma'am, is there any?
Hello, Arya. Okay. Yeah. So, what do you think is the most novel observation in your findings?
Actually, we are observing a gradual increasing of our drug concentration, and we get anti-cancer activity at 82.3 millimolar. So we are using that millimolar for our further studies, like for our apoptosis analysis, and again we are focusing on the making studies. So further our future study means
how does it stand with what is available in the literature?
I'm not audible, ma'am.
No, how does it stand with what is available in the literature? Because disulfide, I think, is used.
Yeah, yes, ma'am. It's already done, ma'am. In so many literature we get, but here our work is first preliminary stage. I'm focusing here, ma'am. But our work is It's an encapsulation stage. Encapsulation means encapsulation with the other compound and in liposomal encapsulation drug delivery system. So, but it's my part of work, not an exactly work, ma'am. It's a preliminary observation only, not the whole research program.
Okay, yeah. Dr. Lata?
So here I am focusing means I have to again ensure that the concentration for my studies.
Okay, yeah. Thank you.
Thank you.
Okay, thank you. Maybe we'll ask the next question.
Now I would like to ask Miss Aruna Devi from Annamalai.
There is a lot of echo happening. I don't know.
There is audio.
Problem with the mic. I muted. Are you connected?
I will.
Miss Aruna, you can unmute yourself. Am I audible?
Yes, Aruna, you are audible. You are slightly visible.
Yeah. Good evening to one and all. This is Aruna Devi,
One minute.
PhD.
And put it in a full
Yeah.
screen mode and then you can.
Do you visible now?
Yes, it is visible, but it is not in the full screen mode. But anyway, you can present. Aruna, you can continue. No problem.
Okay.
Start.
Yeah. Good evening to one and all. This is Aruna Devi, PhD scholar from Annamalai University. I'm going to present the topic entitled Theophlavine, a black tea polyphenol, protects nigral dopaminergic neurons against chronic MPTP-induced Parkinson's disease. Parkinson's is the second most common neurodegenerative disease that affects the movements of the aged peoples. The main pathology of the Parkinson's disease is a loss of nigral dopaminergic neurons and striatal dopamine depletion with consequent disruption of the motor functions. It affects about 1% of the people above 55 years and 4% of the people above 80 years. It is estimated that about 6 million of the people were affected worldwide, and it is expected to be double by the year of 2020. Some of the symptoms that traps the movement of Parkinson's disease patients are tremor, rigidity, akinesia, and postural instability. Various pathological processes are involved in the cause and progression of Parkinson's disease. It includes mitochondrial dysfunction, oxidative stress, proteotoxicity, calcium ion homeostasis, increased iron content, inflammation, and apoptosis. Most of the mechanisms were studied using the models of Parkinson's disease, such as postmortem studies, cellular models, and animal models. Number of neurotoxins are employed to create animal models of Parkinson's disease in lower vertebrates such as Drosophila, rodents like rats and mice, and non-human primates like monkeys. Some of the neurotoxins used to induce Parkinson's disease are 6-OHDA, MPTP, rotenone, paraquat, and mannan. There is no permanent cure for Parkinson's disease, where the levodopa is the golden standard drug used to treat the Parkinson's disease, and also the long-term administration of the levodopa leads to motor complication like L-dopa-induced dyskinesia. Some of the surgical treatments are neural transplantation, deep brain stimulation, and stem cell transplantation. These surgical treatments are only under the experimental level. All these medications and treatments results in severe side effects where the drug blocks neurodegeneration is a milestone of Parkinson's disease. Now, the global scenario of medical research is towards herbal or alternative symptoms of medicine. So, plant-derived products are gaining more attention in neuroprotective studies. Tea is in the world next to the water. It contains apoptotic, anti-inflammatory, and it contains more antioxidant contents. Tan reported that consumption of 2 or more cups of tea daily reduced the risk of Parkinson's disease, and Chaturvedi reported that tea extract possesses neuroprotective function against 6-OHDA-induced Parkinson's disease neurodegeneration in animal models. These are some antioxidant content-rich beverages. 2 cups of black tea is equal to 1 glass of wine, 7 glasses of orange juice, and 20 glasses of apple juice. Where tea is a low-cost beverage we can consume daily, which improves the antioxidant that prevent the cell damage and apoptosis. Some types of tea are white tea, green tea, oolong tea, and black tea. Some of the major components of black tea are, these are the major components, and we have chosen theophlavin as a major component, where it is present 3 to 6% in the whole 100 gram of black tea extract. The major theophlavin derivative in black tea are theophlavin, theophlavin 3 gallate, theophlavin 3-gallate, and theophlavin 3,3-gallate, where it is generally considered to be the most bioactive and effective against neurodegenerative diseases. And it contains more pharmacologies like hepatoprotectivity, anti-inflammatory, anti-apoptotic, anti-allergic, and also it contains more antioxidant contents. We have used, ma'am?
You wanted to change your slides. The slides are not moving. Yeah, it's still on the cover slide, first slide.
My God. I'm moving the slides, but I don't know what's the issue.
Can you stop sharing? Can you stop the share and then redo it?
Yeah, sure, ma'am. The slides visible now?
Yeah, we can see the slides. Yeah, now you can move the slides and check.
Okay.
Change the slides now.
Is it moving?
No, it is not.
Okay.
Why don't you put it in full screen mode? I don't know.
Yes, ma'am. It is in the full screen mode.
Oh, God. Do one thing, then share the whole screen.
Presentation mode.
Yeah, presentation mode.
Is it visible now?
We don't see anything now, Aruna. You're not sharing.
Is the slide visible now?
No, not yet, ma.
Lata madam, can you see anything?
No, I am not seeing anything. I'm just waiting for your comment.
Aruna, actually keep the slide open in the desktop and then click share.
Okay.
Actually, she was in the first slide itself.
Yeah, I understand. Now we cannot see even the slide.
So we have 1, 2, 3, 4, 5, 6 more.
Yeah.
Do we have a tea break?
Actually, we had it, right? Now, if you want, we can.
Okay, let me share.
Oh, yes, please.
Yeah, you stop sharing first.
No, ma, your screen is not visible.
Aruna, try once. If not possible, you can present later.
நான் படிச்சிட்டதுல அந்த 2 மார்க்ஸ் क्वेश्चन எல்லாத்தையும்
Shall we move on and come back to her?
Yes.
Yeah.
Maybe Neha, you can call the next speaker. Aruna, we'll call you later.
Thank you, Miss Aruna. So our next candidate is Mr. Gobinath Murugan from University of Madras. Mr. Gobinath, please start your presentation.
Slides are visible, ma'am. Am I audible?
Your sound, your voice is too low.
Okay. Now?
Yeah, it's okay.
Shall I present now?
Yeah, I can't hear
Just
anything.
maybe increase the volume, or I think your volume is less. Just check it, or else you can go near the mic and talk. But we can see your screen.
I mean, I'm connected headphones, but
Yeah, fine. You can stand close to the mic and then talk maybe.
ma'am.
Yes,
Okay. Shall I proceed, ma'am?
please. Now it's very clear.
Okay. Good evening, all. I am Gopinath from University of Madras, and my topic is.
Can you please put in the full slide mode?
Yes, ma'am.
This is the one. The present. Yeah.
Is it visible, ma'am?
Yes, you can proceed.
Yes. Good evening, all. I am Gopinath from University of Madras. My topic is interaction of phytochemicals of Clitoria tenuti with glycogen synthase 3 in silico approach. Introduction: Glycogen synthase 3 is a serotonin kinase and signal mediator, plays a crucial role in various signaling pathways, including insulin signaling, neurotrophic factor signaling, BNT signaling, neurotransmitting signaling, and microtubule dynamics. GSK3 comprise two highly similar paralogs, GSK3 alpha and GSK3 beta, ubiquitously expressed in all tissues. Dysfunction of GSK3 has been implicated in pathological conditions such as Alzheimer's disease, Parkinson's disease, type 2 diabetes, bipolar disorders, and cancers. Particularly, GSK3 involved in the hyperphosphorylation of tau proteins, memory impairment, and increased production of beta amyloids and inflammatory responses. Moreover, GSK3 is a key mediator of apoptosis and thereby might directly contribute to the neuronal loss in Alzheimer's disease. Hence, GSK3 has been identified as a promising target, and inhibition of its activity is a key strategy in the treatment of neurodegenerative diseases. Clitoria tenuti is a very well-known Ayurvedic medicine, have long been widely used as brain tonic, and it is believed to promote memory and intelligence. This plant is an essential component in brain tonic called as Medhaya Rasayana, which has been used for centuries in regenerating neurons and treating neurological diseases. Aim of this study is to investigate molecular interaction of bioactive phytochemicals from Clitoria tenuti against GSK3 beta by in silico molecular docking approach. These are the compounds going to dock against GSK3. Methods used in this study are phytochemical compounds of Clitoria tenuti root extract retrieved from PubChem database and prepared by using OpenBabel. All the 2D structures are converted to 3D using OpenBabel, and the energy minimization was done by using Avogadro software. Then the protein structure GSK3 beta was downloaded from protein databank, and all the co-crystallized ligand and water molecules are removed by using FIMAL, and molecular docking of compounds against GSK3 beta was carried out by using Autodock 4.2. Then finally the interaction mode of phytochemical compounds with GSK3 beta analyzed by using FIMAL. This is the active site of GSK3 beta. All the phytochemical compounds were docked in this site. Results and discussion. This table represents the docking results of compounds against GSK3 beta, in which quercetin showed lowest binding energy and inhibition rate among the compounds. and chemophenol monoglucosidase and malvidin 3-glucoside exhibit same binding energy and inhibition rate. Delphinidine diglucoside is found to be highest binding energy and inhibition rate among the compounds. This is the binding interaction of quercetin with GSK3 alpha 3 beta. In these pictures, quercetin hydrogen bond interaction forms hydrogen bond interaction with GSK3 beta in the active site. It established hydrogen bonds with lysine 85 and glutamic acid 97 and aspartic acid 133 and valine 135. And this figure represents how quercetin occupies the active site cavity of GSK3 due to the less flexibility of quercetin among the compounds. It tightly bound to the active site of GSK3 beta and blocks the entry of substrate ATP into the active site, so that which inhibit the phosphorylation of GSK3 beta. Conclusion: the phytochemical compounds from Clitoria tantia showed optimal interaction with GSK3. Among the compounds, quercetin is a flavonoid that showed good binding energy and least inhibition constant, hence could possibly be ideal inhibitor because of its antioxidant and binding property. Further in vitro and in vivo studies is necessary to obtain and confirm these results. These are the references. Thank you, ma'am.
Thank you, Mr. Gobinath Murugan. Ma'am, is there any questions?
No, actually, he's trying to plan in vitro and in vivo studies. If so, how you want to proceed?
Now itself, I am not going to do any in vitro in silico cells, but I plan to do molecular dynamic simulation, like further in silico studies, ma'am, to confirm the stability of the compound in the inactive site.
You have studied ADMET and all absorption. ADMET studies.
No, ma'am. No, ma'am. No, ma'am.
Okay. So this is a preliminary we have studied.
Yes, ma'am.
Okay. Any other questions from Asmita, ma'am?
Hello, Gopinath. Did you actually try multiple compounds, or you just tried quercetin?
I tried totally 7 compounds against GSK3, ma'am, in which quercetin showed low binding energy and low inhibition constant, ma'am.
Yes, it was the best out of the list.
Yes, ma'am.
Okay. So then, since quercetin is prevalent in most other plants and other sources also.
Yes, ma'am. Flavonoids. If quercetin is a flavonoid commonly present in various foods and all the foods.
Yeah, there is no point of specificity with that plant that you are talking about, right? Yeah.
But further, in vitro studies may confirm these results, ma'am.
The other compounds.
Yes, ma'am.
Oh, yeah, sure. Yes.
Thank you, ma'am.
Thank you.
Thank you, Mr. Gobinath and ma'am. Now I would like to call Miss Aruna Devi Selvaraj from Annamalai University. Miss Aruna, you can start with your presentation. Miss Aruna, please share your screen.
Yes, ma'am. Is this visible, ma'am? Hello?
Not yet, but we'll see.
Is the screen is visible, ma'am?
No, it's not yet visible. It's showing that you are sharing something, but we can't see anything. At least I can't see anything.
So we may have to wait for a minute or so, depending upon your connectivity. We'll wait for it and see whether it's done. Aruna, actually, we could see only the black screen, but it's showing like Aruna Silvraj has started sharing screen, but we don't see anything. Can you try again? Oh, in that case, we will come to you a little later, Aruna. Maybe we can have your presentation as the last one. You just try it, okay? Aruna, are you there?
Yeah, I think we have lost her. You just move on to the next one.
Yes, that's better. Okay. Neha,
We will move on to the next presentation by Dr. Sujata from Pondicherry University. Ma'am, please share your slides. Dr. Sujata.
1 minute. Dr. Sujata. Yes, she's starting. I think your mic is muted. Are you talking? Dr.
Dr. Sujata, please unmute yourself.
Yeah, it's fine now.
Yeah.
Hello, ma'am. Good evening, ma'am. Hello, ma'am. Good evening, ma'am. I am A. Venkadeshan.
Yes.
Okay.
Project students, ma'am.
Yeah.
Okay, ma, you can start.
Let us start now, ma'am.
Yes.
Yeah.
Good evening to all. I am A. Venkadeshan, project student, Department of Chemistry, Pondicherry University. My principal investigator, Dr. V. Sujata, DST Mobility Fellowship Awardee, Department of Chemistry, Pondicherry University. Our title is Evaluation of Phytochemical Screening and Antioxidant Potential of Diosporus montana. Introduction. Medicinal plants having numerous phytochemicals. These phytochemicals having effective for cure many diseases. Phytochemicals easily reduce the free radical and other diseases. The antioxidant with free radical scavenging activities may have great relevance in the prevention of diseases in which oxidase or free radicals are impellent. So the aim of our present study to discover the good source of natural antioxidants in plants. So the current findings deals with the evaluation of higher antioxidant potential of various extract in Diosporus montana through phytochemical screening and antioxidant activities. So methodologies. So come to our results and discussion. So first study the phytochemical screening analysis. The Diosporus montana leaves, bark, and seed extract analyzed the phytochemical screening analysis. This result revealed that the numerous phytochemicals are presented in the various extract of Diosporus montana various parts. Particularly, methanol have significant new phytochemicals are presented. Next one is quantitative analysis. Estimation of total phenol and flavonoid content of leaves, bark, and seed in various extras. So these results exhibited, particularly the methanol extract showed higher content of phenol and flavonoid, followed by acetone and ethyl acetate extract. So particularly leaves and bark extract methanol extract having higher phenol flavonoid content. So next one is in vitro radical scavenging property. The in vitro radical scavenging property result exhibited, the radical scavenging activity increases, the concentration increases, the radical scavenging activity also increases. Particularly the leaves and bark methanol extract having lower size 50 value. This represented this having a higher radical scavenging ability for methanol extract of leaf and bark. So next one, reducing power activity. This reducing power activity result revealed that the concentration, the extract concentration increases, the reducing power activity also decreases, because this plant Diosporus montana extract, various extracts having lots of phytochemicals, because easily reduce the free radicals and others. So for my conclusion, Diosporus montana serves as a good source of phytochemicals, and higher amount of phenolic and flavonoid content were present, found in leaf and bark methanol extract, which is inferring the hormological properties. Leaf and bark methanol extract possesses significant antioxidant and reducing capacity. Based on the result of time, the leaf and bark methanol extract of Diosporus montana possess various phytochemicals and potent antioxidant compound and promising radical scavenging and reducing abilities. Thank you, ma'am. Thank you, Anandal.
Thank you, Dr. Sujata. Ma'am, please provide your suggestions and questions.
Okay, good. Ma'am, you have questions?
No.
Okay. So if no questions, I think he has done a good presentation. And let us ask the next speaker to talk.
Moving on to the next presentation by Mr. Murugan F. from Pondicherry University. Mr. Murugan, are you there?
From Pondicherry University, is it?
Yes, from Pondicherry University.
Okay.
Mr. Murugan?
Yeah. Now only I have made him the co-host, so he can speak now.
Sure.
Yeah, Murugan.
Yes.
Please start sharing your screen.
Sure, ma'am. Yes, I'm trying. Ma'am, am I audible?
Yes, you are audible.
Slides are visible?
No, it is not. Or maybe we have to wait for 2 minutes. Yes, it is visible now.
Thank you.
Perfect.
Good afternoon to everyone. Myself Murugan, research scholar from Department of Biotechnology, Pondicherry University. My presentation is on MIR-150 inhibits the cell proliferation by downregulating major epigenetic repressors EGH1, 2 and DNMT3A3B. Before going to results, I would like to give short notes on introduction. As we all know, in eukaryotic system, the gene expression is highly controlled at different level, includes transcriptional, post-transcriptional, translational, post-translational level. In recent studies, it is well documented that epigenetic systems also involved in gene expression by modulating the accessibility of chromatin for the gene expression. What is epigenetic system? Epigenetics is referred to as heritable changes in gene expression and chromatin without changes in the DNA sequence. This is well controlled by 3 different modifications called histone modification, RNA interference, and DNA methylation. In histone modification, these histone proteins are undergoing various post-translational modification like acetylation, methylation, etc. Whereas in case of RNA interference, both non-coding, that is the short and long non-coding RNA, are involved in regulation of gene expression. Whereas in case of DNA methylation is the process where the DNA gets methylated by a group of enzyme called DNA methyltransferase that will determine the expression of gene. So here mainly I'm focusing on microRNA, so which is controlling that histone methyltransferase called EGH1 and 2 and DNA methyltransferase 3A and 3B. So what is microRNA? MicroRNA are the small non-coding RNA which spans to 20 to 22 nucleotides in length, which is transcribed and processed by Joshua and Dicer, which will produce the mature microRNA, which will be labeled either microRNA 5P or 3P based on their orientation. This microRNA in turn is loaded with the RISC complex, and it will goes and bind to the 3' UPR of its target gene. So thereby it will inhibits gene expression by degrading its mRNA, or it will inhibit its translational repression. So in recent study, it is shown that this microRNA 150, that is MIR150, is deregulated in the most of the cancer, and it will either function as either oncogene or tumor suppressor gene, depends on the function and the cancer cell type. So results. So first we initiated our study by predicting this MIR150 binding sites in its target genes like all epigenetic repressors. We found that gene MT3A 3-UTR is harboring single binding motif for MIR150 sites, and here it is the screenshot of that prediction. And also we found that EGS2 also is containing a single binding site for MIR153P region. And also we found EGH1 also harboring 3 different binding sites for MIR155P. But unfortunately, DNMT3B doesn't have any binding site for MIR150. So as I told earlier, this both the epigenetic repressors and this MIR150s are deregulated in different cancer cell lines. So we need to find the expression level of these target and the MIR150. So here we have analyzed the differential expression of both epigenetic regulators DNMT3A, 3B, EGH1 and 2. As you can see here, there is a differential expression of all these epigenetic regulators. And also we have checked the MIR150 3P and 5P transcript level also, and we found both positive and negative correlation of this MIR150 5P 3P with respect to its target genes. So next, to validate the MIR150 binding sites on the target genes, we perform 3-UTR assay. In this, we cloned this 3-UTR of 3A, EGH1 and 2 gene under these GFP protein coding regions. So once if this microRNA is overexpressed, it will goes and bind to the 3-UTR region, and it will repress the expression of GFP protein. So as you can see here, under the overexpression of MIR150, the level of DNMT3A, AGH1 and AGS2 the level was decreased. So this confirmed that MIR150 effectively binds to the 3-UTR of DNMT3A, AGH1 and 2. So next we check this transcripted level of this target gene upon the overexpression of MIR150. So we overexpress this MIR150 in HEC293 cell lines and confirm the overexpression by checking the transcript level of both 5P and 3P of MIR150. So as you can see that when compared to the control pCDNA3, the level of MIR150 5P and 3Ps are more in overexpressing cell lines. In that same sample, we have checked the transcript level of AGH2, 1, 3.
Conclude.
And we have checked this protein level also. So this is also significantly showing that reduction in the protein level. And also we checked this epigenetic marks. As I told, it is trimethylation. So we checked the trimethylation level of histone 3, which is also it's getting reduced. And also we checked the global DNA methylation state. And also we checked the impact of this MIR150 on 293 cell proliferation by a trypan-blue MTT assay, neutral red assay, which confirmed that cells are cell growth is getting inhibited upon the overexpression of MIR150, and we analyze the cell cycle upon overexpression of MIR150 with control cells pCDNA3. As you see here, the cells are getting arrested in S phase when compared to the control cells. And also we check the cyclins and CDK level. It should confirm that cyclin D1 and 4,6 there is no significant changes, whereas cyclin A2, E2 and cyclin CDK2 it's getting reduced, which confirm that MIR150 induces S phase arrest in HEC293 cells. We performed the same experiments on HeLa cells also, which is also confirmed that MIR150 affect the cell growth by down-regulating this epigenetic regulators. And also we analyzed the cell cycle, which is further confirmed that MIR150 induces the S phase arrest in HeLa cells. Here is the proposed summary model. So MIR150 it directly binds to the DNMT3A, AGH1 and 2, and indirectly binds to DNMT3B, and thereby it is reducing the expression of this epigenetic repressor, which in turn induce the HEC and the HeLa cells S phase arrest, and thereby it is reducing cell growth or proliferation. So with this, we are concluding that MIR150 could act as a biomarker for different cancer cell line, and it can be used as a epigenetic or cancer therapy. Thank you.
Thank you, Murugan. We have an audience question.
Yeah, sure.
So Athira is asking, when you check the expression level for MIR 150,
Yes.
with what you made the relative analysis that you have presented in the graph, how have you estimated the global?
Actually, this MER 150 is just 20 to 22 nucleotides. So what we did, we just added our adapter sequence with the poly-A. So with poly-A, it is increasing the number of transcripts number of size. So with that, we have used our standard adapters, so which we used as a reverse primer, whereas this microRNA binding site we used as a starting forward primer. So with that, we estimated this MER 150.
Yeah, but when you do relative, you have to do with JPDH or something else?
With GapDH we have checked. Sorry, here we used U6.
Okay.
And here we used methyl specific enzyme, that is MXP1 and HPA2. So here we digested our whole genomic DNA with this enzyme, and with this there is one formula is there. So in that we have estimated this global DNA methylation status.
Very good. Excellent work.
Thank you, ma'am.
Yeah. Okay, any further questions? Move on. Thank you, Murugan. Nice work.
Thank you.
Thank you, Mr. Murugan. Our next candidate is Mr. Vignesh Mariappan from CIT RF. Mr. Vignesh, you can start with your presentation.
Yes, we can see your slide. You switch on your microphone. Unmute yourself.
Mr. Vignesh, please unmute yourself.
Hello, ma'am. Can you hear me? Hello?
Yes, please.
Yes, we can hear you. Yeah.
Yes, ma'am. Good evening to everyone. Myself, Vignesh, doing a PhD in CIDRF, Sri Balaji Vidyapith, Pondicherry. My title of the study is Vascular Endothelial Markers, a predictive of plasma leakage in dengue. So coming to the background of my study, dengue virus is a flavivirus, belongs to a Flaviviridae family, is transmitted to human via two female mosquitoes, and it causes a disease called dengue fever. Once the virus is infected, the patient has several symptoms such as fever, vomiting, leukemia, thrombocytopenia, and other severe symptoms like plasma leakage and bleeding. Based on these symptoms, WHO classified the patient into three different types, such as dengue without warning sign, dengue with warning sign, and severe dengue. Beside that, WHO also classified the patient into three different phases, like this febrile phase or acute phase, critical phase or defervescent phase, and recovery phase or convalescent phase. Critical phase is also called as a deference phase because during this stage, some of the non-severe dengue cases, that is dengue with warning sign and without warning sign, it turns into severe dengue patient. So during dengue viral infection, there are two major processes that happens in the human, which is called thrombocytopenia and endothelial damage. Studies have reported that during dengue viral infection, endothelial cells are activated and release their proteins into circulation. In this aspect, we want to study some of this endothelial released proteins such as endoglin and syndecan and its association with plasma leakage. We have chosen endoglin and syndecan because these two molecules are majorly involved in maintaining the vascular integrity. They are also play a major role in vascular integrity, and they have also studied these molecules in sepsis. However, endoglin and syndican, the role of endoglin and syndican in dengue virus is not known. So overall aim of my study is to study the role of vascular endothelial molecules and its association with plasma leakage in dengue infection. My with this, we have two objectives: to estimate the expression level of endoglin and syndican in circulation in severe and non-severe cases compared to other febrile illness using ELISA, and to find the association between endoglin and hematocrit, an indicator for plasma leakage. Coming to the methodology, in this study, we had 47 dengue cases, 10 OFI, and 8 healthy control cases. The sample collection was done at 3 point of times. Day one, it is within the 24 hours of admission. Day two, not a day two, day of defervescence, which is a median of day 4. On day of convalescence, which is a median of day 11. After that, we have separated the serum for all the samples, and we have quantified the levels of endoglin and syndican using ELISA. After this, we have analyzed statistics using SPSS, and following statistical tests were used for the data analysis, and results were expressed in median plus IQR. So coming to the results, at the time of admission, there is no changes in the endoglin in dengue cases when compared to the healthy control. However, during the progression of disease, we could see there is a significant increase in all the dengue groups, such as all dengue cases, dengue without warning sign, dengue with warning sign, and severe dengue cases. Similar results were also found in the expression dynamics of syndican. During the admission, we couldn't see any expression changes in the endoglin. However, during the progression of disease, we could see elevated levels of syndicans in all dengue cases, dengue without warning sign, dengue with warning sign, and severe dengue cases. We assume that the tremendous increase in severe dengue cases is due to host immune response or dengue virus direct infection with endothelial cells. Based on this, we want to study the efficacy of these two molecules in predicting the severity of dengue. So for this, we have used support vector mission, which is a mathematical model, and we got a result that both the molecules with endoglin and syndican, along with the clinical parameters such as platelet, hematocrit, AST, ALT, and other clinical parameters like fevers, we found that these two molecules could predict the severity with 100% sensitivity. In order to see whether these two molecules have any association with hematocrit, since hematocrit is one of the marker for plasma leakage, we want to study the association between endothelial markers and hematocrit, and we found a positive correlation between endoglin and syndican at the day 7, which is a critical phase, in both endoglin and syndican in all dengue cases and severe dengue cases. So coming to the results, during dengue viral infection, several circulating molecules are released by platelet and endothelial cells. In this aspect, we want to study the two major molecules, that is endoglin and syndican, produced during dengue viral infection. Our result shows that a tremendous increase in the levels of endoglin and syndican during the effervescent phase, that is a critical phase. This shows that these two molecules have a mechanistic role in dengue viral progression. Interestingly, we also found a positive correlation between endothelial markers and HCT concentration, that is hematocrit concentration. This indicates that these two molecules may have influence in plasma leakage. Of note, we also showed that using a vector support mission, these two molecules could predict the dengue severity with 100% sensitivity. So coming to the conclusion, the study reports that the serum level of endoglin, that is both serum level of endothelial markers and endoglin and syndican, along with HCT, could predict the plasma leakage with higher precision in dengue infection, and it also used as a prognostic target for the early prediction of dengue disease outcome. However, further studies are needed to ascertain the role of endoglin and syndican in arboviral infection. Thank you. This is some of the reference. And
thank you, Mr. Vignesh. Ma'am, is there any questions?
Vignesh, I had one question. endoglin. So endoglin is a good for prognosis or bad for prognosis? Like it is, how is the outcome if endoglin is high?
Ma'am, it is hello, ma'am. This is good for prognosis, ma'am, because there it's a critical phase is deprivation, that is day 7. It is a critical phase where this level endoglin and syndican are highly expressed, ma'am. So it can be a good prognostic biomarkers for the dengue.
Okay, so the outcome is favorable for the patient or negative?
No, it is favorable for the patient.
It's favorable for the patient.
So based on this, the clinicians can also give a efficient treatment for this.
Oh, in that sense, favorable or biologically favorable? I mean, you are saying it is favorable because it can be treated early. That way you are saying, or does it have a biological significance for being favorable? What are you meaning?
Ma'am, using this marker, if your patient there are two conditions, non-severe cases, which the non-severe cases will go for severe cases suddenly with unknown mechanism. By identifying this marker, we can say that this patient can go for severe, or this patient cannot go for severe. If a patient have these markers like a very high level at day 7, we can say that this patient will go for a severe cases.
Okay. Okay. All right. Okay. Yeah. Thank you, Vignesh. Yeah. Thank you.
Thank you, ma'am.
Thank you, Mr. Vignesh. Let's move on to the next presentation by Dr. Lada I. Shukla from Pondicherry University. Ma'am, Dr. Lada I. Shukla from Pondicherry University.
Actually, I don't see her here, Neha. I just wanted to know is somebody else's name is there for her? Like, somebody else is presenting on a behalf. So no, if nobody is responding, then we can go to the next candidate, P. Kalaivani.
Yes, ma'am. So with that, let's move on to the next candidate is P. Kalaivani from Wells Institute of Science, Technology and Advanced Studies. Miss P. Kalaivani.
Ma'am. Ma'am, can you hear me? Good evening, ma'am.
Yes, we can hear you. You can share your slides and start.
Yes, ma'am. Is my screen visible, ma'am?
Not yet, ma.
Yeah, 1 second, ma'am.
Okay.
Is my screen visible, ma'am?
Yeah, we can see some Airtel bill.
Only bill you can see, ma'am?
Yeah, my dear. You're sharing that. Kalaivani, open your PowerPoint on the desktop and then you click share option and try.
Yes. Yes, ma'am.
Okay. Or Kalaivani will call Aruna Devi now and after that you can present. Is that okay?
Yes. Yes, ma'am.
Okay. Aruna, are you there?
Miss Aruna Devi Selvaraj from Annamalai University.
Yeah, now only she can talk. Aruna?
Yes, ma'am. Yeah, I'm available.
Yes, Aruna. Then you can present now.
Oh, again, Aruna is coming.
Thank you.
Yeah. Because she has to.
I think she can skip the introductory part because she has already done that.
Yeah. She will directly
and then
go
move
to
on to the
discussion.
next slide.
Is the screen visible, ma'am?
No, dear, not it.
For me, it is not visible.
It's not. It is not. Aruna,
Yes, ma'am.
open the file in the desktop and then you click share. Don't share now. Okay? Open the file in the desktop. Keep the file open in the desktop and then go to Zoom and then share your screen and then click on the your file from the click share. What is it?
Or she can send the file to someone of us and
That can also be done. You can share it to my
Yes,
mail.
ma'am. Let me send the file, ma'am.
Okay, can you send it to
But
this?
somebody must be willing for your sake to.
I will do it. I'll do it from here.
Yeah.
You can send it to
Meanwhile,
me.
I think we can ask.
Sure, ma'am. I will send the. Yeah, may I send you the DDTR 2021 conference mail?
Yeah, you can do that. DBMB conference, right? Yeah, do that.
So what happened to Kalaivani?
Yes, ma'am.
Yeah, I don't know what she has not been here. I don't know. Because I didn't follow.
Yes, ma'am,
I
I
was
have
just having my tea, so I didn't follow. I listened to, Sujata ma'am's thing, her work. Yeah, maybe.
Yes, ma'am. I have shared the file.
The DBMB conference?
Yes, ma'am.
1 minute, let me open. Manoma.
Yes, ma'am. It's
You can see now.
yes, ma'am. Thank you.
Aruna, which slide do you want?
Yeah, I think we have listened to the first bit, so you can skip those. Yeah. You can start.
Theoflavin also she explained, right?
Yes.
Yeah, she has actually given a brief introduction.
So we'll go to directly objectives.
Ask her to go for,
Guys,
objectives.
You can start with this, yeah.
Yes, ma'am. Okay. May I start, ma'am?
Yeah, please.
Okay. The effect of the theoflavine was studied in MPTP-induced behavioral impairments in animal models such as akinesia, open field test, catalepsy, and akinesia. The chronic administration of
Methodology slide.
Next.
Yeah, now you can proceed.
Okay. The chronic administration of MPTP diminishes the peripheral and central movement and rearing and grooming activities in the mice. It also decreases the initiation of movement and enhances the impairment of the movement in mice, where the co-administration of the theoflavine significantly alters the behavioral changes in the mice. Next slide, ma'am. Yeah, next. Yeah, this is a grooming, the peripheral and central movement, and the grooming and rearing activity in the mice. And the next slide. Next slide. Yeah, next. Okay. Okay, this is a mechanism of the action of theoflavine against MPTP-induced behavioral impairments, where the theoflavine inhibits the dopamine accumulation in the substantia nigra, and it just inhibits the neurodegeneration, and it induces the dopamine in the striatum, and it alters the behavioral changes and enhances the movement in the mice. Next. The role of theoflavine on the MPTP were identified using these protein expressions: COX-2, interleukin-1 beta, and TNF alpha, interleukin 6, caspase 3, 8, 9, BAX, and BCL2. Next. The chronic administration of MPTP significantly increases the expression of interleukin 1 beta, TNF alpha, interleukin 6 in substantia nigra and striatum, where beta-actin is used as a control in this study. And GFAP and COX-2 expression was increased in the MPTP treated mice using beta-actin as the control. Next. Yeah, this is GFAP and the COX-2 were expressed in the increased in the MPTP treated mice using the beta-actin as the control one. Next one. Okay. Where caspases 3, 8, 9 also increased by the chronic administration of MPTP. And the next slide, MPTP significantly increases the BAX and BCL2, where beta-actin was used as a control. Where the administration of the theoflavine inhibits the inflammatory and apoptotic proteins such as COX-2, caspases, BAX, BCL2, and interleukins, and it protects the neuronal damage and apoptosis of neuronal cells. And I would like to conclude our work. The outcome of this present study explored the multi-pharmacological effects of black tea containing polyphenol theoflavine against Parkinson's disease model were tested in this study. Hence, theoflavine can be considered as a good candidate for therapeutic intervention of Parkinson's disease. However, further studies were warranted before entering into the clinical trials. Yeah, thank you for listening and thank you for support.
Ma'am, is there any questions?
Thank you, Aruna, for the nice presentation finally. And so why do you think the GFAP was increasing in your MPTP model without theoflavine?
Sorry, I didn't get you.
The GFAP was increasing, right? Without
yes, ma'am.
Yeah. So why do you think that was happening?
Due to the astrological action where the apoptotic process, the GFAP was increased in this.
Okay, so is GFAP a marker for apoptosis?
No, no, ma'am. It is used as a in. Yeah, no, ma'am. It is used as a glial fiber acidic protein, ma'am. And the COX-2 was increased there.
No, I was asking about GFAP. Why do you think GFAP was increasing in your MPTP model? Okay, yeah, think on it. All right. The other
Yeah,
thing is which
let
one
me
has
refer,
more?
ma'am.
Yeah, which one has more theaflavin, black tea or green tea?
Yeah, it's in the black tea.
Okay. All right. Yeah. Thank you. Bye.
Yeah, thank you, ma'am.
So we'll ask Kalaimani if she is available. Kalaimani? Kalaimani, are you there?
Yes, ma'am, I'm here, ma'am. Can you hear me, ma'am?
Yes, yes, we can hear you. Will you try to share your screen?
Ma'am, I have sent my PPT to the conference mail ID, ma'am.
Okay,
Please
fine.
tell me,
So,
ma'am.
Kalaimani. Is she sharing her PPTs?
Kalaimani, I've got your mail, but without the attachment.
Ma'am, I have attached and sent you, ma'am.
No, no, it has not come. I've got your mail. Oh, God, 1 minute. It has come.
Lata Shukla ma'am is here. If she is there, I think we can. Kalaimani?
Yes, ma'am.
You don't have the PPTs with you?
Yes, ma'am, I'm having, ma'am. Just a minute, ma'am.
Ma'am, I have.
Yes, ma'am, that's my PPT, ma'am.
Yeah, fine, ma'am. No problem.
What is that, Vistas?
Ma'am, Wales Institute of Science, Technology and Advanced Studies, ma'am.
Okay, okay.
The shortcut of our university name.
Okay, okay.
Shall I proceed, ma'am?
Please.
Yes, ma'am.
Good evening. Good evening to one and all present here. Today I'm going to present about the in vitro antioxidant and anti-obesity studies of ethyl acetate extract of Boria hispida. I am Kalaimani, research scholar, Department of Biochemistry from Vistas. So let me move on to the introduction. Ma'am, next slide, ma'am.
Yes.
Obesity is one of the major threats to global health in this century. So in both developed and developing countries, in 2009 and 2010, 35.7% of US, 35.7% of US, and almost US adults and almost 17% of youth were recorded as obese. So obese leads to a key factor for health issues like hyperlipidemia, hyper blood pressure, type 2 diabetes, cardiovascular disease, reproductive and gastrointestinal cancers, gallstones, fatty liver disease, osteoarthritis and sleep apnea, and also some of form of cancer such as reproductive and gastrointestinal cancer, prostate cancer. So the natural bioactive compounds, especially from plant sources, including spices, have been investigated for the characteristics and health effects. So plants are more potential sources of natural bioactive compounds such as secondary metabolites and antioxidants. Flavonoids and phenolic acids are the most important group of secondary metabolites antioxidant substance capable of scavenging free superoxide radicals, anti-aging, and reducing the risk of cancer. So the Boria hispida, which is being an alternative therapy for diabetes, and also it also has the ability to cure the cancer. In India, decoction of the plant is used for headache, and the seeds as a stimulant, and for the treatment of internal injuries of nerves and kidney. So in Brazil, the infusion of flowers is also used as an antipyretic and anti-allergic. The root as emetic, and the leaves for anti-diarrheal and for treat erysipelas and hemorrhoids. So the plant has various involved in the treatments. The parts of the plant also involved in the treatments, and it also used to treat the bacterial skin infection and also the leprosy. Next slide, ma'am. So now the aims and objectives. We'll move on to the aims and objectives of my plant. So the aim is to study of ethyl acetate extract of Borreria ispida to evaluate the antioxidant and inhibition of enzyme activity in vitro condition. So objectives: to screen the different phytochemical aqueous extract from the leaves of Borreria ispida, to partial characterization of leaves of Borreria ispida by thin layer chromatography, and to evaluate the antioxidant activity of ethyl acetate extract of Borreria ispida based on their ability to scavenge the free radicals. The methods are ABTS radical activity, lipid peroxidate activity, metal chelating activity, superoxide scavenging, and nitric oxide radical scavenging assays. It's also evaluated inhibition of lipase by ethyl acetate extract of Borreria ispida. Next, we'll move on to the materials and methods. So the first, we have to sterilize all the glasswares by heating at high temperature, and the plant collection. The plants as freshly collected, harvested leaves of is collected from the vegetable market in the KK Nagar on June 2017. So plant was authenticated by Dr. S. Sankaranarayanan, Head Department of Medical Botany, Government Siddha College, Arumbakam. And extraction of freshly harvested leaves of Borreria ispida. The leaves was washed through running tap water, and 100 grams of the leaves were crushed using the blender to a paste-like state for 1 minute. So the homogenized sample was first freezed, dried, and also in order to reduce the moisture content of the sample for a more efficient extraction process. The powder was then soaked in N-hexane to defect for 24 hours. It has then soaked in methanol for 72 hours to obtain methanol crude extract, which was concentrated using a rotary evaporator at 40 degrees Celsius. The sticky residues were partitioned with the chloroform to give chloroform soluble fractions. This was evaporated under reduced pressure and dried using an oven to obtain the flavonoid rich fraction by ethyl acetate. Next, we'll move on to the phytochemical screening. Qualitative analysis, quantitative analysis, in vitro antioxidant scavenging. As I have told, ABTS, inhibition of lipid peroxidase, superoxide radicals, nitric oxide radical scavenging activity, metal chelating activity, and pancreatic lipase inhibitory activity. These are the results. The table 1, which shows the phytochemical screening of leaves extract of Borreria ispida. So it shows the positive towards the phytochemical studies. It was done by the basic standard methods. All the alkaloids, flavonoids, tannins, saponins were also present. Only the glycoside shows negative. And then we'll move on to the next table 2. Yield and phenolic flavonoid content aqueous leaves extract of Borreria ispida. So the yield of extract gram per 100 gram of deflated content is 92.53. And the total phenolic content is 88.79, and the total flavonoid content is found to be 61.3. And then the table 3 shows the free radical scavenging ability using ABTS assay of ethyl acetate extract, Borreria ispida. So it was taken in different concentrations like 25 ml per ml, 50, 75, and 100. The EC50 value shown by these is of ABTS is for ethyl extract, it shows 72.26, and ABTS radical scavenging with the standard vitamin C shows 89.63. Next, we'll move on to the inhibition of lipid peroxidation activity of ethyl acetate extract of Borreria ispida. This also with the same different concentration 25, 50, 75, and 100. The ethyl acetate extract shows 93.12, whereas the standard shows the 98.52. In superoxide, it shows 93.12, and with the superoxide vitamin C, it shows to 112.36. For methyl chelating activity of ethyl extract, extract of Borrelia hispida, the ethyl extract of Borrelia hispida shows EC50 value of 96.31, and with the standard vitamin C is 102.36. Nitric oxide scavenging activity, it shows EC50 value 98.23 and standard 110.23. Inhibition pancreatic lipase activity of ethyl acetate extract of Borrelia hispida with different concentration, and finally it shows a EC50 value of 79.63 and standard it shows 84.56. So the discussion, Borrelia hispida has been extensively used in the system of medicine for various conditions, including the decreased blood sugar level. The ethyl acetate extract of my plant are the greatest, shows the great antioxidant activity against ABTS, and it shows the protective capacity from radical hydroxy radical than the vitamin C. The results indicates that the extract contain rich phenolic compounds in the ethyl acetate extract of Borrelia hispida, which plays a protective role from free radical induced damage. So it also inhibits the degree of lipid peroxidation. This inhibition of lipid peroxidation may be either due to the chelation of iron or by trapping of the free radicals. In the present study, the nitric oxide radical activity of the ethyl extract has been detected and compared with the standard ascorbic acid. The ethyl acetate extract of Borrelia hispida exhibited the maximum percent inhibition of 53.8 at a concentration of 100 ml per ml in a concentration dependent manner. So the nitric oxide is a potent pleiotropic mediator of physiological process such as smooth muscle relaxation, neuronal signaling, and inhibition of platelet aggregation, and also regulation of cell-mediated toxicity. So from the data of the present study, which results obtained, maximum percentage of lipase inhibition was shown by ethyl extract of Borrelia hispida. It shows a percentage of 78.92, though it shows a minimum activity of 69.23%. So by conclusion, polyphenols are valuable plant constituents for the scavenging of free radicals because of their phenolic hydroxyl groups. So this together with the obtained results suggests that the amount of polyphenol compounds increases the antioxidant and anti-obesity activity, which also increases. So the present study demonstrates that the ethyl acetate can protect extract of Borrelia hispida, can protect the body from oxidative stress from reactive oxygen species, which may be due to the phytochemicals present in the polyphenols that occur in the plant. So thus this results indicates that the commonly used green leafy of Borrelia hispida significantly inhibit the activity of pancreatic lipase, which can be attributed to the presence of saponins, phenols, flavonoids, or alkaloids, which is comparable to the standard. So these may be used in the nutraceuticals and the food industry. So the additional studies are necessary to develop a method for the fractionation and identification of polyphenols and to determine the most active antioxidant compounds in the Borrelia hispida. So these are the references. Thank you. Thank you, ma'am.
Thank you, Ms. P. Kalayani. Ma'am, is there any questions?
No, nothing from me.
Okay. No questions, I think we can ask the next speaker. Is there anyone else left out? Smita, ma'am.
I don't have any questions.
Soundarya is here. Soundarya. Sorry.
Soundarya, she is not in our list at all.
No, actually she couldn't present in the morning, and so.
Kavya, what about Kavya? Is she here?
Kavya is not here. I'll just make Soundarya co-host, and then if she can present.
Smita, ma'am, has Kavya presented?
Yes, madam. Kavya has presented.
Kavya presented.
Kavya has presented. Everyone has presented in this list.
Except for Soundarya.
Except for Dr. Lata Shukla.
Yeah, Lata Shukla ma'am has not done, right?
No. Soundarya, are you ready?
Soundarya, ma'am?
Yeah. She's from the previous session.
Okay.
It's Soundarya from Bharatiyar University.
Okay. Yeah, she's shared her.
Am I audible, ma'am? Am I audible, ma'am?
You are audible, but you feel quite audible because there's a problem with the mic. There is an echo. Meaning that you have two equipments together. Mobile phone and laptop or something is here in your room.
Am I audible, ma'am?
Cannot hear you right.
Ma'am?
There's a lot of
ma'am.
You remember one girl presented.
Ma'am. Hello, ma'am.
Go ahead.
Okay, ma'am. Good evening. I am Pamela from Bharatiyar University, Biochemistry Department, and I'm here to present present a part of my work. That is pharmacogenic induces our process in A549 lung cancer cell. And coming to the introduction, cancer is one of the major causes of the death in the world. Among them, lung cancer is the leading cause of cancer death. And in the global mortality, it is started to decline, which reflects the decrease in the smoking habit. But in case of India, it is increasing day by day. So the natural compounds are believed to be a promising alternative for many therapeutic remedies. So I'm here selected Nigella sativa, which is most studied medicinal herbs in different healthcare and anti-cancer studies. And the thymoglobulin is a major natural component of Nigella sativa seeds, and it possesses antibacterial, antioxidant, anti-allergic effect. My aim of the present study was to analyze the anti-cancer effect of thymoglobulin on A549 lung cancer cell. And coming to my methods, first I have done the anti-cancer activity of thymoglobulin against that lung cancer cell. I'm doing NTT assay versus cell validity assay. So 10,000 cells per well was incubated and treated with an increasing concentration of thymoglobulin. That is 20, 40, and 60 micromolar. And then media was replaced, and 100 ml of MTT reagent was added and incubated for 4 hours. And after that, 100 ml of TMSO was added to form a violet chromosome crystal, and the violet was calculated. And the next one is apoptosis assay. 1 lakh cells of A49 were seeded and incubated at 24 hours, and then treated with increasing concentration of thymoglobulin, that is 20, 40, and 60 micromolar. And stained using our Muse, Anaxin, and Dexter reagent of 100 ml and the suspension from 100 ml, and incubated for 20 minutes at a dark. And the percentage of apoptosis cell was analyzed. And then cell cycle assay, we did with 10 lakh cells and seeded with the 6LP and treated with the same increasing concentration of thymoglobulin. And also control was kept. The cells are incubated for 20 and 40 hours and centrifuged for 5 minutes at 1500 rpm and washed with PBS. And then cells are fixed with ice cold ethanol 70%, and then after wash, then add 20 ml of new cell cycle reagent, and it was analyzed. Coming to the result, in the MPT assay, concentration decreases with the and concentration increases with that increase in the cell inhibition. From that, I got the activity value of 47 micromolar. And coming to the apoptosis result, it shows that lower right quadrant is viable cells, and the lower right quadrant is early apoptosis, and the upper right quadrant is late apoptosis, as well as upper left is dead cell. So I got the result of 20 micromolar. I got 27.2% of apoptosis, And then at 40, 43.14% and 60, 32.5%. So from that result, I conclude that upper process on 40 micromolar got that 43.14 increase in concentration when compared to control, which has 96.45 lyses. And coming to the cell cycle analysis, thimaquinone in A549 cancer cell, which arrives the cell cycle in G2M phase. The same for 40 micromolar, I got that G2M phase cell arrest and got 56.3% of cells. And this conclude that thimaquinone on A549 cells, which arrest in the G2M phase. When it compared to lysis, G2M phase was lower. And of course, for 20, 40, 60, the 40 concentration got higher number of G2M phase cell arrest. So coming to the conclusion to sum up, thimaquinone proved and is still proving to have a potential anti-cancer effect in several tumor types. And it was proved that thimaquinone induces upper process in A549 lung cancer cell in 40 micromolar in 24 hours and cell cycle arrest in G2M phase. And I got that result in 24 hours efficiently. So that's all, ma'am. Thank you.
Thank you, Ms. Soundarya.
Yes, thank you.
Is there any queries, ma'am?
What way do you use this component for yourself?
Just I have referred several articles, ma'am, and thimaquinone is most effective in that particular seed, ma'am, Nigella sativa seed. So I have chose that thimaquinone.
Ma'am, also any question? Okay, today's the last. Yes, ma'am, thank you.
No questions from me. Yeah.
Any other questions? Asmita, ma'am?
No questions from me. Fine.
Okay, then the candidate can, I mean, she can.
Thank you, ma'am.
Yeah.
Dr. Lada, I should.
Yeah, there is an echo. I don't know. I have to check.
Soundarya unmuted her mic. It's okay. I just muted it. So now
Rukmani?
Yes.
Ma'am, today so we are.
Yes, ma'am. I think we have come to the end of the session.
Yeah, I think
so
if
we can
Dr. is not coming, right?
Ma'am, Asmita, ma'am, can you tell about the speaker, I mean presenters today who are presented for our session alone?
Yeah, the names you want. You want the names?
I mean, so it is.
Soundarya Kalevani from the.
Was the first one.
Oh, the first one is who? First one I'll tell you.
First one
is not there. One student has presented, then Kavya, Daisy.
Yeah, all of them have presented.
Except Lata Shukla, ma'am, na?
Yeah, exactly. And one additional Soundarya. So total.
Yeah. Okay, so then we'll pass on the scoring card to Rukmani, ma'am.
Yeah.
Finally, give to her when you are planning to give.
It is like, ma'am, it is the mark for all the 3 sessions together, ma'am. It's not for the single session. So we will just consolidate everything after the 3rd oral session. And we
Madam, I'm emailing. I'm emailing to you, Dr. Sudha, Dr. Lata, and Dr. Tulasi. Okay.
Yes, we will decide that later. Yes, ma'am. Fine. So with this, we have come to the end of the session, and I thank all the participants and of course the speakers and the technical team especially for your kind cooperation. We will meet tomorrow exactly at 8 AM for next interesting sessions. Thank you very much, all of you. See you all tomorrow at 8 AM.
8 AM, madam?
Yes, because their time doesn't match, so 8 AM it's like, I think their time is 10 o'clock in the night and they are doing for us here.
Okay, sure.
So it's 8 AM tomorrow morning.
Thank you.
Thank you, all of you. See you tomorrow.
Yeah.
Thank you, ma'am. Thank you.
Neha, thank you. Is she here or she has already left?
Yes, ma'am. Thank you, ma'am.
Yeah, Neha, thank you.
Manju.
So, by then tomorrow we'll meet by 8 o'clock.
Yes, yes,
Evening
ma'am.
8
At 8 a.m.
o'clock.
Definitely.
Yeah.